as I mentioned yesterday, we will cover the entire syllabus of physics. Yesterday was first part, another 4 sessions, we will cover the entire syllabus of physics. And for radiobiology on genetics also, we will cover the entire syllabus in 4 sort of. So 5 days will be physics on Saturdays, and 5 Sundays will be cancer biology and radiobiology. So people may ask why on Sunday, because unfortunately most of the doctors, if you keep it on a weekday, they will not be able to join. So that's the only time when medical doctors can really attend these things. So that's the reason. This is what we covered yesterday, and today in the first 3 talks, I will talk about genes, gene structure and mutation, a brief overview, then something about Mendelian genetics and human genetics, and then finally about the cancer genes, oncogenes and tumor suppressor genes, and I'll explain a phenomenon known as Knudson hypothesis, which is very relevant, a little bit about genes and related to cancer. So this is a diagram which shows we always see 2 things. In between we don't see. We see either chromosomes, okay, chromatid pair, and DNA. But in between that also there are a lot of things which are happening in terms of the structure, how it is, all the genetic material is packed. As you see the double helix, which we all know, then it is complex with histones. So there are different histones. Okay, H1, H2A, H2B, H3, and H4 histones. So as you see, it forms a nucleosome. So this is wrapped around octamer of H2A, H2B, H3, H4, and then H1 sort of binds it together, and then sorry, there's a noise disturbance. I don't know whether it is coming on YouTube or it is on a local system only. And then a nucleosome and this thing, a chromatosome, and the chromatosome folds up to form fibers, and then they coil, and then ultimately we are into a chromatid and a pair of chromosomes that we have. This is how the human genetic material is packed all in the nucleus. This is what I'm talking of the nuclear DNA. So gene, when we sometimes just synonymous gene, DNA same, but gene is only a specific sequence of DNA when we call it a gene. So it's a functional unit of inheritance. This is a functional unit of inheritance. So it has a very specific structure and sequence of nucleotide in the DNA. And the gene, I'm talking of the protein coding genes. Not all genes code for a protein. Some code for an mRNA or microRNA. So it directs the synthesis of a specific polypeptide chain. Protein is nothing but a polypeptide. And these proteins then have biological function, and that's what we are interested in the biological function. Mind you, only a small fraction of DNA is protein coding genes. We don't know exactly how much, maybe just 1% or 2% of the DNA is protein coding. Rest.
Interrupt once. After this, shall we shift to the other side? The menu.
So, vast majority of the human genome is non-coding DNA, which includes some functional elements. So basically, they're non-coding genes and regulatory sequence also. And rest is not expected to have a function. So you could ask, what is like, there are some pseudogenes or duplications, or some of these things which are Murphy's law. So even though only 2% of the human genome codes for the protein coding genes, still the remaining part is also important. That's why we do genome analysis, mostly for research. And we also do studies like genome-wide association studies. So sometimes variants, it could be a single nucleotide variant,
Auditorium is ready. Mini talk auditorium is ready. 5 minutes if anybody wants. We can shift there.
Just one second. Sorry, there's some noise in the audio we're trying to see. Let us figure it. So you should see now whether you're getting the noise. Okay. I don't know whether people who are in the auditorium, there's some noise is coming. People who are on YouTube, whether they are also hearing this noise. Okay, that's fine. So on YouTube it is clear, so we'll continue. Sorry about this. So any genetic material, which either it is protein coding or non-protein coding, but if it regulates gene expression, it could be altered. Basically, any DNA sequence in the entire genome could be altered. And if it is in the protein coding gene, or it is in a regulatory part of the genome, it could have a phenotypic effect. Okay, and we will talk about the phenotypic effect later. So what when we call it gene? So gene has, broadly we understand gene has exon and introns, but it is more than just exons and introns, and that we need to understand the moment we start looking at genetic alterations. So when we analyze genes, we don't just look at the exons or don't just look at the flanking introns. Sometimes the variants come up in other parts, and we have to understand what the rest of the genome does for us to make sense out of it. So in this nice diagram, what they have shown is that, so this is a central dogma of molecular biology. So DNA after transcription, pre-mRNA, and then post-transcription modification, then translation of mature RNA into a protein. And then protein also has post-translational modification. That is also important, but this is just a broad thing. So what you see in the red part is the coding sequence. Okay. In between the coding sequence is the intron. The coding sequence is exons, in between the introns. But you look at the regulatory sequences, so they control the expression of the protein coding region. What will be expressed as a protein, that depends on the regulatory sequences. So these are the on 5 prime. So it starts from the other 5 to 3 it goes. So 5 prime UTR is untranslated region. It is not translated. And in the end, there is 3 prime UTR or untranslated region. Then there are promoters, enhancers, silencers, and terminators. Okay, so this is the at the DNA level, and then you would see finally the mature transcript, mature mRNA, and it is translated into gets to a protein, and the protein is the functional unit, biological functional unit, but what creates a biological effect. That's why we're interested. And anything which happens in the gene regulation or in the sequence of the DNA, whether it is mostly exonic or sometimes intronic, we will explain it later. It could have an impact on the protein that is made out of this DNA, and that will have an impact. So promoters and enhancers, they regulate transcription into pre-mRNA, which is modified, and the 5' cap and the poly A tail is then sort of removed. So all of us wonder why different codes give rise to different amino acids. Same amino acid is coded by different codes, and there is a reason. So this is I'm saying. So this is the first base, this is the second base, and this is the third base. We know triplet codons, so they are triplets. And if you look at each total 20 amino acids are there, but these combinations are many more. I think they are total 64. So there is degeneracy or redundancy. So that is multiple 3 base pair codons combinations for each amino acid. So for each amino acid, like phenylalanine, you see these are things for leucine, so many possible combination, isoleucine, methionine, valine, etc. And each of these amino acids, so they have properties of these amino acids. These are biochemical properties. So it could be polar or non-polar, it could be acidic or basic. So these are the various properties of amino acid. And for it to happen, so there's a start codon, which is AUG, methionine, and there's some other uncommon things which could help start, and the stop codon, so UGA or called TGA or UA or UAG or TAA or TAG. So these are the stop codons. So this is how the whole nucleotide that we have said, when they make a protein or amino acid, this is how the different combination of nucleotide bases make triplet codons and they code for an amino acid. Now talking about, so now talk about the abnormalities which could happen. So at the broadest level, there could be a abnormality of the chromosomes. So chromosomes, we know we're talking of humans. There are 22 pair of chromosomes, 23 pair, 22 are autosomes, 1 to 22. And then we have a pair of sex chromosomes. So XX in females and XY in males. So the abnormalities that can happen at the chromosomal level, it could be a numerical abnormality or aneuploidy. That means one chromosome is missing or one is extra. Actually, addition is more common, extra chromosome, and it can result, and we're talking of only live birth, because when a miscarriage happens, we do not know what happens in that unless until the fetus was, the product of conception was examined and looking at karyotyping. So it can result in miscarriage, congenital defect, or cause syndromic features. Sometimes the syndromic features are mild or manifest later. Only an experienced geneticist or pediatrician can, say that, okay, this is I'm suspecting this underlying chromosomal defect. And the most common chromosomal anomaly is trisomy 21 or Down syndrome. Okay, so one additional this thing. But also in addition to this, of trisomy of chromosome 18 or chromosome 13, then we have 40X, 45X Turner syndrome, 47XXY, which is Klinefelter syndrome, and several others. So these are the chromosomal, basically number. One chromosome is added or one chromosome is missing, generally what is there. If many chromosomes are happening like that, it will not be compatible with life and it will result in miscarriage. Okay? Sometimes that is a cause for recurrent abortions. Then the structural abnormality, that means a part of the chromosome has broken. It may or may not have joined. If it has joined, it may join in the incorrect set. So it could be balanced or unbalanced. Balance means the entire chromosome material is there, except that it has gone somewhere else and something else has come somewhere else. Or it could be unbalanced. Sometimes when the two broken ends of chromosomes join together, it forms a ring chromosome. And some of these translocations, especially the balanced translocations, are tolerated, but they can also cause a problem. And others cause disease. So they cause disease because some of the genes which were contained in that segment of chromosome are missing, or they are duplicated, or it creates a aberrant fusion protein. So when it goes sit somewhere else, it is now sitting next to a, in the class sometimes teacher used to say, these two boys will not sit together. They will create a lot of nuisance. So one will sit on the left side, other will be made to sit on the right side. I don't know the class teachers do that anymore or not, but it used to be nowadays. So this is what the fusion protein requires, and they're very famous examples. I will not tell. Some of you should know what are the examples of this, and they're targeted therapeutic. Any answer? BCRA. Very good. BCRA. So zooming in from chromosomes to the other part. So there could be a structural variation. Just part of the chromosome is deleted or duplicated or inversion. It just flips around, or translocation. Okay, that we already mentioned about translocation. Then S and V. S stands for single, N for nucleotide, V is variation. So it could be a single nucleotide variation, and the single nucleotide variation would have various effects on the protein which is it codes, and I will talk about it later. This is just to the overall picture. And there could be indels, like in the lower diagram, you see that you could have deletion or addition of few bases, 1, 2, 3, 5, 10. So these are short indels, but there's a large, then we call it different thing. It's a large deletion or large duplication. So what really happens? What is the impact of this? Because as I mentioned that ultimately after the translation, the protein is formed. Protein has a structure, not just a function, but also has a structure. Okay? It has sheets and groups, and primary, secondary, and it has to be like that because that is a functional molecule. If that is changed, then it may have an impact on the functioning of the protein. Okay? So that is important. So what happens that if a large part of the gene, protein coding gene, is deleted or duplicated or inserted, so almost always it will have a major impact on the protein which it codes for. And because the protein will be misshapen or half or completely messed up shape, it will not be able to function normally. And then depend what is the normal function of that protein or the gene, there will be a phenotype. If it is a important protein, which is a very crucial vital role, there could be human disease, it could be a severe human disease. Cancer is one of those human diseases. But you could have just 1 or 2 or 4 or 5 or 7 bases which are deleted or inserted. So in this, not always, whenever it is green, that means it may not have a deleterious effect biologically. So if it is in frame, suppose only 3 bases are deleted from the start of a codon, then all you will have, suppose BRCA1 has 1863 amino acids. It will have 1862 amino acids. Maybe somewhere here, one amino acid is missing, but it has not really affected the structure and function of the protein. So some in-frame deletions, that means just 1 or 2 amino acids are missing or added, but rest to the left and to the right, upstream, downstream, everything is same. It may or may not have. May have an effect, but it may not have. You might think that, well, it is deleted, 3 bases are deleted, why it should not have it? So that's the reason. But then you have a frame shift. Suppose 2 bases are deleted, the entire reading frame is changed. So what was, becoming, it was suppose alanine, serine, alanine, threonine, methionine. Suddenly it will change, and at some stage down the line, it will introduce a stop codon. Okay. So what we were showing that in the diagram, the stop codon will be introduced. The moment the stop codon is introduced, the further translation will stop, and you will have a truncated protein. So only maybe this much of the protein is made, and most of these proteins, almost the entire length of protein has importance. Sometimes you do not understand the importance of what is only some of the domains of the proteins are solved. Not all domains of even important proteins are solved in terms of structurally as well as functionally. The issue comes when you have a single nucleotide variation. So single nucleotide variation, as we saw that degeneracy or redundancy, silent means although you have a change in a nucleotide, but SNV, but the amino acid is same. And because amino acid is same, generally it will not have any impact. And these are the ones we call single nucleotide polymorphism. That means sometimes they could have a weak modifier effect. They could slightly affect the expression of the gene. Okay? And rarely they could actually be introduced. Suppose it in the last base of the exon. Okay? then or the first base of the exon. So they may affect the splicing. So by affecting the splicing of this, it causes an aberrant splicing, and that is how it could be. So for vast majority of silent or synonymous changes, one nucleotide change, they are tolerated. They may have no consequence, or they may have very weak, impact, and they are called as single nucleotide polymorphism. A lot of association studies were done with this to see whether if this SNP is there, whether the disease risk is slightly increased. But these are not like Mendelian genetics. They are not studied in terms of Mendelian genetics, but for the association studies. Now, this sense, when it codes for a different amino acid, a new amino acid has come instead of alanine, now you have threonine. So the amino acid has changed. So whenever there is a missense change, most of these remain as SNPs, single nucleotide polymorphisms without any impact. But some of these are pathogenic. And I will talk later, especially in the oncogenes, when we'll talk about it. Oncogenic mutations, they are mostly single nucleotide variants. So just one missense change because they are gain of function mutations. I will talk about it later. Okay. And rest of these are what we call variants of uncertain significance. That means there is a variant, but the significance of the variant is as yet unknown. Okay. Then there are splicing variants. So splicing is a very complex subject. In the subsequent courses, we will have a full lecture on splicing. I will just mention that when there is aberrant splicing, that may result in either retention of an intron or retention or deletion of intron or exon. So that is they will always be pathogenic because the protein impact on the protein would be quite substantial. So they will be pathogenic. And then nonsense. Again, I will show some examples. When the amino acid changes to a stop codon, whenever there is a little bit truncated protein, and most of these proteins, especially tumor suppressor genes or other genes also, the moment the protein is truncated, you would expect a severe phenotypic effect or disease related to it. So when we talk about germline variant, when we do sequencing, whether it is next-gen sequencing or Sanger sequencing, we only look at the nucleotide sequence. And we know what is the reference genome, we know what in humans it should be, there are different versions of that. So if in that person whom we have tested, in that sample, if at one point instead of T, we find it is C, type to C, or there are one additional base is there, or two bases are missing, or something is different, then we have to interpret, just note that this is a change, and then the interpretation, what this change would lead to. Like a radiologist, when they look at a chest X-ray, they will see, okay, I'm seeing a shadow which normally I don't see in most of the chest X-ray, 99%. Then it is an interpretation, what this shadow could mean. Okay, shadow could be an artifact, shadow could be old healed infection, shadow could be a cancer, shadow could be tuberculosis, active tuberculosis, so many things. So same way, here also we like to see. So pathogenic or likely pathogenic are the ones which are expected to have a major phenotypic effect and disease. And we're talking of genes which are important in biological process. So if that gene is very important biological process, and if it is a pathogenic variant in that, there will be a genetic disease associated with that particular gene. And most of such genes are already characterized, we know what happens. And so these are the mutations. So how will we know the type of mutations? As we previously said, again, emphasizing they would either truncating mutations. Truncating mutations are either stop codon, which is nonsense, or it could be frame shift mutations, or these are the two main truncating mutations. Or if it is a splice site mutation variant, a large genomic rearrangement, or it is a missense, but it has been characterized to be a pathogenic or likely pathogenic. So these will be this thing. Then, because they are harmful, they are having a major phenotypic effect, they should be very rare in the healthy population genomic database. So there are many databases where they would have done sequencing, DNA sequencing of tens of thousands or maybe hundreds of thousands of individuals. So we know, so those are the people who don't have any disease when they give the blood sample. Some of them may develop later. And they see that at this particular position in in most of the people, this at this point it is A. And very rarely in 0.00001% people, it could be instead of A, it could be C. Okay, so we know this is a reference, so the wild type and the variant allele. And it will never be or extremely rarely as a homozygous in a healthy population database. Because sometimes if it is a missense change, it may have a weak phenotypic effect, but homozygous would have a much more severe effect, okay, as we will see later, much more severe effect. So a pathogenic or likely pathogenic variant is you do not expect it to be in a homozygous state in a healthy person, healthy adult, if, depending on the gene also. We know in which some genes or homozygous pathogenic variants are not known, but in some genes, homozygous pathogenic variant, we know it creates a very severe phenotypic effect in childhood or something else, and those individuals would never be sampled to come into the healthy population. So, just the converse of that, what is a benign or likely benign? So, it's a type of mutation. I've written mutation, actually, we should call variant, because mutation has a connotation of harmful or deleterious. So, what we describe as variant. Okay? So, the variant has a deep intronic. So, it is 100 bases away from the intron exon boundary, or it is in the exonic region, but it is synonymous, or a missense change, which is a known single nucleotide polymorphism. So these will be benign or likely benign. So they may be there in 1 out of 1,000 people, 1 out of 3,000 people, 1 out of 100 people, 2 out of 100 people. So we see that, we know that, yeah, it happens sometimes. No action on that is left. And they are not so rare, okay? Or they may occur as a homozygous state in healthy population. So they could be there because it happens. So because it has hardly any effect, it may be there in 1 out. If it is present in 1 out of 100 people in a heterozygous state, it may be present in 1 out of 2,000 people in homozygous state without causing any phenotypic effect, and it doesn't. Or else they are a missense change, and people have done functional studies or family studies and have shown that they are not pathogenic, they are not segregating along with the disease or some other ways. So whenever you are unable, when you find a variant, you are unable to fit them into one of these categories, pathogenic, likely pathogenic, or benign or likely benign, but it is a variant and it is a rare variant, you call it a variant of uncertain significance. So any rare variant, or it's a novel variant, no one has described it before, if it is not fitting into this criteria, is a variant of uncertain significance. Okay? So when you will read a report or when you do a research, when you find a variant, you first have to go. So there are many databases for the healthy population database. Genome AD is one of the best ones, and the latest version also has. So it also gives for different populations like South Asian, Latin American, Caucasians, okay, Africans, Chinese. So in different, so a polymorphism, which suppose is present in 1 out of 200 people in a particular community or caste or group in India. In the healthy population database, which is predominantly Caucasian, it may look as a very rare 1 in a million, because that population where it is a polymorphism is not represented in that healthy population database. So to allow that this thing, they have subdivided into different ethnic groups. So you look at, basically it is just a stretch of nucleotides, DNA, that's a stretch of nucleotides. And you have to make sense, suppose here instead of a C, it has become T. Okay, that is what you find. So explaining it in a different way, you know that DNA, genes, and proteins have biological function, and which is normal growth and physiology. That's the function of proteins, okay, structure and function. Now, because of either spontaneously or due to a mutagen or a carcinogen, you have an altered DNA sequence. So when you have an altered DNA sequence, you have a gene with genetic alteration. So it's a genetic alteration. You call it mutation or a variation, whatever it is, it is there. Now, what will be the impact? So the fact is that only some, which I explained earlier, only some of the gene variants alter the structure or shape of the coded protein. Others have no impact either on the structure or the shape of the coded protein. Okay, so that is one thing. and only some protein alteration. So protein is altered, but their biological function is not affected. So that also happens. There are many examples of that. And some changes in the only some of the changes in biological function of a protein will result in a disease, congenital anomaly, or evolution. So, like I've talked about the redundancy or degeneracy in codons, similarly, proteins also, sometimes for the same function, the human body, eukaryotic and prokaryotic, there are multiple proteins or a family of proteins which will do that function. So even of my GST T1, glucagon S transferase T1 is not really fully functional, a null phenotype, I've got other GST. So CYP, GSTs, etc. So many of these. So because of the change in the nucleotide sequence may or may not affect the gene, the protein structure or function, and may or may not have the effect on biological function. Change in biological function may or may not have a disease. That's why we get many variants of uncertain significance. So there's a variant, but the clinical significance. Okay, what we are interested in as doctors or even in the lab when you do research, you want to know whether if this change will affect the cellular function or some other important parameter. So there are organizations like IR, International Institute for Research in Cancer, ACMG, American College of Medical Genetics. So they have divided when they find a variant, with this I'm describing for germline variants. So not pathogenic or not likely not pathogenic, that means benign or likely benign. Then uncertain, likely pathogenic or definitely pathogenic. So this is the based on some concept of posterior probability. If the probability is very low, extremely low, then it is benign or likely benign. When it is very high, almost certain, then it is pathogenic or likely pathogenic. And whenever it is, and this is used for advising the patient and the family. I'm not getting into the details, which we will cover later when we talk about clinical cancer genetics in the subsequent courses. So there are ways in which you can make an effort to characterize a VUS. So a comprehensive one is a bioinformatics. So there are many tools which can tell what or predict, not tell, but predict what will happen. So AGV, then so look at the evolutionary conservation. So it is believed that any protein or any amino acid in a protein, if it is really very important, it will be conserved through evolution. So at that particular place, it will always be, say, serine or methionine. So that is important. Then functional studies, co-segregation. If suppose there are 5 people in a family who have a particular disease, genetic disease, and on that comprehensive analysis, you find one variant and in one of the tested person, and that variant is as yet PUS, no one really knows whether it is pathogenic or not. If that was the cause of the genetic disease condition in that family, you would expect it to be present in all those who have that genetic condition. So co-segregation. Okay? And then co-occurrence, if suppose a particular variant do not know, but it has occurred many times with a pathogenic variant in a gene which explains the disease in that person, then we know this is a bystander, it has nothing to do with the disease. Okay, that is one. And then you do family studies and syndrome characteristics. I will show later when we talk about the VUS what to do. And then you see how the presentation is there. The gene in which you have found the VUS is the person's, the age and tumor characteristics are very typical characteristic to that gene. Like for an example, gene called P53, TP53. There's some tumors which are very characteristic to it. So they are otherwise very rare, like a type of brain tumor known as choroid plexus carcinoma. If in that patient who has a choroid plexus carcinoma, on comprehensive testing, we do not find anything else but a very rare variant in the TP53 gene, which as of now cannot be classified as likely pathogenic or pathogenic. So you strongly suspect, because in this person, there's a very classical P53 associated rare cancer, and I'm finding this variant. So there are multiple ways of looking at it. I'll just show some of the things that we do in the lab. Like this was functional characterization. So in our cohort, we had found many variants. Some of these were novel variants like C1697Y and S1722P. So one of our my co-student, I was co-guide for her, Lumbini. She did various functional characterization. So looking at loss of beta galactosidase activity. So this is an indication that this variant is affecting the function of the protein. So functional characterization. Then you could do in a different way, like this is another student of mine who had done it earlier. So we found BRCA1 intervening sequence, IVS plus 1 GYA and plus 3 GYA. So both of them were, as you have seen, this is the transcript analysis. So on the cDNA, you see that they resulted in retention of 153 bases in the mRNA transcript. So that ultimately it was aberrant splicing, and the protein which was formed would not be same because you had so much of extra, amino acids in that, and it will cause disease. So to end this first talk, what we know about human genome, a lot, and as an assignment, I want you all to do some interesting reading about the Human Genome Project milestones. I had first kept a slide on Human Genome Project. I said, well, if I just tell you, there's no fun. So you all should read, okay? Anyway, while on your way back, you can just read about Human Genome Project. What was the milestone? How it was conceptualized? Who were the main players? What came out? Okay? So I think Human Genome Project, although you say completed, I don't think it will ever be completed. At least next 50 years, it will not be completed. And the reason is because we have 3 billion base pairs in a haploid human genome. 3 billion, imagine. Okay? And only less than 2% of human genome is for protein coding genes. Number of protein coding gene is anywhere from 20 to 22,000, and this actually turned out to be much less than we had previously expected. People thought they thought there are 100,000 human genes, then 50,000 human genes, then I remember it was 30,000 human genes. Now we are sort of saying maybe around 20 to 22,000 human genes. Okay? So because some other primitive organisms have equal number or even higher number of genes. So we humans function with just 22,000 genes. Now, this is another question for you. I'm not telling you the answer. The number of proteins. So I've talked about, let us say genes are 20,000. How many proteins humans have? Same number, because genes make code for protein, okay? Or less or more than the genes. So this you all have to figure out for yourself, and this will be part of the. Then, although we talk about DNA variation, gene variation in terms of disease, but it is a hallmarks of all life forms. Without that variation, evolution would not have been possible. For evolution to happen, you have to have the underlying variation, otherwise evolution cannot happen. And as of now, only some DNA variations have been understood in terms of their functional and or phenotypic effect. And that means many of these gene variants we do not yet know what will be the functional impact, leave alone the phenotypic impact. So for phenotypic impact, you have to have studied a human being which has that. Sometimes it is not possible. But even functionally, they have not been characterized. But on the other hand, we have several well-characterized gene variants, and they show a strong association or causation with a particular trait, not always a disease. Traits also have, when we say human, these physical traits, physiological traits, they have this or a disease. So this is what about the first lecture. Okay, so assignment for you all, in addition to understanding everything, is read about Human Genome Project, the milestones, and when maybe some of you will be involved in having a better version of the human genome, what we have, and the proteins. Here now I think everything is settled down now. You will continue here. Yes. Okay, so now I will switch next half and I'll let by 15 minutes I will cover because all this. Human genetics and Mendelian genetics. So I will again not go into the details of this thing, but briefly I will talk about the important aspects of human genetics and Mendelian genetics. So Gregor Mendel, I used to call him Gregory Mendel, I realize it is Gregor. He was a monk and a botanist in Austria. And I think he had a lot of time or interest. More than time, I think it is an interest. We always find time what we want to do that. He, in his greenhouse, he sort of did crossbreeding of pea plants. Okay? And in the process, so what he was looking at various characteristics, the seed, whether they were round or wrinkled, cotyledons, the flower color, the pod form of the pod or the color of the pod, where is the inflorescence, okay, tall or short. So this is through these studies what he found out, and he used to just note down what happened. What is the progeny when we have this and you cross. Okay? So what he showed that inheritance was not blending as Darwin had thought. Darwin had thought that it blends. If these two things happen, then like you mix two colors, it will just blend. It was not blending, it was particular. So through this, he came out with a law of dominance that some alleles, that time genes were not known, some alleles are dominant. If that even one, so that two alleles, even if one comes, it will manifest in form of the trait. So he was looking at traits. Okay, it substitutes for a disease also, but for the trait, even one law of dominance. Then law of segregation. So the two alleles, only one of the two alleles from the parent will pass on to the progeny. Both will not pass. Okay. Then law of independent assortment. That means if suppose there are 5 or 6 alleles, each one of them would have an equal chance of being passed on to the progeny. It's not that if one goes, all 4 will come with that. Of course, there are exceptions to this because maybe both the alleles are on same chromosomes or nearby to each other. So they will go together, not all. So these are by and large correct. And based on this, what he had found, so essentially dominant and recessive. Single variant, both variants. Single allele, both allele. Okay. And he published this in 1866, but people did not, who would pay attention to some monk doing some pea experiments and publishing in some journal. But after almost 30, 40 years, this was rediscovered. And that was the basis of modern genetics. People started understanding. Genes were not known at that point of time, but just through this simple observation. Okay, so this is the power of doing experiment and observation and applying your mind to it. I'm sure when he started doing this, he would have never thought that this is the inference he will come up, okay, which will change genetics completely. Even now in human, this thing we call it Mendelian genetics. Okay. He worked on that, so this is so profound. Ultimately, we are also life forms made up of DNA and genes. So Mendel had looked at these very distinct characteristics, phenotypic features, what we call phenotypic features. In humans also sometimes you have very characteristic features. So on the left side you have one albino. This is an autosomal recessive condition in which there is mutation, homozygous mutation, a compound heterozygous mutation in a class of gene, which helps in the development of pigment. So because of the mutation, they lack pigment completely in the entire body, hair, skin, eye, everywhere. On the other hand, there is a very rare condition, again autosomal recessive, known as xeroderma pigmentosa. It is in the XP genes, which is there. Again, it is a homozygous or a compound heterozygous mutation, and that results in UV hypersensitivity. So phenotype is the appearance of observable nature. So what I've shown here is a very stark black and white. Okay, but the fact is, and this is happening because of effect of gene-environment interaction. So what is the gene-environment interaction? On the left side, you can see that genetically his skin and hair have no color, but because there's no pigment, so more of the UV rays are able to penetrate, and that's why he has developed a cancer over the year. Here, he is trying to put more, his skin is making more pigment, but still as a sunscreen, but whatever little UV rays goes in, they are sufficient to cause DNA damage, which is not being repaired, and it causes cancer. Okay. So unlike this black and white, actually there are a lot of gray shades. So experienced doctors, they will be able to make out and tell, well, this is a different, distinct phenotype. So there are always shades of phenotypic effect. So broadly speaking, what is a genotype? Genotype is the genetic constitution of an individual. You can apply it to a single locus or all the loci collectively. So that's the genotype of that person. And when you apply it to the entire genetic constitution, then we say it is a genome. So genome of this person. So genome would have so many variants of a person. Would have so many. So coming back to Mendel. So Mendel, as he said, there are dominant and recessive alleles. So autosomal, when we talk of autosomal, you're only looking at autosomal. So they are dominant and recessive. In recessive conditions, both the alleles of that gene have to be dysfunctional or mutated for the effect to come. Whereas in dominant, even if the other allele is normal, that variant or the mutation, only one copy of that gene is sufficient to cause that phenotypic effect for the disease. Then Mendelian sex link. So sex link is interesting. Again, that I am not showing here. This is for something as a homework that you all have to read. So when you read and understand sex linked disorders, then suddenly a lot of things about genetics you will understand. Okay? In males, in females, who can remain a carrier without having a phenotypic effect, who can be affected, why only males could be affected, who can be carrier, all those things. So X-linked, most of these are X-linked disorders. And then non-Mendelian. So non-Mendelian is makes a little confusing. So what is non-Mendelian, but how it can be inherited? We only studied that for inheritance it would be Mendelian. So I will briefly describe some of the things. So what happens is, we were thinking of a high penetrance allele. That means if that gene has a mutation, there's a very high lifetime risk of expressing it in form of a disease. Okay. So like P53, BRCA, retinoblastoma, RET, I'm talking of cancer-related genes, but in some other conditions also there are diseases which have these genes. Then there are intermediate penetrance gene. That means it can increase the risk of the disease several fold. It could be as high as 30, 40, but not 100% or 95%. Then a lot of variants have either low or uncertain penetrance. Could be an allele, like the xenobiotic metabolizing enzyme, CYP, GST, SALT, genes like some of the DNA repair genes, NBN, MRE11. So the lifetime risk of a specific disease or cancer may increase, but may increase by only 1.5 times or twofold. So if otherwise the risk of, say, lung cancer was 4%, it may increase to 6%. Or if breast cancer risk was again, say, 5%, it may increase to 8%. So it is increasing it, but just a little. And most of these are complex, chronic, multifactorial, polygenic disease. So complex, that means they have not been understood. Chronic, they develop over a period of time, not just, like an acute infection. Multifactorial, that it is not just that gene, but, gene environment interaction, and polygenic. Like for an example, diabetes. Diabetes, if you have diabetes, the family, the others also have a higher risk, but it is not a monogenic Mendelian disorder that you can just, So there are multiple, so many mental health, diseases, psychiatric diseases, diabetes, hypertension, cardiac diseases, most of the common sporadic cancers, these are polygenic multifactorial. And they have not been well studied because of various reasons. So some of you would know this very famous this thing, Michelangelo David. This is in a museum in Florence. I visited it, but I've only seen this. But if suppose this man was going in, sometimes, and he was obese like this, and as a researcher, what would you look at? You would look at genes or the environment or the gene-environment interaction. Okay? If you focus only at genes, you will never learn anything. Because his genes, he was a Roman man, so his genes are same. But now his genes will be expressing only the shape of his nose or the colors of his eyes or his hairs, because that is only purely genetic. It has nothing with environment. So your color of eyes will not change with environment. but rest of it is because of his environment. So if you have to study, you have to study the environment. That means his lifestyle, his habits, his food habits, his sedentary habits, and also his genes. And the genes will be not high penetrance genes, but the genes which have a weak modifier effect, what I called a polygenic. So obesity is also mostly a polygenic. Obesity, diabetes, hypertension, etc. So non-traditional inheritance, I will cover, just show some examples. I kept more slides, but I will skip those slides. So the inheritance, we know it is being inherited, but it does not falling into the chromosomal, a single gene or multifactorial categories. So these are like could be digenic, mitochondrial, imprinting disorders, and genetic anticipation. I will show one or two examples of each one of these. So there is a disease known as retinitis pigmentosa. So the retina degenerates and results in blindness. So in this, there are two genes. So RDS1 and ROM1. Okay. So when there's a mutation in both copies of this gene, heterozygous mutation, then it manifests. Otherwise, it does not manifest like this. In the shaded ROM plus minus, that means one is normal wild type, other is mutant and RDS. So this is affected and this person is affected. And whenever only one of the gene has the variant, mutant variant, and the other is normal, it doesn't. So it is two genes, digenic. So it was monogenic, then there is polygenic, in between there is digenic. Okay. Then mitochondrial. So what is mitochondria? Mitochondria you get only from the mother. So mitochondrial genome is much smaller compared to, we say, the human nuclear genome is haploid genome is 3 billion bases. And mitochondrial genome is very small, and you get only from the mother in the ova. Sperm doesn't carry any mitochondrial genome. So different mitochondria. So when it forms, you might find that in this one ova there was 80% mutant, 50% mutant, or 20% mutant. So it may change in each of this, and as a result, so in this one you might find that the child which is born with this could have a severe disease, and in this one may have no disease or with mild disease. So in mitochondrial inheritance, the manifestation varies. It is coming from the mother side, and the manifestation, the spectrum is really highly variable. Okay, like this is mitochondrial and Leber's optic atrophy. I'm just showing this. So it will only come from the mother side. That is important to know that. Like this, the father, male with mitochondrial disease gene, a man cannot pass it on to his sons or daughters. Whereas females, if this lady has this mutation, she has passed on to some of her children. And of course, we are not showing here the severity of the phenotype, but that is how it will be there. Now, imprinting disorders, very briefly. So these are syndromes. So they are result of altered expression of the genes that are imprinted. So this complex mechanism is little complex. I will not go into more details, and I will just give some examples of some of the disease and what happens in that. So there are three genetic mechanisms and an epigenetic mechanism of imprinting. So one genetic mechanism is that there is a deletion or duplication of a chromosomal region which has the imprinted gene. I'll show the example. Or a mutation in the genes that are imprinted, or in there, there's a region called as imprinting control region, center. Or there is uniparental disomy. That part of that gene or DNA is coming from the same parent, both copies, either maternal or paternal inheritance. So when we were talking about Mendel's law of segregation, so here sometimes there are always exceptions to those laws. That is this thing. And then there is an epigenetic mechanism. That means the DNA sequence is not changed, but because of the methylation on the maternal or paternal allele, that expression is changed. So there is a group of syndromes known as Prader-Willi and Angelman's syndrome. So there is a PWS gene and there's the AS gene. So in this, what happens is deletion of the paternal, this region is deleted. 15q11.13 is deleted on the father side, and when there is a maternal side, then the Angelman in the AS gene, then there is a deletion of the maternal. Okay. And there could be a uniparental disomy, maternal uniparental disomy, that is both has come from the mother side, and there is a mutation in the imprinting center region mutation. So again, don't have time to go into the details of it. As a result, you could have Angelman syndrome. Angelman syndrome is maternally inherited. Our mothers are angels, so Angelman syndrome, or a Prader-Willi syndrome, which is from the paternal side. So this is a phenotype. They could have severe phenotypic mental retardation, speech abnormality, dysmorphic facial features, microcephaly, and several other neurological problems, including epilepsy. Behavioral phenotype, they could be hyperactive, sleeping problems. They, in general, are very happy kind of people, but an inappropriate laughter. So there are various things of Angelman. Then there's a Beckwith-Wiedemann syndrome, and it's a disorder for imprinting, growth, dysregulation, and tumorigenesis. So they have, large macro glossia, big tongue, viscera could be, enlarged, some of the visceral organs, and they could also develop some cancer. I think. So briefly, the last syndrome I will talk about, which causes non-traditional inheritance, is the trinucleotide repeat expansion. So normally it was thought that between generations, whatever variant is there, it will not change, it will be stable. So then this was found that the trinucleotide repeats, there is intergenerational expansion of the number of repeats. And this was found, so the different repeats, CGG, CAG, GAA, CTG. So Fragile X syndrome, Huntington's disease, Friedreich's ataxia, myotonic dystrophy. So these are all neurodegenerative disorders. Okay, so number of repeat, this thing is increased. So as a result, what would happen that these are the number of repeats, more than 200. So in the grandfather may have a very mild phenotype, but as generation increases, second generation, third generation, fourth generation, number of repeats increase, and you may have a more severe phenotype. So the grandfather may have just had a premature cataract, but second generation, by the time they come for the third generation, the child is crippled or paralyzed by the time the child is 12 years old. So that is because the number of the repeats has increased. This is just an overview of the symbols. When you talk of genetics, you have to draw pedigree just to show. A lot of things you understand by looking at pedigree. Males and females, square and round. When the sex is not known, and whenever someone is affected by a condition, you shade it. Disease is you cross it. Pregnancy, and whenever there's a particular proband or consultant, you put an arrow to show that this is a person with whom we are starting getting all the information. You have first evaluated this person. So these are lines. So this is a line of marriage. This is a separation, and this double line means consanguineous. That means they're married between their blood relatives, then consanguineous. And in the recessive traits, it becomes important because when there's a consanguinity, there's a high probability both the parents are carrying the same variant in the child. So you say child is born out of consanguineous marriage. So this is the line of relationship, line of descent. This is for individual, and this is the shipship. That means they're ships. Twins, a lot of studies that happen. So twins could be dizygotic or monozygotic. Monozygotic, they are identical. And there are twin studies that are used to look at nature versus nurture. So monozygotic twins, the genetic constitution is exactly the same, but they may have grown separately. There was a time when they were given for adoption to different parents. So they have, in different states, in different homes, they have grown up. So their environment is completely different, but the genotype. So when you look at what age, what diseases they get, it will tell you how much of it is genetic and how much of it is because of the upbringing or lifestyle. So so kindred is related individuals in a family. Genotype, what we have said, genetic constitution, Phenotype is the observable characteristics of an individual. I showed you some examples. Penetrance, I mentioned again, just to recap it, is the frequency or probability of appearance of a trait or a disease in individuals who carry a variant or a mutation. And genetic anticipation, I showed in the example of prime repeat disorders, is a worsening of a disease phenotype, like earlier age of onset over succeeding generations. And we have said who is a proband. Okay, so I think I've covered up for the time which we had lost. Now this will be the last lecture. Just let me bring some more. This will be my last for in 2 days. Any questions in between you all have? No? How many people are watching online? Just want to give a little 1 minute rest. Today is Sunday. Anyway, by the end of the day, more than 1,000 people would have watched. So far we had not talked much about cancer. I mean, obviously, when we talk about genes, somewhere reference had come to cancer. Now I will talk specifically about cancer and genes and the two classes of genes known as oncogenes, tumor suppressor genes, and a very important mechanism known as Knudson's 2-8 hypothesis. So we know cancer, it's a dysregulation of the most fundamental biological process. You know, when the most fundamental biological process are dysregulated, like cell replication or its control, then one of the outcomes that could happen is cancer, because you know it is so. And the factors which govern the replication of cell and the integrity of the cellular phenotype, they can either suppress or accelerate development of cancer or the progression of cancer. So when we talk of genes, we know that what is the function of these genes? Most of these genes, so they've got gatekeeper genes, housekeeper genes, etc. etc. So all cancers are a genetic disease. So this is a statement I'm making. Since genetic alterations, either in the genes or chromosomes, are a hallmark of a cancer cell. No one has ever seen a cancer cell where all the genes and chromosomes are absolutely normal. The defect is only in cell membrane or some other organelle. It cannot happen. Okay, or some physiological change. So because for cancer to become autonomous and behave like a cancer, it has to, gain or many other properties or have lost some inhibition or controls which otherwise a normal cell, lives and grows under those controls. Okay? So, but when we say genetic disorder, most of times it is somatic genetic disorder. That means it is in the cell, cancer cell, and it will be in the daughter cell of that cancer cell. So any genetic change will be always passed on. The question is whether it is passed on to the daughter cell or passed on to the daughter. And when it is there, a germline genetic disorder, because every cell of the body has, then in addition to that mutation in the cell being passed to the daughter cell will also be passed on to the daughter or may be passed on, depending on the laws of Mendelian law of inheritance. So very important to distinguish between somatic and germline. Okay, sometimes we just say mutation, we're describing it. We're talking about basically a variant in the tumor, somatic, but we talk as if it is a hereditary this thing. So soma means body or tissue, somatic, psychosomatic illness. Okay, so what is it? And germline, germ through which something germinates, and not germs which cause disease, germinates. So germline, which is inherited past. And then the tumor suppressor genes are oncogenes. So this is a distinction you have to have in the mind. So cancer is it's a process, malignant transformation. It's a multi-step process. It just doesn't happen just like that. Like if you fall immediately, you may have a fracture. Okay, but cancer doesn't happen like that. So, in which there are additional genetic lesions at each step, and it's a cumulative effect of all of these genetic alterations which gives rise to malignant phenotype of the cell. And these, once a cell is transformed, it undergoes clonal expansion, and the subsequent genetic alteration results in it also causes metastasis or treatment resistance phenotype. I'm putting it in a very simplistic manner for you to remember. So first, through multi-step process, multiple alterations transform it, and it becomes a cancer cell, transformed cells. Then that transformed cell undergoes clonal expansion, and then after clonal expansion, when it forms a tumor, it undergoes further changes, and then it could gain a metastatic potential or resistance to therapy, etc. So now the two classes of genes, oncogenes and proto-oncogenes. So basically, oncogene is a proto-oncogene in which has been activated. So without activation, it is proto-oncogene. After activation, it becomes oncogene. And how does this activation happen? Is because it's the mutation in a specific domain. Okay, it's not just spread anywhere. So we say codon 12 mutation or codon 18 mutation when you look at a different gene. And it's like an accelerator of a car that is stuck in full throttle. So suppose you were driving and you press the accelerator. Okay, you are from here going to Pune on Ghats. You press the accelerator gets stuck. No matter how good a driver you are, you will not be able to control the car. Okay, and the result will be an accident. So that is what oncogene. So it becomes a driver. The cell goes mad. When an oncogenic mutation happens, it sort of just goes into overdrive. Okay? But an oncogenic mutation is also easy to target. Like, the only defect in your car was that the accelerator was stuck. If somehow where the place where the accelerator gets stuck, you block it, you don't let it get stuck, then the effect is immediately lost. Okay? Then your brakes you'll be able to use your brakes and the steering control and everything. So that is the basis of targeted therapy, where you find it's a targetable. Okay, so whether there is a kinase mutation or whatever different kind of. Then the tumor suppressor genes. So they are like natural, the suppressor. Okay, they are like natural brakes of the car, which fail when the both alleles are made dysfunctional due to inactivating mutation. So many times we forget that in a car we have two brakes. I hope during emergency time we will remember that there is another handbrake also. Otherwise, why would they make this inconvenient thing? It's always inconvenient to have a handbrake. So same way there are two brakes. So both alleles are important. And I'll show it with an example. So the Knudson's two-hit hypothesis. So Knudson actually he wanted to be a mathematician, but then he became a doctor, and then a pediatrician and a scientist, and finally he's into public health. So way back in 1971, before the RB gene was known, he published. You look at people, so mind is always more powerful than anything else. Not Gregor Mendel published all that in 1866, okay, before genes were known. And after 40 years, it was shown that what he said was very profound. And Knudson published in 1971, before that time genes was known, but RB1 gene was not known. The disease was known. So what he explained in his two-hit hypothesis of cancer causation, so it explained the relationship between hereditary and sporadic cancers. He proposed a mechanism of penetrance in hereditary cancers. And he predicted that there would be tumor suppressor genes. I mean, he did not say it like that, but his work basically is telling you that there will be a tumor suppressor gene, there will be two alleles, and both have to be, So retinoblastoma, for those of you who are from a non-medical background, is a childhood disease. Unfortunately, it affects the eye. And if not treated in time, child might lose the eye vision, and also sometimes lose their life if it spreads before it is treated adequately. So it has been long known that there are two types of retinoblastoma, either familial or non-familial. Non-familial ones we call sporadic. So about 2/3 of retinoblastomas are sporadic and 1/3 are familial. So the sporadic ones are generally almost always unilateral, they affect only one. Whereas, and this what you're seeing, this picture here is the white eye reflex, because the tumor is growing. Okay, so it's a cat eye reflex. Normally on a photograph also you would see that looking at the child. So the hereditary retinoblastoma, which is about say 1/3 of the case, 1/4 or 1/3 of all retinoblastoma case, and in majority of the cases they are bilateral. So what Knudson did, what he published in 1971, is he's just simply plotted the age at which they get cancer, present of cases not yet diagnosed with at age indicated. So at birth, none of them had retinoblastoma. Okay, and these are the ages he has written for the bilateral cases, and these are the cases he has written for the unilateral cases of retinoblastoma. So, and you look at it, he has, because he had a mathematical bent of mind, he had plot on a semi-log. Okay, it is not a so it is a log linear plot. So this is linear and this is log 2, 4, 6, 8. So comes like a straight line. The bilateral one comes like a straight line. And later on in the day, when you will see radiobiology, you will see many places that they are showing this depending on the type of cell kill. Okay, single hit or double hit. You will see this kind of curve, straight line, or the other one, which is a shoulder, and then later on it is a straight line. Okay. And he just basically looked at 25 cases of unilateral, 23 cases of bilateral retinoblastoma, just plotted this, and what he found was so profound. So he proposed that for this, for this line to happen, this shape of the curve, it has to have two hits. And for this, single hit is there. So as in radiobiology, you will see later for single hit and double. So in sort of you explain it, in one of the book chapters, I've drawn this diagram. So hereditary form, if you can see that little cross I've made on one copy of the RB gene or a chromosome. That time it was not known also there's an RB gene, but knew it is inherited, that means through genes. The first hit is inherited in all cells at birth, because your germline, you are born with every cell of the body would have the first one copy of the RB gene would have the mutation. Whereas in the sporadic one, because it is not supposed to be hereditary, they will be all normal. Then second hit would happen. So second hit in this hereditary form, it could happen in any cell of the body, because every cell of the body has that RB gene mutation. In any cell, you just have mutation, the probabilistic event. Any cell of the body has this thing, it will cause, so it has happened. Whereas in this, the second, here the first hit has happened. Okay, so the chronology. The first hit has happened in one or few cells of an organ. And then the second hit happens in the sporadic forms in one cell, and then there's a clonal proliferation of one transformed cell. And because the probability of this is very less, so you will have one unilateral cancer at a later age in life. Whereas here, already there were few cells had a second hit, so you would have clonal proliferation of multiple foci of transformed cells whenever there's a double hit. And so what will happen? As these are, so clonal proliferation of several transformed cells, so that's why it could be multicentric, more than one tumor in the eye, could be bilateral, both eyes could be affected, or multiple primaries, eye cancer and then maybe sarcoma or something else. And because it has got the lead time, this thing, so it will occur at a younger age. And now we know that because they are driven by a particular genetic pathway that one particular gene was causing, so you might find organ specificity, like in this case, preference for the eye tumor and sometimes sarcomas. They could have specific immunohistochemistry features or molecular markers. Or in some of the genes, they could be a syndromic feature. So through this study of retinoblastoma, sporadic and hereditary classes of retinoblastoma, plotting the ages and finding out that there will be one hit or two hits. So he basically explained all the features of a tumor suppressor gene. So this I have put together so proto-oncogenes and tumor suppressor genes. So allele affected is one in proto-oncogene. It's like a stuck accelerator, only one accelerator in car and gets stuck. The effect of the mutation is enhanced, and it is also called as activating mutation or gain in function mutation or dominant mutation. Okay, just one thing is enough. Whereas in the tumor suppressor gene, both copies have to be mutated, like we showed in the Knudson hypothesis. the effect is reduced. That means if it suppose it was the braking action was there, so the braking action is lost. Right? It was there, and it is also known as inactivating or loss of function. I have also written recessive in bracket because when we talk of human genetics or laws of inheritance, recessive means both parents should be carriers. And if the child is born with both copies mutated, then only it manifests. That is what we understand recessive, what it is. But here it is recessive at the cellular level. At the cellular level, when one copy is mutated, it doesn't sort of cause that. But when the second copy is also mutated, and that is true for many cancers. Second copy in the tumor suppressor gene is not lost by inherited mutation, like in this one. So the second copy could be lost by many things. Okay? Deletion of the other chromosome or some other mechanism or epigenetic silencing or some other features, it sort of inactivates the other copy. Okay? So that is why it looks like it is called recessive, but in inheritance terms, it is dominant. So like retinoblastoma. Okay? Both copies are required to mutate for the tumor to form. But inheritance of one copy, mutated copy is sufficient to cause the disease as an autosomal dominant, because anyone born with even one mutated copy has such a very high lifetime risk that one or other copy of the gene in some cell will be mutated. Okay? So if you are born with one mutated copy, you have a 80 to 90% lifetime risk of having mutation in the second copy and development. So at cellular level, it is recessive, but in terms of Mendelian inheritance, it is dominant. That is true for BRCA gene, that is true for mismatch repair genes, all those tumor suppressors. Okay? So earlier in the exams, when you write about RB gene, so they would say it is autosomal recessive. It is not autosomal recessive. It is an autosomal dominant disorder. But at cellular level, now that we know how genes function, at cellular level, it is a recessive. Okay, at the cellular level. So I have just again shown it for you to remember. So this is a car simulator when you learn driving. Okay. So oncogene is like an accelerator stuck in full throttle. And tumor suppressor is like foot brake and hand brake. Sorry, the arrow has moved somewhere. Okay. So that is it. And whenever this happens, plus there are many other controls also in the car. This just shows the multi-step colonic, the carcinogenesis process. Vogelstein is a very famous scientist. He came up with this model in using colon cancer. So like APC gene. So he studied many patients who had a rare syndrome known as familial adenomatous polyposis syndrome, FAP. There's a gene which causes this syndrome known as adenomatous polyposis coli gene, APC gene. So like a normal colonic epithelium, so you have a loss of function mutation in APC, inactivation of beta-catenin activation, and you form an early adenoma pre-cancer. Then you have a activating mutation in KRAS or BRAF. Then you have an intermediate adenoma. Then, so this is tumor suppressor gene, oncogene, then SMAD4 or TGF inactivation, loss of function mutation, late adenoma. Then P53 loss of function mutation, then carcinoma, and then some other mutation, it causes metastasis. So this is just to don't take each and everything seriously in terms of genes, because this model is not entirely correct. Okay? We have studied in our lab, one of my PhD students, but broadly as a conceptual level, it is good to understand that at each step from pre-malignant to malignant to metastatic or treatment resistant, there are genetic alterations which are adding in different genes. And it is said that normally 1 or 2 activating mutation in an oncogene and 3 or 4 inactivating mutations in a tumor suppressor genes are required for a normal cell to become a cancer cell. This is a generalization. It could be more, it could be less, but that's what it is. So I'm just showing some examples now. So this is a recap of the first talk that I gave. So the mechanism of activation of proto-oncogenes or inactivation of tumor suppressor genes. So it could be point mutations, missense could be P53. Of course, oncogenes like red, it is generally point mutation, but even P53 BRCA, you could have missense. frame shift mutation, I'm showing an example here. So it's in this, if you see that there's a deletion of 2 bases after that. So it looks like you've got a bad chromatogram, but actually it's the reading frame is shift. So in one copy, it is normal. The other copy, it is running 2 bases ahead. Okay, and that introduces a stop codon later. Then nonsense mutation. So this one nonsense. So this is a missense, like in the red proto-oncogene at one place, it is causing this cysteine is changed to another amino acid. And this is a cysteine-rich domain of the red proto-oncogene. It is the extranuclear domain. It is very important. Then you could have translocation, BCR-able, gene amplification, like HER2 new. If it is amplified, again, it is an oncogene, or you could have a deletion. I'm just showing the chromatogram. So sometimes you'll see what happens. So this is one of our cases of last year. So all it is happening, you can see single nucleotide variant. At this place, there is a, instead of G, there is a T. Okay. So wild type, you see there is a G here. So here you see two peaks, one underneath the other. That means in one copy, it is G. In another copy, it is T. And as a result, what is happening? It is introducing a stop codon. Okay. So just nothing has happened, just one nucleotide has changed. And as a result, it so happened the change happened at a place, and the type of change was such that it introduced a stop codon. And because it's stop codon, it will, so it's a glycine at 484th amino acid, ter means terminate. Okay. It terminates. So as a result, you would not have a full length BRCA1 protein. And as we explained in the earlier lecture, it will be pathogenic, and you will have a BRCA1 associated cancer risk, which we will cover later. Now, this one I am showing another BRCA1 exon. This is a, so normal, this is a chromatogram. Chromatogram is basically different colors, different nucleotides are depicted in different colors. Okay. So G is in green, A is in green color. So here there is again deletion of 2 bases, 68, 69. At 2 bases A, G are deleted. As a result, the reading frame is shifted. And when you say P dot GLU at glutamine after 23 codons further down the line amino acid, it will introduce a stop codon. So you will not have a full length BRCA1 protein. So when we do a genetic testing, so how do we use all this study? I'm just giving an overview of this. We'll have a separate in next sessions or next month or so. Is when do we testing and how we define whether it is of clinical relevance or it is only for research. So we know that 5 to 50%, some of the cancers even 50%. I gave an example of retinoblastoma, almost 25% inherited. I gave an example of a rare brain tumor, almost 50% are hereditary. So they would have a genetic alteration in the germline, which is inherited. Or many of them would have only in the tissues, which is somatic, and that can be tested and found out. So this could explain why they developed a particular cancer at a relatively young age for that cancer type. Their future cancer risks and surveillance for second primary, if they are cured of the first cancer, are they at risk for some other cancers? If their first degree or other relatives, they are also at inherited risk because they have inherited the same mutation. And now increasingly based on the finding genetic alteration, whether a particular type of drugs, like say platinum agents, work better for mutations in BRCA gene and some other genes. Targeted therapy, immunotherapy, patients who have mutations in mismatch repair genes, their tumors are hyper-mutated. They have more mutations, more neoantigens, and they respond better to the immunotherapy called immune checkpoint blocker. Then targeted therapy, there's an approach known as synthetic lethality. So in that, like those who have mutation in BRCA1, BRCA2, or other homologous recombination repair pathway genes, their tumors are more sensitive to PARP inhibitors. Okay, so before the testing, counseling process is important. And just very briefly, I'll tell what happens in the counseling process. There's some 3 steps. Pre-test counseling, you inform, you take the family history, you do the phenotyping, you do a counseling, why you are doing this, whether genetic testing is indicated, how it could help, and you inform them of the various options of testing, facilitate decision making, find out who all in the family could be at risk in case you find a mutation, and obtain the informed consent. Then genetic test is not just one test. It's not like just checking for your blood glucose, one test. Okay. So genetic test is a broad term. Within that, whether it is germline testing, whether it is somatic testing, whether it is one gene, whether it is 20 genes, whether it is all the exome, whether it is the entire exome or only 3,000-4,000 genes of relevant what we call clinical exome, or a genome, or a microarray. So there are various kinds of things. So depending on what you are likely to expecting to do that, and use in a lab technique and this thing where you will get good results, reliable results. And finally, most important, when you find someone is carrying a mutation, then post-test counseling in which you disclose what you have found, provide support, it could be psychosocial support or logistics, and help them make a plan for the next step. But all this would happen, this is for the doctors, because only if some doctor has suspected inherited predisposition and refers for genetic counseling or testing. Okay? If you only don't suspect, patient will, of course, some patient directly ask, well, can I do BRCA testing? But that doesn't always happen. Again, for the doctors, syndromic diagnosis or differential diagnosis will be made only by the doctors. Patients never go to the lab or PhD researchers directly. Okay? And they will first go to the doctor thinking that my doctor who's treating me will know all this. They will suspect when he or she has to suspect, and will ask for genetic test. And one of the things we make is a differential diagnosis whenever we sit there. So the differential diagnosis will be based on the type of cancer, age at which that cancer was diagnosed, whether it is unilateral, bilateral, or multiple cancer. So all this is, remember, Knudson's fluid hypothesis. So hereditary retinoblastoma at a younger age, more often bilateral and more likely to have multiple cancers. Same is true for any form of hereditary cancer, which will tend to occur at a younger age, more likely to be bilateral or. Then sometimes more and more the pathologists are alerting. There are particular features, histological or immunohistochemical features, which will alert about the possibility of an underlying gene mutation. Like in mismatch repair gene mutation, MLH1, MSH2, if there is a germline mutation, they could have on immunohistochemistry, there will be loss of expression of these proteins, what we call MMR deficient, loss of expression, or MSI high. So finding that not only it could help their treatment, I said immunotherapy, other family members could be tested, and a lot of research is happening in terms of prevention. You all must have heard of the drug aspirin. Aspirin is not just a painkiller or anti-inflammatory. People who have had heart condition or other condition, in them, aspirin is given to them to reduce the risk. Aspirin has a lot of biological effect. Same way, it is also being tested as a chemopreventive agent. So in people who have a mutation in the mismatch repair gene, what we call Lynch syndrome, in them, aspirin studies have shown that it can reduce the risk of them developing new cancers or subsequent cancers, both colorectal and other cancers. So, and the phenotypic features are not always in the tumor. So they could have polyps, skin, macules, eye, and cyst. So whenever we talk of genetic testing, it is important to talk about the ethical, legal, and social implications. So just forget about genetic testing. People can figure out in this family, this disease is running. Okay. And cancer, there's stigma associated with cancer. So there are a lot of things. So there's a myth. I also thought that in India, it's one large family, everyone shares everything. That is a complete myth. Each family and each person is unique. When you counsel them, talk to them, then you realize, well, this person, if there were two sisters, they may be thinking very differently just because they're two sisters. There may be two very good friends. Their thought process may be different. Just because you're a good friend doesn't mean that you'll be thinking alike. So then they have some common issues, some unique issues. So when they're concerned about their children's future, about their daughter's marriage, which is very common, sometimes because of this in the family, in the family with the siblings, and their concerns. We have cost of genetic testing is high. Some of you maybe would help to reduce the cost of genetic testing. Then counseling has to be timely and culturally sensitive. Okay. You have to know how that, what is the belief system of that person, what is the family structure, what is the family relationship. So it should not harm them. It is very important. Okay? And of course, you require medical financial. So these are the principles of genetic counseling. I'm just introducing this because otherwise genetic counseling will not come as part of any of your core curriculum or this thing. I'm just introducing this because you all deal with patients or deal with samples with that. So the principles are autonomy, empathy, beneficence, non-maleficence, non-directiveness, informed decision, confidentiality, privacy, and fairness. And the way medicine is practiced, unfortunately, especially in busy hospital, more often than not, one of these principles we will not adhere to, and they have consequences. It may not happen immediately, it may not happen for the doctor, but for the patient or the family. So I talked about the VUS earlier in this thing, but remember that VUS, you have to see whether it is in somatic or germline. If you have done the NGS of the tumor, if you find a VUS, you did it only to find out whether you can give a particular drug or not. On the VUS, you will not bother about it for it. But if it is in the germline, it is not just about that person's treatment. It is whether that person is at risk for other cancers, whether the person's daughter or a brother or a son or mother also have, who may, because, and if I have a VUS variant, my first degree relatives have 50-50 chance of having the same. So then you see whether it is in a gene which is related to cancer type and the phenotype, whether the gene is actionable or not. So like I'll give an example of VUS in a gene which has a classical phenotype. Like in this case, very commonly we find a variant in a APC gene in a patient with some other cancer other than colorectal cancer or papillary thyroid or hepatobiliary cancer. And because APC has a lot of repetitive regions, so it is prone to have these errors. And if you find in that person and that person or the family, no one has a FAP related, APC related this thing, we know, well, it is in a gene which otherwise has a classical phenotype and this person is not, you will ignore it. Whereas other case which we had, I'm showing a real case, a gene which has a classical phenotype. So this person had adenocarcinoma, stomach and papillary thyroid cancer. And we were suggesting, the pathologist had sort of this thing, he was suggesting another syndrome known as Peutz-Jeghers syndrome, in which the mutation is STK11 because of the polyps and the way the polyps look hematomatous, which is jumbled up tissue. But what we found that there was a in-frame deletion. Remember, I was talking about various things. So in-frame deletion, that means only one of the two codons were removed. And this was a novel not reported anywhere. It may or may not have been pathogenic. We would have called it otherwise VUS. But this person had a classical phenotypic features of Cowden syndrome. He had macrocephaly, his head was larger than normal. Normal head circumference is around 55, 56. Okay, so his was 62. And he said that he works in a school as a peon. Hats don't fit his head. And he said, my elder son also has this problem. So he has a macrocephaly. He had facial hematomas, skin trichomomas, and oral mucosal papules. So he had a classical phenotype. And we were finding a VUS in the gene which is associated with that. So bingo. So I don't need any other information. It's a very rare or novel variant. And it has been found in a person who has a classical phenotypic feature. So it becomes very easy. But these are the black and white cases, but most of the time we struggle and we are unable to. So what I'm saying is that, and because again, doctors, so we find this lady who had triple primary cancer, ovarian cancer, endometrial cancer, then rectal adenocarcinoma, and it also showed loss of MSH2, MSH6 expression. These are two of the MMR gene proteins, and she had testing done outside, and in a very good lab, and it was negative. So twice it has been done. So what then we did, we found a mutation. So why it was missed, just to show that you can see poly A region. Okay, AAA. Whenever there's a repetitive region, this is a chromatogram. Normally earlier you see that different colors, just remember continuous A. So whenever this kind of thing happens in the sequencing, you might miss something. So this I am putting this example both for the clinicians as well as for the researchers who work in the lab, that you have to suspect. I would strongly suspect there is an underlying genetic basis why this person has developed multiple cancers, okay, and why on tumor it is showing loss of expression of these two genes. Genetic test is normal, so you repeat the testing by a different method, and there is a reason. So it is not that it was bad technique. The region of the gene was such which is prone to that. So as technology improves, we are able to detect those things. So my last slide is that we know that there is a genotype and phenotype. So phenotype could be risk of cancer and for a particular site histology of cancer, age at onset in which they develop, or other phenotypic features. Genotype is site and the type of variant that we find that. And many times even now it is a challenge to decipher the biological basis of a phenotype. So you have a phenotype, you do not know why this phenotype is happening. Okay? Why this person developed cancer at the age of 18, which otherwise normally people get in their 50s and 60s. Or what is the phenotypic effect of a genotype, that we have found this genotype in a person, but we are able to figure out what is the phenotypic effect. So in terms of research, a new approaches for learning is that, look at different humans are just one of the animals. Okay? Don't have to put. So if you look at same genes are shared across all animals. So if man, mice, Drosophila, zebrafish, if you do comparative, reverse comparative genomics, a variant you find in human. Suppose if I find this variant in the APC gene, you will be surprised that there is an APC main model for even zebrafish. Even zebrafish, they will get polyposis like humans. Okay? So through this process in genetics, we are, although different species, but we are one living world. So in genetics, we try to correlate with all life forms. Okay? The genomes of all life forms and try to see what is the phenotype in different things, even in terms of bioinformatic analysis, what is evolutionary conserved. So it's a lot of genetics is still not known. The genome variant, what is the phenotypic effect is still not known. A lot of phenotypes are still not explained the underlying genetic basis. It's a distinct phenotype. It has to have a genetic basis. It is not just because of environmental exposure. Okay? So, but it is not understood. Hopefully, in years to come, we will know a lot. And the studies will not be just on humans, but also through comparative studies through various life. So this is what I use for counseling. This is summarizing everything from DNA to proteins. And when there is a family history or early onset cancer, bilateral or multiple cancers, basically Knudson's this thing, or some phenotypic feature, you do counseling, then do genetic testing. And in testing, whether you find a mutation, don't find a mutation or a VOS. And if you find a mutation and it's a monogenic disorder we're talking about, then in terms of treatment, either because of the gene-specific treatment, chemotherapy, target therapy, immune therapy, or for screening, early detection with scans, MRIs, copies, blood tests, and for prevention. So either chemo prevention with medicines or with surgery. So thank you very much. So we now start with the radiobiology module. Professor Jain Goda, he is the professor of radiation oncology and also is a clinician scientist. He runs a radiobiology labs and is a very passionate radiobiology teacher. So Jain, please.
Thank you, sir, for the introduction, and very good morning to all of you. People are still coming in, I think. Maybe I'll just start it.
Yeah, please start.
Yeah, because otherwise it's going to get delayed. So yes, I'm Jain. And yesterday, Dr. Sareen had taken a class on physics and radiation therapy, but that was physics. Now, radiation therapy, when it enters the biological system, it produces changes. And that's exactly what we are going to talk about today and maybe thereafter in future classes. And this is what is called as radiation biology. So I'm going to talk about the basic concepts of radiation biology as it pertains to clinical radiation therapy. Now, To start with, let's talk about the milestones in radiation treatment. As you all know, Professor Sareen must have discussed about it yesterday itself in 1895. Röntgen actually discovered X-rays. And so these milestones can be actually divided into 4 phases. The phase 1, where with Röntgen's discovery, we started the use of X-rays in the clinical settings. But also there was some misuse of this radiation, especially with the dropping of the atomic bomb in Japan. And then slowly over a period of time, because of these misuse of radiation, safety norms came into vogue, and lots of organizations actually developed various safety norms for the use of radiation therapy on humans. And then over a period of time, with further discoveries, slowly and steadily, the role of non-oncological uses of radiation reduced. However, it did not completely go off. And then ultimately, we are in a phase now, that is the phase 4, where the use of radiation therapy has actually evolved a lot. And now what we actually practice in clinical setting is the modern radiotherapy in the form of right from where we have now evolved from 2D radiotherapy to 3D conformal radiation, and now we are using intensity modulated radiation treatment, and also other things like flash radiotherapy, high LET radiations like protons and carbon ions. So all these changes actually have evolved over a period of time, and all these changes have come out of a biological understanding of the radiation. And when we talk about radiation, it is nothing but ionizing radiation. So what are the learning objectives for this particular lecture? So we are going to understand the importance of radiation biology. We will introduce some terminologies, understand the historical background in the field of radiation biology, and also understand the evolution in radiation biology in the modern era. So why do we need to study radiation biology? So since we are giving radiation treatment to cancer patient, what is the main aim? We have to maximize the benefit of radiation treatment in this patient. And in order to maximize this benefit, I have introduced this term, which is called as a therapeutic ratio or a therapeutic gain. The first terminology has now come to you, therapeutic ratio. What exactly does that mean? You're going to know about it later on. It just means that we are going to give radiation treatment so that it is maximally beneficial to a patient in terms of curing the patient from the cancer, and at the same time, reducing the complication rates. So this is what is called as a therapeutic ratio. It is the ratio of tumor control to the ratio of normal tissue toxicity. Main aim is to maximize tumor control and to minimize the toxicity. And we also sometimes interchange it with what we call it as therapeutic gain. Next, so first terminology I've already introduced here. Then we need to understand the other terminologies, and also need to understand what are the exogenous factors and the endogenous factors that will determine the radiation response to the tumors and normal tissues. So, okay, now when we are talking about radiation treatment, I might interchange it with ionizing radiation also. Okay, so ionizing radiation, when we give it, it not only basically damages the tumor or kills the tumor cells, but also may have effect on the surrounding normal cells. Okay, so when we are talking about radiation therapy, we always try to balance it between the tumors and the normal cells. Okay, so here we are going to discuss a few factors that will determine the response of what ionizing radiation does on tumors as well as certainly normal tissues. And as I said, we are still evolving. Our techniques have improved in the field of cancer in the field of cancer radiotherapy. As a result of this, as a biologist also, we have to be in par with the radiation oncologist. So we also have to evolve in the field of cancer biology so as to understand the biology behind these evolved radiation techniques, right? I've spoken about protons, I've spoken about carbons, so we need to understand the biology of that also. So we have also evolved in terms of radiation biology. So what is the role of radiation biology in clinical radiotherapy? So the contributions can be at different levels. One, to provide a conceptual basis for radiotherapy, for identifying mechanisms and the processes that underlie the response of tumors and the normal tissues to radiation. So we need to know a bit of knowledge about hypoxia and what it does to the tumors. And when there is hypoxia, there can be some oxygenation, what we call it as re-oxygenation of that of the tumors. Then we need to know about tumor cell repopulation, because cancer is a proliferating tumor. The cells will be repopulating. So we need to know about tumor cell population, repopulation, and at the same time, the repopulation of that, the normal cells also. Then ionizing radiation's basic target is the DNA. So we need to understand the mechanisms of the repair of DNA mechanisms. So Dr. Kumar will be taking those things also later on. And next is the develop specific treatment strategies. When we understand these things, we are in a position to basically develop various treatment strategies, which is the best for our patients. It can be in the form of utilizing hypoxic cell sensitizers. Because when we say that why hypoxia is not good for the radiation, so as a result of this, we have to have some sensitizers, some compounds which will actually sensitize the hypoxic cells and improve the benefit of the radiation which we are given. We have got targeted agents. So we have to develop treatment strategies wherein we can use all these things along with that of radiation to improve the therapeutic ratio. Then I've spoken about protons, which are high LET radiations. We're going to know about that later on. We have to use, instead of now, we are using X-rays. Dr. Sarin must have spoken about X-ray. I don't know how many people attended his class yesterday. He must have spoken about X-rays, which are something which are not very beneficial, but the more benefit can be given from these the radiations, which are high LET radiations, such as the protons or the carbon ions. Okay. Then where we have, from the basic understanding of biology, we are now able to give the radiation dose, a curative radiation dose over a period of time, what we call it as fractionation, right? And over a period of time, we have also understood how to fractionate. Means when we have to give, say, about a curative dose of, say, 70 gray to the tumor, we don't give it in one day, because that's going to damage the normal cells also. Our main aim is not to damage the normal cells, but to basically damage the tumors. In that 70 gray, what we can do is to divide it over a period of time so that it efficiently kills the tumor, but also doesn't cause any problems to the surrounding normal tissue. So we have understood how to do fractionation changes in EBRT, means external beam radiotherapy. And then we have got so many chemotherapies. Radiation is not the only agent to kill the cancer cells. There are so many chemotherapeutic agents also. So now we have started to use chemotherapy along with radiation to again maximize the benefits of radiation therapy. And ultimately, understanding radiation biology, we are on one side, we are knowing the clinical effects of radiation treatment. On the other side, we are understanding the mechanisms of how ionizing radiations are bringing about changes in the biological matter. So these two together are married to develop mathematical models, okay, to predict whether this person in the future will be cured of tumor or not, or to predict whether this particular patient will develop any toxicity of late toxicity of radiation treatment. So we are developing all these models. We call it as LQ model or a linear-quadratic model. You don't have to understand that now. Dr. Kumar will take it. Some models, what we call it as normal tissue complication probability models and tumor control probability models. So these are using the knowledge that we gain from clinical observations and the knowledge that we have gained from the biological observation, marry them together to develop these mathematical models. Okay, so that we can predict in the future whether the tumor will be controlled or not, whether the patient, that particular patient, will develop any normal tissue complication. Okay. All right, now let's get into the crux of radiation biology. So when radiation therapy is given to any biological tissue, and in that, for that matter, any ionizing radiation. So there are three things happening, and that is based upon time scale of events over a period of time. So the three things which are happening. So when suppose Dr. Sarin told us that electromagnetic radiations are all, they travel in the straight line. Okay. So when a single electromagnetic radiation or a photon, single photon enters a biological cell, it can cause, the first thing that will happen is the physical changes. The physical changes can be either excitation or ionization. Everybody knows about what excitation is and what ionization is, okay? And this is a very, very, very fast process. And this can happen within 10 to the power minus 18 to 10 to the power minus 12 seconds. It's that fast a process. Once that is done, then a phase of chemical changes will take place. That can be in the form of enzymatic reactions, or because of this enzymatic reactions, there can be some free radical changes happening, free radical reactions happening. These are all part of the chemical processes that are happening after the physical process is completed. And because of this, the target cell, target structure within the biological matter, which is the DNA, gets damaged. But again, every cell in a body, every substructure in a body will have a mechanism to compensate. Although it gets damaged, there can be some reparative process happening. And all these things come under the chemical changes that are happening, okay? And then comes the biological process. Once the damage done by the radiation is fixed, then all the biological processes will start. And it can be anything between a few minutes to as long as years together, okay? So they can cause, so it can, when the biological process happen, we call it all these three things are the biological processes. That is the early effects. And the early effects usually happen from few minutes. When you give the ionizing radiation to the biological tissue, it starts from few minutes and can continue for about few days, the early effects. All right. Then the late effects, after few days to few months to even few years also. And also second malignancies, what we call it as radiation-induced carcinogenesis, is also a biological process that can happen after years after giving the ionizing radiation, okay? So some facts about it. So when 1 gray of radiation dose is given, there is an excess of 10 to the power 5 ionizing events which are happening. And that will happen within a volume of every cell that photon interacts with in a diameter of about 10 microns. These ionizing and excitation leads to breakage of the chemical bonds. This is the chemical process, and formation of broken molecules, which we call it as free radicals, right? And these free radicals are responsible for the damage they cause to the DNA. So damage happens at the subcellular level. The target, as I already mentioned, is the DNA. And at the cellular level, what will happen is that the clonogenic cells or the stem cells, everybody has heard about stem cells, they get damaged. And ultimately, this is expressed at the tissue level when there is an organ damage, okay? And the majority of these lesions, which occur in the DNA, can compensate and get repaired. But few cannot get repaired. And those lesions which cannot get repaired result in the cellular death, okay? So in order to understand what are the biological processes over a period of time, maybe it's more than 70 or 80 years old, lots of biological assays have been developed. It has started from in vitro biological assays, that is outside the body at the cellular level, to in vivo in the mice and in the higher animals, and later on in the humans. So let us start. What all biological assays are? It's just the basic concepts I'm giving. What biological assays have been developed to understand the biological processes that the ionizing radiation causes to the biological matter. So so when ionizing radiation is given, we have to quantify the effects of that ionizing radiation on the biological matter. So in 1942, the first of the in vitro biological assays were developed, and they were developed in the form of chromosomal, by studying chromosomal aberrations. And you can see the radiation, these chromosomal aberrations were actually studied in plants, basically. not even in the cancer cells, pollen. Okay, and they could show that there is aberration in the chromosomes. Multiple types of aberrations can happen with ionizing radiations, right from translocations to that of mutations to deletions, and also you can also have certain different types of chromosomes which are called as ring chromosomes and dicentric chromosomes. Of all these sorts of chromosomes, the dicentric chromosomal aberrations, the dicentric aberrations, and the ring chromosomes are the most lethal. That can lead to where the repair usually doesn't happen. That can lead to cell death. So this is one of the earliest radiobiological assays that were developed to understand the processes, radiobiological processes of ionizing radiation in the form of chromosomal aberrations. Next, 1953, clonogenic assays were developed. What I said earlier, the stem cells or the clonogenic cells are one of the most important cells which are being targeted by the ionizing radiations. So people started to study about clonogenic assays. What is important for you people to understand is that clonogenic assays doesn't talk about cell death. It talks about the reproductive integrity of that of this. Okay, so when you're giving this ionizing radiation, if you are able to stop the reproductive integrity of that particular clonogene, then basically it is the ionizing radiation has had its effect on that particular cell. So here we basically talk about reproductive integrity, and the survival of the cell is equivalent to the loss of is equivalent to the maintenance of the reproductive integrity of that cell. And in order to understand this, we basically count the colonies of the after seeding the cells and giving them ionizing radiations. The entire basis of it, Dr. Kumar will be talking about as to how we do it. And this can be done both in adherent cells as well as in the suspended cells. And once we do these clonogenic assays, and by giving different doses of radiation treatment, we can plot a survival curve for these particular cells, okay, over a period of radiation doses that has been given. And that will tell you about the reproductive integrity of that particular cell, okay. And next, based upon that, using various mathematical models, we have developed these cell survival curves from these clonogenic assays. And these are still, remember that, the clonogenic assays are still the gold standard in vitro radiobiological assays. We still do that. Even after evolution of so many biological assays, we still do this clonogenic assays. And from these clonogenic assays, we basically draw the cell survival curve. The cell survival curve will allow us to know so many things, of which one of the most important thing is the intrinsic radiosensitivity of that particular cell, which we call it as D0. Sometimes we call it as D37 also. What exactly it means? Means that when you give 1 gray, or which is equivalent to 96 Roentgens, or equivalent to 100 centigrade, it reduces the survival by a factor of 37%. It never happens that a cell, when you are giving radiation to a group of cells, all the cells will not die. And so only a few cells will die. And to quantitate that few cells, we have used this terminology called as D0, which says that when you are giving 1 gray of radiation or 100 centigrade of radiation or 96 Roentgens of radiation, it reduces the survival by a factor of 37%. That is, if 100% cells are there before radiation, after you give 1 gray of radiation, only 63% of the cells will be surviving. And this tells you about the intrinsic radiosensitivity of that particular cell. Next, after the clonogenic assays, many people in the 1970s, they wanted to look at the damage to the DNA by the ionizing radiations. So they have started using these assays, using micronuclei assay. And so what happens is that when you give radiation to these cells, what you will be able to see is that there are 2 nuclei within that cell. And if there is if a single nuclei is divided into two nuclei in simple terms, that means the DNA damage has happened, and you count these basically two nuclei and then quantitate the amount of DNA damage that has been done. It's a simple assay. And it's not only for ionizing radiation, it's also for any other genotoxic agents, even for UV radiations or for chemotherapeutic agents. These assays can be done, and we do it for ionizing radiation. However, over a period of time, many other assays have come, and it has slightly become more historical now. So next is this: in 1990s, the COMET assays for DNA damage and repair were developed. So you are able to see these photographs. They look like comets only. They tell us there are different types of COMET assays, and each type of COMET assays talks about a single-stranded DNA break or a double-stranded DNA break. Even COMET assays can tell you about, give information about interstrand crosslinks and even intrastrand crosslinks. So this is what we do. We give different doses of radiation and see the amount of DNA damage. Now this is the head, COMET head, which means that the DNA is intact. It does not cause anything. And this is the tail of the comet, which says that the DNA has damaged in very simple terms. And the length of the tail will give you the amount or quantitate the amount, either the double-stranded DNA break or the single-stranded DNA breaks. There are many more things which can be, the COMET assay inform us, but I'm not going to talk about that. In simple terms, COMET assay is one of the important radiobiological assays to quantitate not only DNA damage, but also the DNA repair. In 2000s, this particular assay came into view, a book, gamma H2X assay. We all know histones are there which are coiled with that of the DNA double helix, and H2X is one of the histone which is present in an intact, which is coiled with that, which is associated with the intact DNA. When you're giving this ionizing radiation, what happens is that it acts as it undergoes phosphorylation and gets converted into gamma H2X, which we see using various instruments like microscopy or even with flow. And this tells us you can see that these are the foci, okay? These are the gamma H2X foci, which each foci says, basically tells that each foci is equivalent to one single double-strand break. So it's a highly specific double-strand DNA break assay. So people usually use this for understanding the double-strand breaks after ionizing radiation. And this is also one of the earliest events which happen after giving the ionizing radiation. So it's also called as a sensor for ionizing radiation. Okay, so far so good for the in vitro assays. I'm not going much deeper. There are so many other assays, but these are the most important assays which are being done to understand the biological processes at the in vitro level. Now coming to in vivo assays. What I had said earlier was that ionizing radiation can damage not only the tumors, but also can damage the normal tissues. So we, using mice models, we not only look at what is happening to the tumors, but also what is happening to the normal tissues. So using mice, we look at jejunal crypt assays, skin clonogenic assays, and even stem colony assays or bone marrow assays for understanding the toxicities which are happening in these normal tissues. And these tissues are highly proliferating tissues. And radiation produces very early effects in highly proliferating tissues. So these tissues are basically used to understand the damage or the early reactions which is happening to these tissues, okay, by giving ionizing radiation. One is the kidney assay. Kidney assays are usually done at a late stage after giving radiation. You can see that we do it at 60 weeks, which is after a year or so. So these are used to understand the late effects of radiation treatment. People also do for tumors. We also do 3D spheroids, or we also call it as 3D organoids. And here we basically make a 3D model of the tumor cell, which mimics the tumor itself. And it will have all the features like you can have the tumors You can have the you can also have the necrotic cells. All those things which are there in the tumor in the mice which we develop, we can develop it in the lab also. So a lot of things are also done using these spheroids to understand the effects of ionizing radiation on tumors. So you can very well see here. It's a 3D organoid model of a colon cancer, which has got in the center. It has got so much of necrosis, and surrounding there is all viable tumor cells. And as we go from center to the periphery, we can see that the level of nutrients and the oxygen levels are much higher. And when you come to the center, there is necrosis cells, so there's so much of hypoxia. The nutrition is also quite less. So we can actually mimic a in vivo tumor by using these spheroids. This is very important for us as radiation biologists. We use a lot of mice models to understand the effect of ionizing radiation on the cancer cells. So we use syngeneic tumor models, which are nothing but inbred, genetically identical, or even sufficiently identical and immunologically compatible to the other species. And these type of syngeneic tumor models are basically used for bone marrow transplant studies in radiation therapy, because radiation therapy is an important component for bone marrow transplantation. So we use these type of models for that. So examples like bulk C57BL by 6 and 6 CCH, and all these can be used for in leukemia model, or there's something called as Lewis lung carcinoma model. These are a murine lung cancer, where in this particular mouse, you can actually spontaneously develop a lung cancer. And if you're interested in seeing the effects of ionizing radiation on lung cancer, you can use these particular models. Sometimes sarcoma models are also used. So these are all syngeneic tumor models, which are genetically identical. Okay. Next is the xenograft tumor models. More commonly, these are used than these. And these xenograft tumor models are basically immune compromised or immune suppressed mice. Examples are the nude mice, in which there won't be any B cell immunity. Skid mice, also called as not skid mice, where both the B cells and the T cells are all suppressed, and the NSG mice, where all the types of immune cells are suppressed. The B cells, T cells, NK cells, and the macrophages. And this is all done for tumor implantation studies and tumor growth studies. Most commonly, we use these nude mice and the skid mice for various types of this thing. But nowadays, if you might have heard about CAR T cell therapy, in CAR T cell therapies, we have to use, in order to do the preclinical studies, most common, we have to use these industry mice. So they can be either, so xenograft models, commonly they are used for basically understanding the effect of any genotoxic agent on cancer, and these tumors are implanted on subcutaneously, right? But we also use orthotopic models, or PDX models, that is patient-derived orthotopic models, where the tumors from the patient is taken out and then implanted into the organ of origin in the mice. And these are now commonly used, especially for that, for understanding the effects of ionizing radiation on brain tumors. Transgenic mice and knockout mice models, not commonly used. So they are mostly used in the knockout mice. If you want to use, you basically use it to understand the function of that of the gene. But as of now, we don't commonly use these knockout mice models. But if you are interested in understanding the function of a particular gene, what it does after ionizing radiations, then you can basically use these knockout mouse models also. All right, so we are now basically transcended from in vitro biological assays to animal models. And now we are coming to humans, where the radiation therapy is used. Even before radiation therapy was used, so whatever biological models were used, radiation therapy was ionizing radiation, or radiation therapy was given as a single dose of radiation, 1 ray, 2 ray, 3 ray, 4 ray, or even 6 ray or 10 ray. So this guy, Claude Regard, actually wanted to basically simulate whatever was done in vitro and in vivo in mice models. And he wanted to see what it is to give a single dose of high dose of radiation to a ram's testis. It's a very important and a commonly used experimental, in our all lectures, basically we use this particular slide because this actually gave us the idea of fractionation, where the entire dose of radiation has to be divided over a period of time. So what he did was that, so the testis of the ram was considered to be a tumor. So in one group, he gave a single tumoricidal dose of radiation. And what he saw was that there was necrosis, okay,
Of skin.
of the surface of the scrotum, basically, okay. So the next experiment, what he did is that he fractionated. So with fractionation, he got that desired result, wherein he was able to achieve what he wanted, basically completely sterilize the testis without having any toxicity of the scrotum or any toxicity of that skin or the skin of the scrotum. So this was done in 1920 in Paris, and this actually gave us the insights of fractionation in radiotherapy, of what we do nowadays. So I've spoken about fractionation. When we talk about fractionation in radiotherapy, there are actually 5 hallmarks or the 5 pillars for fractionating this radiation dose. We call it as radiosensitivity, or in other words, the intrinsic radiosensitivity of the tumor, repair and recovery. Okay. This is basically for normal tissues. I said ionizing radiation not only damages the tumors cells, but also damages the surrounding normal cells. So repair and recovery of the normal tissues. Then reoxygenation of the tumor cells, redistribution of the tumor cells, and repopulation of the normal cells. All these 5 actually are the pillars for doing the fractionation of radiation treatment on humans, right? Now, the 6th R has come, which is reactivation of the immune response by giving radiation treatment. So all these R's have biological effect on ionizing radiation. And of these all 6 R's, the 5 R's are the pillars for fractionation in radiation therapy. That you have to remember that. So when we give radiation treatment, so you got the tumor and also the surrounding normal tissues. So radiation, as I said, will affect both the tumor and the surrounding normal tissues, which are, it's not avoidable because it's all within, they are all very close to the tumor, the normal tissues. So you will have some effect on the normal tissues. So the response of the tumor will be regression, and it can also regrow. But failure to regrow is a good thing because then the tumor is controlled. And when it comes to normal tissues, you can have what I said is early reactions or even late normal tissue reactions, which is detrimental, right? So we have to avoid the early and the late normal tissue reactions, and at the same time, we have to have a local control. That is our main aim in radiation treatment, okay? And all these things will depend upon and what I said was, for tumor control best, you have to optimize it, isn't it? The tumor control has to be highest and the normal tissue reaction has to be lowest. And what is it called as? Therapeutic gain or therapeutic gain. And that therapeutic gain will depend upon the total radiation dose that we give and the time and the volume that we irradiate, okay? So this is a very commonly used slide when we teach our students. This is called as the Hall-Thiessen curve. And it tells about the amount of radiation dose that has to be given to achieve the optimal therapeutic effect. On the right side, so this is left side, sorry. The curve just talks about the tumor control probability or the radiation dose that is required to give to produce the local tumor control. On this side is the radiation dose that will produce normal tissue complications. So we need to have that differential between the two so that you get that therapeutic gain. And our main aim is to separate these two curves by whatever way you want to do. This is our main aim, right? So just to illustrate that, when these two curves are separated, this is a favorable thing for us, for clinical radiation oncologists. But when these two are close to each other, it's quite unfavorable for us, because for a given radiation dose, you're curing the tumor, but at the same time, you're also causing a lot of normal tissue complications, which is not good, right? So this is what we always try to achieve. And this is what we call it as a therapeutic ratio. It's the ratio of probability of tumor control by the likelihood of normal tissue damage, right? So as I said, the tumor control probability depends upon that of the amount of radiation dose that we give. So the first dose response curves for radiotherapy were basically published in 1936, all right? But they were published for skin toxicities, and they extrapolated it to the tumor control, right? And they have shown that after about 50 gray or 50 Roentgen, I think they have put it in Roentgen, there is not much of not much of response. So they basically said, and the endpoint basically was the telangiectasia. It's a type of skin reaction that we see after giving ionizing radiation. And this, they extrapolated it to the tumor, because what they thought was that when you're getting this toxicity, you can't go beyond this particular dose, and that should be good enough for tumor control, which is not the correct thing. But at that time, they used this method to basically extrapolate it for tumor control. Then came the importance of time and duration of radiotherapy. So what you can see is that when you're giving radiation therapy over a period of time, the toxicity is also affected, right? Now, for example, you're planning, say, about 60 gray of radiation treatment in, say, 14 days, in about 10 days, and about 18 days. Let us see, there are three points where radiation is being given over 14 days time, over 10 days time, and over 18 days time. When you're giving radiation in about 14 days time, you are able to basically, you can see that this is what is called as radiation epidermitis, I think, yeah. And also mucosal toxicity. So when you're giving this over a period of 14 days, you're able to reduce the toxicity, okay? So there is mucosal toxicity, but it is reducing. And then again, you're giving radiation, you're able to reduce that in that 14 days time. But if you are able to if you are reducing the duration of the radiation treatment, what is happening is that the toxicity is not completely getting reduced. So here you are getting more amount of radiation toxicity when you are reducing the total duration of radiation treatment. And if you are increasing the total duration by to 18 days, of course, you are reducing the toxicity, but there's always a chance that your tumor can also regrow. So the importance of time duration of radiation treatment was shown by these experiments. And now also we actually follow these things. So this is another important thing in fractionation for radiation therapy. Then the importance of volume was also expressed at that time. So tolerance doses in radiotherapy was this is the paper which was published way back in 1942 in BGR. What they showed was that when you are irradiating a small volume, you are able to increase the dose. So it's a field size is about 6 into 4 centimeters. You are able to give 20 gray. But when the field size is increasing, you have to reduce the dose because of the toxicity that can cause. So in the same way, when you're giving single dose, even with 1 week, 2 weeks, 3 weeks, you can see that with small field sizes, the small volumes, you are able to give higher doses. But when you're increasing the volume, you cannot give that high doses. Okay. So those are the three important things apart from the pillars, 5 pillars of 5 hours of radiobiology, these are the 3 important things also, that is total dose, time duration of treatment, and also the volumes are important in fractionation radiotherapy, right? So now we have basically evolved over a period of time, and you can very well see that we started from 2D RT in the 1890s and went on to do 3-dimensional conformal radiation treatment, where we are able to completely shape the radiation beam to the shape of the tumor. And with that, we are also having all these delivery techniques, which we call it as IMRT, SPRT, 4D RT, and particle therapy. What you can able to see is that with these refinements, we are able to give radiation more to the tumor and also spare the surrounding normal tissue. And that's the very reason why also, because we have evolved in delivery techniques, we also have to evolve in the radiobiological aspects of these newly evolved radiation therapies. So the new radiation biology is on inducible responses. We are trying to understand the genomic instability also of these various ionizing radiations. And I've already spoken about flash radiotherapy and all those things. So we are trying to understand what it does. We are trying to understand about the bystander effects. And nowadays, systems biology and omics are being incorporated into the radiation biology paradigm as well. And since we are not only using radiation therapy alone, we are also giving chemotherapy and immunotherapy along with that of radiation treatment. So we are using multimodal therapy. So we have to understand the effects in the radiobiology parlance. Thank you very much.
Thank you.
So next is Amit.
You have 2 talks back to back. How much is each talk? So maybe we'll after one talk, how much what is the duration?
Around, I have made just general, so I was not doing something, but around 30 minutes, half an hour.
So after one talk, we can have a lunch break, and then again you can come back. Okay.
So I have a continuous 4 lectures with them. So
Yeah,
around
yeah.
I will stop.
Yeah, so maybe you have 120 or 30.
I will stop. So after lunch also I will continue. So
Yeah,
I can
yeah.
finish and then I will.
Yeah. You have a PDF.
Yeah. So actually, you should have some board kind of thing, so we can actually teach with them.
Oh, yes.
Next time we can have that.
We'll have a board. Actually, in this also there's a whiteboard also. So we have it.
Good. So good afternoon. So first of all, I will introduce myself. So I am Amit Kumar from Bhava Atomic Research Center. So I am from Division of Radiation and Photochemistry. Basically, I am a biologist. Then after doing my training, I joined to chemistry group, and currently I am working with a group for the development of radio protectors. So I generally take the class of radiation biology in CBS, and this time Jan told that I have to take some classes here also. So Jayant has introduced you about the radiation biology, and I will be taking some fundamental basis how radiation causes damage to biomolecules. That we need to understand, then only we will be able to know that what kind of a strategy and other things we can develop to manage the radiation toxicity, not only in normal cells, but also in cancerous cells. So radiation biology is the amalgamation of chemistry and biology. There is a lot of chemistry and there is a lot of biology. James showed you the timelines of damage. The moment radiation falls on a cells, the events start taking place. The first event that takes place is the physics that we call as radiation physics. How the radiation interacts with the matter. Matter, when I am saying it is the atom. Everything is made up of atom. So finally, radiation interacts with the atom. There are different types of interaction, Compton interaction and other things. If you want detail, I can talk in the next lecture. But those are the radiation physics. And then the next phase is the radiation chemistry.
And what is covered yesterday, Compton and photoelectric.
Okay, so chemistry is the next phase where because of the interactions, you will have a lot of changes, and then electrons and other things will start interacting with those things are chemistry. And biology is the response, how cell responds to those damage. So we have 2 branches, radiation chemistry and radiation biology. Radiation chemistry talks about the all the chemical changes which happens when the radiation falls inside the cells. And radiation biology is the field where we study the responses of the cells to all these changes. Now, what I will teach you about, because all these introduction has already been given, what I will teach you is that how the damages happens to the biomolecules. So in that respect, so we have, the cells are made up of, what is there inside the cells, you all already know. So we have in cells around 70% of the water. And this is the region for the damage of the cells after radiation, once radiation falls on a cell. And then we have all these biomolecules. I call it biomolecules, although these are known as organelles also. So we have DNA, and cell membrane is made up of lipids. Then we have a proteins, and mitochondria has again DNA, membrane, and protein. So basically, if you talk about the cell has 70% of the water, and rest 30% are the protein, lipids, and nucleic acid. So radiation, when radiation falls, and we are talking about ionizing radiations. Ionizing radiation means radiations which have very high amount of energy. The moment radiation passes through a medium, it deposits energy in the medium. So we are talking cell as a medium. When radiation passes through the cell, what radiation is going to do? It is going to deposit energy inside the cell. Now when we are talking about ionizing radiation, the energy deposition is highly non-selective. See, this is also a radiation. This is also depositing energy to this environment. But here, the environment will not absorb the energy of these radiations because this radiation has a very low energy. This will be absorbed only by the molecule if the difference in the electronic energy level matches with the energy of this radiation. But when we are talking about ionizing radiation, it has quite a lot of energy. So energy deposition becomes non-selective. It will deposit energy to the material which has the very high volume. So inside the cells, you have 70% of the water. So what it is going to do? When radiation will pass through the cells, it will deposit maximum amount of energy to the water molecule. And that leads to radiolysis of water. We call in radiobiology, we call it radiolysis of water. The water molecule will start breaking down. And because of the breakage of water molecules, we have a generation of free radicals. And those free radicals will actually cause damage to all these biomolecules. So this is what we are going to study in detail. So if you classify the types of radiation damage, we can classify into two types. We have a direct action, and another we have indirect action. Now, what is direct and indirect? That you have to understand it very nicely. When I talk about radiations, we can classify the radiation based on the quality in two types: low LET radiation and high LET radiation. I think you are aware about that. You know whatever. So these two types of radiation interact, and their mechanism of biological damage is quite different. And that is why it comes to direct action and indirect action. So what is direct action? When the radiation interacts directly to the biomolecule and deposits energy to biomolecule. So inside the cells, we have a biomolecules, DNA, protein, and lipid. So if the radiation is depositing energy directly to those biomolecules, then we call it direct action. And what is indirect actions? Indirect action, when radiation deposits energy to the water, and because of the radiolysis of water, you have a generation of so many species, reactive species, and those reactive species, if they are interacting with the biomolecules, then it is called indirect actions. So low LET radiation generally follow indirect actions, and high LET radiations follow direct actions. Now, so this is the basic mechanism how radiation causes damage inside the cells. So once radiation falls in a cells, what will happen? I told you water is the 70% volume. In case of low LET radiation, this energy will be deposited to the water molecule. And then water molecule will be broken down. So it will break in a very fashions like this is the H2O plus. So, and there will be generation of electron. Now this electron is high energy electron because radiation was having the energy. That energy had been given to break this water to this water molecule. Because of that, this water molecule has got ionized, and it has generated the electron. So whatever extra energy the radiation, some energy will be consumed in breaking the water molecule. Rest of the energy will be transferred to this electron. Now this electron will go and interact with the nearby water molecule, and this kind of chain reaction will keep on going on till the energy of this electron is thermalized. What do you mean by thermalized? Means that electron does not have any energy to cause further ionization. And that is called aqueous electron, EAQ. Aqueous electron means now this electron does not have any energy to cause further ionization. So that much event will take place when radiation will fall inside the cell. So now you can see that you have a H2O plus, and now another event, what happens, that breaking of the water molecule. So this is the excitation event. Water molecule gets excited. I have written H2O star. Star means excited water molecule. And this excited water molecule undergo bond breaking. And because of that, you have a generation of hydrogen radical and hydroxyl radical. So what is radical? When I am talking about radical means, radicals are the atoms or molecules having unpaired of electrons. So they are unsatisfied molecule. That is why they are called highly reactive molecule. They either need electron or they want to give extra electron. And that is why these molecules are highly reactive. They will start interacting with the nearby molecules. And this becomes the region of damage. So we have ionization, excitation. Now there will be further reactions. See, this H2O plus will react with another water molecule. It will make H3O plus, hydronium ion. And this, along with hydronium ion, again you have a generation of hydroxyl radical. So ultimately, if you see the radiolysis of water, what is the end product? Radiolysis of water, you have a high energy electron, which is doing again excitation and ionization till it gets thermalized. Then you have a hydroxyl radical generation and hydrogen radical generation. Now, out of hydrogen radical generation and hydroxyl radical generation, the most damaging one is the hydroxyl one, because it does the ionization. It does the, what it will do, it will oxidize the nearby ball. It will oxidize the DNA, it will oxidize the protein, it can oxidize the lipid. However, this hydrogen radical is a reducing type. So it is not that much damaging. Perfect. Now, what will happen but the problem with hydroxyl radical is that it has very short half-life. When I talk about half-life means, these are the species which are being generated inside the cells. And the time for which these species will be alive for reaction is called half-life. So half-life of hydroxyl radical is a microsecond, 10 to the power minus 6 seconds. So the moment radiation falls on the cells, you have a generation of hydroxyl radical, but this hydroxyl radical is going to be present inside the cells only for 10 to the power minus 6 seconds. So during that time interval only, it will get the time to oxidize nearby molecules. So practically, theoretically, hydroxyl radical is very damaging, but practically it is not damaging, because in cells, radiation falls. If you see the diffusion constant, the cell has a nucleus and nucleus has a DNA. Suppose hydroxyl radical is being generated in the cytoplasm, it does not have time to reach to the nucleus. By the time it will be converted into 2 hydroxyl radical will combine into hydrogen peroxide. And now this hydrogen peroxide is the long-lasting molecule. It's not a free radical, it's a molecular species, but it's a highly oxidizing. Now it can remain inside the cells even for hours. So after radiation, you will get damage not because of hydroxyl radical, but you will get damage because of the production of this hydrogen peroxide, which is going to be present inside the cells for hours. Now, I told you this hydrogen radical as such, it is not damaging because it is reducing type. But what is there inside the cell? We have an oxic environment. We have high concentration of oxygen because we are respiring. Each of our cell is oxygenated. Now, in presence of this oxygen, this hydrogen radical will make hydroperoxyl radical, HO2 dot. Now, this is again a very highly oxidizing reagent. And it also has a long half-life. So this is also, one of the major culprit inside the cells after radiation exposure. Now, what this hydroxyl radical or hydrogen peroxide or hydroperoxyl radical will do? They will react with the organic molecules. So all the DNA, protein, and lipids are organic molecules. In chemistry, we denote these molecules as RH. So these all will oxidize the RH and they will make organic radical, R radical, which again in presence of oxygen will make organic peroxyl radical. And this is the chain reaction that will keep on going inside the cells. You can imagine what can happen after radiation. These all events will take even for hours because these have the half-life now in hours. And because of that, you will have a DNA damage, protein damage, and lipid damage. So all these events comes under indirect action. So this is the water radiolysis. Free radical generation, now you understand what is the free radicals, which of the free radicals are damaging in nature, and this will lead to all protein, lipid, and finally it will lead to the cell death. That we will study that how the cell death happens, how the apoptosis and other events happen. Now another condition is for high LET therapy. You might have been heard by proton therapy, carbon therapy. So these are all high LET therapy. High LET therapy does not follow this route. It goes for direct damage because here we can localize the radiation. You might have heard about the Bragg's peak. We can actually pinpoint the radiation to the place where we want. So what it will do, the radiation will deposit energy directly to DNA, directly to biomolecules. And because of that, it will lead to fragmentation of DNA, fragmentation of protein, fragmentation of lipids. So, and ultimately it will result in cell death. So this is the direct action. So this is direct, this is indirect. Direct is followed by high LET, and indirect is followed by low LET. Now we want to know that what kind of damage happens to DNA. So we need to know the basic structure of DNA. Here I will not give a lot of emphasis because you might be knowing everything about this. So the building block of DNA is nucleotides. And nucleotides has three components: nitrogenous base, pentose sugar, and phosphate. And they join together to make the nucleotide, and then they polymerize to form the polynucleotide chains. Now, what you need to understand here that we have two types of nitrogenous bases. One class is called purines, and another is called pyrimidines. So pyrimidines, you have a thymine, cytosines, and purines, you have adenine and guanine. And this is the structure of nucleotides where, nitrogenous base is joined to the pentose sugar, and pentose sugar is joined to the phosphate group. Now, here you have to know two important things. So nitrogenous base makes a covalent bond with the first carbon of pentose sugar. And this bond is called N-glycosidic bond. Very important to understand the damage of DNA. So this is called N-glycosidic bond. Now here, if you see, this is the first carbon, second carbon, third carbon, fourth carbon, and this is the fifth carbon. Now this fifth carbon is joined with the phosphate group, and this is called phosphodiester bond. So there are two important bonds: N-glycosidic bond and phosphodiester bond. Now this is the double helical structure of DNA, and the diameter of DNA is around 20. How much? 29. Perfect. And these are anti-parallel strands, So these are anti-parallel strands, and then you have a base stacking between the two strands. Now, if you talk about one helix, the one helix has the length of around 34 angstrom. So this is the structure of DNA. Now, when you talk about DNA damage, so DNA damage, you see, so radiation, there will be indirect and direct. These are the generation of radicals, and radicals are now start interacting with the DNA. So where the interactions can happen, I told you all the free radical, either they want the electron or they want to donate the electron. So there are three chemical species inside the nucleotides: nitrogenous bases, pentose sugar, and phosphate. Now the targets for damage are the pentose sugar and nitrogenous bases. So all the radicals, whether it's a hydroxyl radical, hydrogen peroxide, or you have hydroperoxyl radical or organic peroxyl radical, during the indirect damage, they interact with the nitrogenous base and pentose sugar. And because of that, what will happen? Now you can imagine you have a two important bond: phosphodiester bond and N-glycosidic bond. Because of the oxidation of either nitrogenous base or pentose sugar, there will be delocalization of electron, and that delocalization of electron will lead to the breakage of either N-glycosidic bond or phosphodiester. If the N-glycosidic bond is breaking, then you have a generation of A-purine and A-pyrimidine sites. You might have heard that is a DNA damage. You have a generation of A-purine and A-pyrimidine sites. And if you have the breakage of phosphodiester bond, then you will have single strand breaks or double strand. Clear? So indirect damage, this is mediated by radicals. Radicals are oxidizing nitrogenous bases or pentose sugar. Now in case of direct damage, when you have a direct where radiation is directly interacting, radiation can deposit energy directly to these bonds: phosphodiester bond or N-glycosidic bond. And because of that, bonds will start vibrating. And the moment energy level goes beyond the vibration synchronization, then the bond will break. So that is called direct damage. It's not moving. Not moving. It's not working. Sorry. Okay. Okay. So now coming to, I told you that what are the different types of radiation damages that you can see. So first one is I have given you some structure. These are the structure of oxidized nitrogenous bases. I told you that all the radical species which are generated after radiations, they actually oxidize the nitrogenous bases. So here now you can see that these are the derivatives of guanine. These are the oxidized derivatives of guanine. These are the oxidized derivative of cytosine. These are the oxidized derivative of adenine. And last panel is for oxidized bases of thymine. All these are oxidized nitrogenous bases which are generated due to oxidation of, okay, perfect. Due to oxidation of nitrogenous bases by the radical species. So this is here 8-oxoguanine. What is this? This is the 8th position where oxidation has happened. And that is why now it has been called 8-oxoguanine. Later on, it gets hydrogen gets attached here. So now we famously call it is 8-hydroxyguanine. Okay, this is the 5-hydroxyuracil. This is the 8-oxoadenine. These are the thymine glycol. So these all are the modified nitrogenous bases which are found due to oxidation by the radical species. Now, suppose these are the bases when present inside the DNA, what problem they can cause? Can you imagine what problem the DNA will happen because of these modified bases or oxidized bases? They will lose their Watson-Crick base pairing pattern. Now, you know that purine has to pair with the pyrimidines. Now, because of this modification or oxidation, they will mispair. They will not follow the proper Watson-Crick pairing, and that leads to mutations. In the next round of replication, it will lead to mutations. Okay. So next round of replication, it will lead to mutation. Clear? Now, next is the strand break. So now here you can see that there is a difference. Now, here you have put two drawings. Now, can you tell me that which one is the single strand break and which one is the double strand break? Whether can we call it as a double strand break? No. This is the difference between the single strand and double strand break is that when the information is lost at the same position in the both strands, then it is called double strand break. So you have a breakage of phosphodiester bond at the same location in the both strands, it is called double strand break. And if then only one of the strand is damaged, then it's called single strand break. Now for cell, double strand break is much more lethal. The cell can survive with single strand break can be repaired, double strand break can be repaired, but even a single double strand break, if it remains inside the cell, in the DNA, the cell is going to die by apoptosis. So it is the double strand break which is the most dangerous or lethal thing that happens after radiation exposure. Now the next is the intra-strand cross-link. What is intra-strand cross-links? So in this, you will have the cross-linking between the nitrogenous bases of the same strand, or there is a possibility that the nitrogenous bases of the opposite strand can also cross-link. And because of this cross-link, again the topology of DNA will get disturbed, and finally there will be repair will start happening, and that repair will lead to the strand. So ultimately, you have whatever the kind of damage you are creating, this type of damage will lead to mutation. But all whether it's a strand break or it's a cross-link, that will lead to the cell death by apoptosis.
Sir, could you repeat that? Which one out of the you are more lethal?
Double strand break. So you have single strand break and double strand breaks. Double strand break is very lethal for the cell. Even a single double strand break, if it remains inside the DNA, if it remains unrepaired, the cell is not going to survive. They will undergo apoptosis. So this is the example where I have shown that this is how this is the single strand break. It looks, and this is the cross-linking of the nitrogenous bases. Now we move to the next molecule. I told you that is the protein. Protein is again made up of amino acids. The building blocks are amino acids, and we have 20 natural amino acids. I have given you the list, which you can classify chemically into acidic amino acid, basic amino acid, hydrophobic amino acid, sulfur-containing amino acid. We have a different types of classification. And apart, you might be knowing that we have 20 natural amino acids, but now this list has grown to 21. We have a 21st amino acid. Any one of you know that which is the 21st amino acid? selenocysteine. That is the selenium containing amino acids. So that is the unnatural amino acids, means it's a 21st amino. We have the genetic codons now. The terminator codon codes for the selenocysteine depending upon the secondary structure which is present in the mRNA. Okay, so now these are the building blocks for protein. If you see the chemical structure, so this is the chemical structure of simplest structure of amino acids. So we have a carbon which is attached to the hydrogen, and here I have written R. This is called side chains, which will be different for different amino acids. And most important thing that it has a carboxylic group and an amino group. And when it gets polymerized, this carboxylic group combine with the amino group of next amino acids and make the polypeptide chain. Perfect. So how this is how it links. So this is the peptide bond, which is, and these are the successive amino acids which are linked by the peptide bond, and this is the polypeptide chain. Now this polypeptide chain as such is not active for doing any function, biological function. It has to get folded into three-dimensional structure. So this is called primary structure of protein, and then once it gets folded, then we call it tertiary or secondary structure of protein. And during the tertiary or secondary structure, lot of forces come into play which stabilizes these tertiary structures. So this could be hydrogen bond, hydrophobic interactions, disulfide linkage, electrostatic interactions. So you can see that this is the kind of means bond is formed between the two cysteine containing amino acids. So once, and you have a different types of secondary structure, alpha helix. So this is the structure of helical, that's why we call it alpha helix. Then we can have a beta sheet, and it may differ from protein to protein. So different proteins will have a different secondary structure, and when the secondary structure will again get folded, they will acquire the tertiary structure. Some proteins also have quaternary structure, and quaternary structure is present only in proteins which have more than one polypeptide chain. If the protein has only one polypeptide chain, it can go only up to tertiary structure. The proteins having more than one polypeptide chain will have interaction between its individual polypeptide chain also, and that is why it is called quaternary structure. So once the protein achieves the tertiary structure and quaternary structure, it becomes biologically active. It becomes ready to do function. Perfect. Now, but now what happens when radiation falls on a cell? I told you that there will be generation of reactive oxygen species or radical species, which can now do the same kind of interaction. So chemically, so there is a very complex reaction. If you go, this is the just a representation of a peptide chain. It goes to many chemical reactions and finally forms the protein carbonyls. So protein carbonyls are the ultimate product of the protein oxidations by radiations that we measure in the lab also. Now, but in brief, what you should understand as a PhD student or as a biologist, that these are the brief reactions that you can understand. So protein can be represented by RH. Any organic molecule can be represented by RH. So they have hydrogen to donate very easily. And these are the hydroxyl radical. So hydroxyl radical snatches hydrogen from the proteins and it makes the protein radical. This is the protein radical, R dot. Now this protein radical can react with another protein molecule and it will generate another protein radical and water. Now two protein radicals can combine with each other and make the protein-protein cross links. Very important to understand. So you have a cell, radiation passes through the cells. It causes oxidation of one protein. That protein generates its radical. Now you have another protein. That protein also generates radical. Now two protein radical combines and make protein-protein cross links. If you have made a slide for microscopy, you might have used glutaraldehyde for cross-linking of the proteins. Fixation, cell fixation. Glutaraldehyde is a reagent that we use for the cell fixation. What it does? It cross-links the protein. Same thing radiation does. When the radiation passes through the cells, it leads to the cross-linking of protein, and why it happens, now you can understand the chemistry of that cross-link. Okay, fine. And some of the protein radicals in the presence of oxygen, they will make protein peroxyl radicals, and this will get degraded and make the protein carbonyls. Now those protein carbonyls are the ultimate end product of protein damage. Apart from that, due to all these modifications, protein is going to lose its secondary and tertiary structure. So protein will no longer be available to do its biological. Clear? So these all are the consequences. There are many proteins which are very important enzymes which are present. So there will be loss of enzyme activity, altered cellular functions such as energy production. So mitochondria has a lot of proteins which play a role in the energy production. And because of radiation, since those proteins are going to lose their physiological functions, you will have no energy production. Inactivation of there are several transcription factors, then changes in redox potential. There are some of the proteins which are maintaining redox homeostasis inside the cell. So they are also going to be changed. Now you can imagine what will happen to the whole pool of proteins which are present inside the cell after radiation passes through the cell. These all are the consequences. So DNA is damaged. You have a lot of damage to protein. Now the third molecule is the lipids, which is also present in the membranes. Every membrane is there in each organelle, even plasma. So everywhere lipid is there. So lipid, what is lipid chemically? So lipids are the esters of glycerol and fatty acid. So we have, this is the glycerol, and you can see that each of the carbon has the free hydroxyl group, and these are the fatty acids. So here I have written R, R1, and R dash. R means this chain can be any length, it can be, we have a different types of lipids. So this R will vary depending upon its chain length. Now these fatty acids get saturated to each of this hydroxyl group, and you have a triglyceride. And this is the simplest structure. Now if you have phospholipids, where you have one of the hydroxyl group is attached to a polar molecule, phosphate group and phosphate group. And because of that, this molecule is the amphiphilic in nature. Amphiphilic means it has a hydrophobic tail and it has a hydrophilic head. So it looks like just like this. And this makes the lip. You have a plasma membrane. So this is the basic structure. Now how the damage can happen to the lipid. Same. So now here you can understand lipid is the LH. It has a lot of hydrogen which can take part in the reactions. Now hydroxyl radical will snatch hydrogen from the lipids and it will make lipid radical. This lipid radical will react with another lipid and it will make another lipid radical. Though two lipid radicals will combine and make lipid-lipid cross-links. Then again, lipid in the presence of oxygen, it will make lipid peroxyl radical. Now this lipid peroxyl radical can combine with another lipid, can attack to another lipid, and it will make lipid hydroperoxide. And that lipid hydroperoxide can undergo degradation and make aldehydes and ketones. So if you want to do some experiment in the lab, just take the cells, radiate the cells, and make the lysate and evaluate for the aldehyde and ketones. You get a lot of aldehydes and ketone in the irradiated cells. Generally, we don't measure. Now, the one of the aldehyde which we call as 4-hydroxy nonenal, 4-hydroxy HNE, which is even measured in the clinics also for checking any kind of oxidative stress. So 4-HNE you can measure in the lab. So that is nothing but you are measuring the effect of radiation on the lipid peroxidation. So clear? So this is how lipid damage happens. This process is known as lipid peroxidations. This process is known lipid peroxidation. This happens even when you do cooking in your house. When you warm the oil in the anything, there is a lot of lipid peroxidation that happens during that process. And it depends upon the unsaturation. So unsaturated fatty acids are more prone for undergoing lipid peroxidation. Because of lipid peroxidation, now again, what will happen? So this is the product, 4-hydroxynonenal. This is the aldehyde product, and the malondehyde is also there. So this is the ketone, and this is the aldehyde. Sorry for that one. This 4-hydroxynonenal is a ketone product, and malondehyde is an aldehyde product, which are generated from lipid hydroperoxide, which were produced from the lipid peroxidation process. And you can measure these things in the lab after radiating the cells. So what will the consequence of membrane damage? Because of this lipid peroxidation, fluidity of membrane changes. And because this will affect any cellular uptake. All the nutrients are taken inside the cell by diffusion or osmosis, and some receptor mediated process. So all these things will be affected because of the lipid peroxidation. Apart from that, this lipid peroxidation results in the production of ceramides. And ceramides are again the signals for the cell death. The moment ceramide concentration will go above certain limits, it will give feedback mechanism to the cells that you need to undergo apoptosis. So lipid peroxidation independently can lead to apoptosis through ceramide pathway. Is it clear to everyone? So now what I taught you, that once radiation falls to the cells, which is the major place where radiation is going to deposit the energy, Water, because cell has 70% of the water. So the major event that is going to take place inside the cell is the radiolysis of water. And because of the radiolysis of water, what will be the generation? What it will produce? It will produce long-lasting hydrogen peroxide is the major culprit. Even if you see books everywhere, they say that hydroxyl radical, but hydroxyl radical has a very, very short half-life. The hydroxyl radical which are generated only in the nucleus can cause the DNA damage. The hydroxyl radical which are generated in the cytoplasm, they cannot reach to the nucleus. So I'm not saying that hydroxyl radical will not cause the damage, but the long-lasting damage. Like Dr. Jain told that, okay, this biological phenomenon will keep on going even after days. Who is sustaining this hydrogen peroxide, reactive oxygen species which are generated after the radiation exposure, which have long life, they keep on causing damage even for the weeks. So hydrogen peroxide, all the peroxyl radicals, they will type, and this is a chain reaction. I told you what is chain reaction. One molecule attacks another molecule, one molecule attacks, this keeps on going inside the cell. So that is the thing. Now, what are the targets of these reactive oxygen species? DNA, protein, and lipids. Now, DNA, what kind of damage it can do? It can lead to the breakage of phosphodiester bond. It can lead to the breakage of N-glycosidic bond. Due to breakage of N-glycosidic bond, there will be generation of apurinic site, apyrimidinic site, and that can lead to mutation. And due to nitrogenous base oxidation, they will lose their normal Watson-Crick pairing pattern, and that will also lead to mutation. And if the phosphodiester bond is broken, then you can have single strand break, double strand break. And double strand break is the most dangerous, lethal thing. Okay, and intra-strand cross-linking also happens because of the modification of the oxidation of the bases. Now, with respect to proteins, I told you there are reaction that is called protein carbonylations. Ultimately, is that you will have a protein carbonyls. As a result, the protein will lose its secondary and tertiary structure. They will stop functioning, and that will be not good for the cell. And third is the lipids. So I hope you are clear up to here. Now, what are the consequences of these all damages? There are 4 consequences. Either cell cycle arrest, or there will be apoptosis. So see, the cell will division delay. This is called reproductive cell death. And that is why we do clonogenic assay, that I will teach you. So these are the 3 fate of the cell, fate of irradiated cells. So there will be cell cycle arrest, there will be apoptosis, and there will be loss of clonogenicity. So we should measure this. If you have radiated the cells, we can measure this. I will stop at this place, and then after I will take another class for clonogenic assay. That's all. And
any questions?
Very good. I also learned some new things today.
No, it's okay.
So,
We usually talk only about hydroxyl radicals.
Yeah.
That is one important thing
Yeah.
is told that it's the hydrogen peroxide which remains for a
Yeah.
Longer time. See, hydroxyl, it is not that they don't cause damage, because
Yeah.
there will be some hydroxyl which will be generated inside the nucleus, also has water. But cytoplasmic hydroxyl is not going to travel to the nucleus.
so most of it is with low LET reagents, sparsely ionizing radiation. If you want to clarify that.
Yeah, all this damage, indirect
Yeah.
damage I told you, they are valid only for low LET.
Yeah.
X-ray, gamma, electron. But if you go for proton therapy, direct damage will come. So direct and indirect difference is only there. Direct, the energy will be deposited directly.
Low LET means sparsely ionizing radiation versus densely ionizing
Ionizing.
radiation.
So definitely high LET like proton will do dense ionization,
Yeah.
and this will do the partial.
So all this is for the sparsely ionizing radiation. Okay, I think now.
Can any cell escape these damages?
Cell escaping this damage depends only upon what are the intracellular concentrations of thiols. Cells have a redox couple, glutathione. I will teach you that cells have, nature has given cells to protect from any kind. So this toxicity is not different from phototoxicity. Phototoxicity also generates a lot of reactive oxygen species. Only the nature of reactive oxygen species are different. So we have a lot of intracellular antioxidants. So we have a lot of intracellular antioxidants, all the repair pathways. So nature has given all those things to take care of any kind of genotoxic stress. Genotoxic, proteotoxic, and lipidotoxic. There are 3 types of stress that radiation is doing. It is doing genotoxic, proteotoxic, and for taking care, cell has given. But the amount of stress which is generated after radiation, even the intracellular mechanism that we have, they are not enough to take care. And that is why the cell undergo apoptosis. See, we are getting every day 10,000 mutations, but you know, it does not mean that all the mutations are getting repaired, na? Or damage. You see, every day we are getting DNA damage. Even the sunlight exposure causes some level of DNA damage, but they are getting repaired. But radiation leads to such an extent of damage that whatever the mechanism, they become inefficient to completely repair.
Overwhelmed.
And one DSB will lead to apoptosis. So you have a 10,000 DSB, the cell has repaired 99,999. One DSB left and the cell will undergo apoptosis. That is the reason.
Thank you very much. It was an excellent talk. I think all the students and us, we learned a lot. And I think on this YouTube video, when people will see, they will understand. So see it again and again and again, and then your concepts will be clear. And this was a very important foundation, you know, what case. So I think we'll break for lunch. And those who are watching it on the YouTube live, we are pausing the share for some time.
So now, in my first lecture, I told you that what are the fate of a radiated cells. So in brief, the radiated cells undergo cell cycle arrest, then they undergo apoptosis, and these all things happen because of the changes inside the cells, which are called redox changes. So now I'm coming to the topic. I will give you the details of DNA damage response pathway, repair pathways in my next lecture. But now I am moving to some biology assays that we do in our lab. So that comes under molecular assays. So whatever the molecular assays that we do in the lab, we do for measuring either the cell cycle pattern changes, or we do to measure redox changes which are happening inside the cells, or we do to measure the cell death, apoptosis and other thing. So now in that series, today I will give you clonogenic assay. Today I will talk about clonogenic assay. This is one of the molecular assays that is routinely done in any radiobiology lab. Now, why this assay is important, what information this assay gives, that is what I am going to talk about. So now clonogenic assay, as Dr. Jain told, that it is considered as the gold standard assay for radiobiology. Now, the first question, why it is considered gold standard assay? What information it gives? So ultimately, it helps us in making the survival curve or dose response curve. What is dose response curve? Survival of a cell as a function of radiation dose. Whether survival, you are talking about the survival of a normal cells or a cancerous cells, you expose those cells to varying doses of radiations and measure how much fraction of cells are surviving to the dose of radiation. So it helps us in making the dose response curve. But now we have many other molecular techniques. I can simply do MTT assay. Have you heard about MTT assay? So we can do simply MTT, or I can do flow cytometry based PI assay, propydium iodide based assay. But why not? Why only clonogenic assay is considered as the gold standard assay? So the reason is that whatever the other method that are available to measure the cell death, they all are specific to a particular cell death pathway or process. For example, PI-based analysis will help us to know the cell cycle change in the cell cycle pattern, or it can help you to measure the apoptosis. And the timeline of apoptosis or cell cycle pattern changes is just 48 to 72 hours. Because you know that most of the doubling time for the cell varies from 24 to 72 hours. So we don't allow the cells to go for multiple divisions. We want the cells to just pass one doubling time so that we can capture the changes which are happening in the cell cycle pattern. So if you are doing PI-based analysis, this will help us to know the changes in the cell cycle pattern or apoptosis, which happens in a time frame of 24 to 72 hours. Clear? Now, if you go for MTT-based assay, again, you cannot do the MTT analysis for a long. You can hardly do for 2-3 doubling times. But now, if you talk about radiation toxicity is a very, very complex toxicity. The radiated cell has multiple fates. Some cells are undergoing cell cycle arrest, some cells are undergoing apoptosis, some cells are undergoing autophagy, some cells are undergoing mitotic catastrophe. Now, all these pathways, if you are doing just MTT assay, what will happen? You will underestimate actual toxicity of radiation. Because you will be able to, that method will give you the indications only because of the death happening due to certain pathways. If you're doing PI, again, you will underestimate. So clonogenic assay is the only assay which will help us to know exact toxicity of radiation on the cells. Got my point? So that is why it is called gold standard method. Region. Now, why it will give the exact? The principle of clonogenic assay is that we allow the cells to make a clone. We allow the cells to make a clone. And what is clone? We classify in radiobiology clone as a group of 50 cells. So one cell, unless until it becomes 50 cells, we cannot call it as a clone. So in clonogenic assays, we allow single cells to make its clone. A single cell will make a clone only after minimum 6 to 7, 8 divisions. And that is why what we do, we put single cells into the culture dish, leave the cells for at least 2 weeks so that single cells become a clone. Now suppose those cells are radiated. If the cell is not able to make the clone, you will get less survival. But that cannot be detected by MTT, that cannot be detected by, so a radiated cell, whatever method, whatever pathway it is following for undergoing cell death, that will be detected by clonogenic. Suppose it is dying by apoptosis, that is also going to not make the clone. If it is going to undergo cell cycle arrest, then also it is not going to make the clone. If it is going autophagy, then also it is not going to make. So whatever the death mechanism it may follow, it is not going to make a clone. And that is why it gives the exact toxicity of radiation in a particle on a cell. And that is why we call it as a gold standard method. Is it clear to everyone? So now we'll know the method, how it is done. So it is nothing but the measurement of cell killing. I told you that radiation toxicity in terms of ability of a cells to make a clone. So in vitro, we have clonogenic assay, and in vivo, if you are doing, we have to do the, we have to grow the tumor in the mouse model. So we call it genograft when human tumors are grown in the mice. In situ tumors were syngenic, that Jayant already told, and we monitor the survival. But I am now concentrating on this in vitro clonogenic assay. So this is already I explained you what is clonogenic assay, what is its principle, and why it is called gold standard assay for radiobiology. Clear? So now in brief, so what you do in clonogenic assay, you can take tumor tissue and you can actually make a single cell suspensions. Okay, and now single cell suspension cells are cultured for at least 2 weeks to allow them to make the clone. Okay, now you have a normal tumor and radiated tumor. From there, you will make a single cell suspension after radiations, and you will allow them to make the clone, and then you will determine how much of clones have formed. So this is the basis. You have a single cell suspension. Now these single cell suspensions are seeded onto culture dish, and you put different number of cells. So you know that how many cells you are seeding on a dish. Suppose in one disk you have put 100 cells, in another 400 cells, 1000 cells, 10,000 cells. So that many cells have been seeded into the culture disk. Now these cells will be allowed to, you have given radiations, okay, you give them radiations and then allowed to form the colony. Now after 2 weeks, when the colonies will be formed, you have to count those colonies. But how you will do the counting of the colony? Either you can observe those colonies in the microscope, or you can stain those colonies. And for staining, we use crystal violet. So first of all, on the end of experiment, you take this culture disk. First of all, you will fix the cell, and then after you will stain the cell with crystal violet. Crystal violet, what it does, it goes inside the cells. During the fixation, cells gets permeabilized, and the crystal violet goes inside the cells, and it stains the colony. Now you can visually see the colony. You don't even need microscope to see the colony, because colony is a group of 50 cells that can be seen from the eye. So now you can count the colony, how many colonies are formed. So now from this analysis, you will calculate survival fraction. What is the survival fraction? So simply the number of colonies developed divided by number of cells seeded. Suppose you have 400 cells seeded, how many colonies? So how many colonies have formed after your culture? If you will take the ratio, that will give you survival fraction. Now you tell me, suppose you have a cells which you have not radiated. Now from that bunch, you have seeded 400 cells, and you have allowed them to form the colony for 2 weeks. Whether you will get 400 colonies? You have seeded 400 cells. Those cells were not given any radiation, and you have allowed them to form the colony. So after 2 weeks, how many colonies you will get? You should get 400 colonies, na? Because you have seeded 400 cells. No, it does not happen. It does not happen. because even without any kind of stress, the clonogenic potential of each cell type, or you can say if you are talking about cell lines, each tumor cell type will vary. This is called plating efficiency. This property is called plating efficiency. What is the ability of a cells to make clone? So plating efficiency is the number of colonies formed divided by number of cells seeded. That is the first parameter that you have to optimize for your cell type. Suppose you are working on a particular cell type, you calculate the plating efficiency of your cell type. That I am seeding 400 cells, how many colony it is making without any stress. I am seeding 800, and that you have to do at least 3-4 different seeding density. It is not that you seed just 400 cells and allow them to form the colony and then count how many colonies are formed. No, you should seed the cells at different density and calculate the plating efficiency. And then you calculate the average plating efficiency. So suppose you have seeded the cell at 200 seeding density, 400, 600, 800. And then each cell density you calculate the plating efficiency. Theoretically, it should come very close to each other, but no, there is always biological variation. So you'll get some variation, and then you calculate the average plating efficiency of your cell type. That you have to do for every cell type. Again, I am emphasizing the plating efficiency for each cell types are different. This is called without any stress. Now, this plating efficiency will be used for making dose response curve when you are actually evaluating the radiosensitivity of that particular cell type. So now when you have to evaluate the radiosensitivity of cell types, you will seed the cells at different number, you give them radiation. Now here again you have to follow one principle. Now suppose here I have given no radiation, 0 gray, here I have given 2 gray, here 4 gray, 6 gray. Now can I seed the same number of cells for different dose of radiation? No, you have to change that, because what will happen if you are taking suppose 400 cells and you are giving 2 gray with increasing dose of radiation, there will be a lot of toxicity, and at the end of 2 weeks you will not be able to see any colony. And you will, so how you will calculate the survival fraction? Because you need certain colonies to be formed so that you can calculate the survival fraction. So you need to vary the number of cells depending upon the extent of toxicity. As you are increasing the dose of radiation, you will vary the number of cells also. Seeding density also will vary. Okay, fine. So after doing all these things, now this is the type of table that you have to draw. Here you have colonies counted, number of seeding density, how much cells seeded, and then you calculate the survival fraction. Survival fraction is this divided by d multiplied into 100. But now this has to be normalized with respect to plating efficiency of that cell. Because even in the absence of any kind of stress, the cells don't form the cell number of colony. So for normalization, then you have developed, so this is how it looks. So this is the crystal violet stent cells. You know, the cells have been fixed and stained with crystal violet. Now you can see this is the effect of radiation. So here you have a less number of colonies, here you have more number of colonies. Can you tell me any other feature that you are seeing in this photograph?
You do carry on.
So can you tell me any other feature that distinguishes these two type of this? Very good. So not only the number of colonies are important, but the size of colonies is also important. Because I told you the definition of colony is that the colony should have at least 50 cells. But during the 2 weeks time point, some colony will have 55 cells, some may have 100. Now you have given radiation, so you see some colonies have become very, very small. And some colonies, see, size of the colonies are also smaller than this, and number of the colonies are also smaller. So you have to observe these two phenomena during the radiation experiment.
That's
What
mean by naked eye.
Naked eye, yeah. If you have the colony of 50 cells, you can see the clones by naked eye. If you have some, suppose the colonies are very close to each other, we use microscope for accurate counting because sometimes colonies may overlap or close. So that is why we take the help of microscope to correct counting. But you can do by naked eye if the colonies are like this. Perfect. So you can see that these are the small colonies, these are the bigger colonies. So now you understand these are the two things that you will observe after the colonies are developed. Now this is the formula, which is that plating efficiency is the number of colonies counted divided by number of cells seeded into 100. So this talks about the clonogenic potential of a cell type. This is without any kind of stress. Now survival fraction formula is that this is when you are doing actual radiobiology experiment. So in those cases you will count the colonies, divide by the cells seeded, multiplied into plating efficiency by 100. So this is a kind of normalization, correction factor. So you have to correct, you have to apply correction factor to your survival fraction data. Okay, so now from this analysis, what you will get? Survival fraction. And now that survival fraction, you have to plot as a function of dose to develop survival plot that we call a survival curve. So this is all I have explained you. These are the steps. It is available in this thing that you can read. But now this is called survival curve. So what is this survival curve? This is, you see, x-axis you have dose of radiation and y-axis you have a survival fraction. Now, if you see, there is one feature. On y-axis, I have put survival fraction in log scale. On x-axis, I have put radiation dose on the linear scale. This is very important. So this is a semi-logarithmic graph. And sometimes you see some of the publications, they will put this also in linear, that is wrong. Whenever you are plotting survival curve based on the survival fraction data calculated by clonogenic assay, always make semi-logarithmic plot, because then only you will get the concept of D10, D50, D37. In linear, you will know those are not correct survival curves. Perfect. So now from these survival curves, you can get certain radiobiology parameters. These are called D10, which Dr. Jain told D0, or no, D37 he told as a D0. Then you can calculate D10, D37, D0, and you can sometimes people calculate D50 also. Now what is this? So what is D10? So D10, from this curve, you can calculate D10 is the dose of radiation which reduce the survival fraction by 1 log cycle. This is the correct definition of D10. So D10 is the dose of radiation which reduces the survival fraction by 1 log cycle. It means, so it is the dose of radiation which kills 90% of the cells. So suppose 400 cells you have seeded. Out of 400 cells, 90% cells have died. So only how many cells are left out? 10%, 40 cells. So the dose of radiation which is needed to that much killing is called D10. Perfect. Very nicely you write down. So D10 is the dose of radiation which reduces the survival fraction of the cells by 1 log cycle. Now D37. So D37 is the same way. It is the dose of radiation. Now Jain told that 1 gray of radiation you are giving and it reduces. That is the only, they have done the exercise for X-ray. But suppose if you are doing for proton, if you are doing for gamma, if you are doing for, then D37 will be different. It will not be just 1 gray. So it is the dose of radiation which reduces the survival by 37%. Clear?
Here, there are two clinical interpretations of it. The D37 tells you about the radiosensitivity of that particular tumor cell. And the D10 tells you about the radial curability of it.
Yes, in that way also you can calculate. But it is basically the dose of radiation which is leading to that much percentage of killing in the cells. Perfect? Any confusion?
Why the 37 number?
30, very good questions. So now the 37 number, I will tell you. See, when you give the radiations, I told you radiation is going to cause both single strand and double strand breaks. Okay. So now it is a probability that suppose in a population 100 cells you have radiated, out of 100 cells, the probability of double strand breaks is just 37%. So this is the dose of radiation in which at least there will be 1 strand double strand break in 37% of the cells. You got my point? I will repeat again. From where the 37 came, it is very important. It is the probability. So when you pass the radiation through the cells, there is a probability of single strand and double strand break. Okay. Now double strand break is only going to leading to cell death. Now it is the dose of radiation which will create at least 1 double strand break in 37%. That is why this dose is very important.
Partially ionizing radiation.
Partially ionizing. Not for. These all concepts are for low LET. High LET, there is not such concept. The curve, I will teach you, the curve will be just linear. What is the difference between the low LET and high LET survival curve? I will teach you. So high LET is a completely different. Most of the radiotherapy principles are for low LET there, because high LET is just emerging. Now we have got the proton here. Now means these things are emerging. So we'll be coming more, we'll be knowing more about high LET dose response in future time.
Basically X-rays and gamma rays.
Nama. Yeah. So any other questions here? So you can calculate different dose parameters from survival curve. Now these are the survival curves, but we have to give them some mathematical modeling to that. Then only you will get all those parameters. So for fitting those survival curves, we have different mathematical models. Now, this is just low LET what service is saying, that low LET, these all survival curves. In low LET, you have a mainly damage by free radicals, and high LET means dense ionization. So that is a different, that curve is a different. Whatever we are talking is for low LET. Now, how you are going to fit this survival curve? So we have, for that, we have a theory. This is called target theory. So target single hit theory and multiple hit theory. These are the two theory which actually developed mathematical model to fit those survival curves. Now, what is this single hit theory? So according to single hit theory, entire cell is considered as a single target. Entire cell is considered as a single target. So when radiation hits a cell, it is going to either kill or not kill. So it's a single target, it is being hit, either you get a kill or it will not get, the cell will not be killed. Now multiple hit targets believes that the cell has a multiple targets, and when radiation hits the cell, it can hit to different targets, and different targets will result in the cell death. So now multiple hit targets led to the development of linear-quadratic model, and single hit theory led to development of linear theory, where survival is directly proportional to the dose of radiation. S is equal to E alpha D, you know. So this is the linear relations. Survival fraction is linearly related to the dose. So for this, the curve will come. If you plot a curve, so you have y-axis like this, the curve will be just linear. Now, if you take linear quadratic, it will become hyperbolic. You got my point now? I don't have this thing, so I'm drawing with this one. Okay. So this is multi-target model in which you have multiple target inside the cells, and because of the damage to multiple target, you have a cell death. So definitely here you will not get the linear response. Now, earlier when radiobiology experiment started, at that time that single hit theory came into picture, and it was, you know, dominant theory. People were believing that always we will get a linear response. But multi-target model later on developed, you know, then they realized that no, there will be multiple target. Like I told you, DNA is also target, protein is also target, lipid is also target. And because of multiple target model, that linear model developed into linear quadratic model. So this is linear hypothesis, linear quadratic model. Linear hypothesis, I told you that there will be always, there is a single target which is considered as a DNA, and because of the damage to DNA, the cell will undergo apoptosis, and they will lose the reproductive integrity. Now, in case of my linear quadratic model, now here you have a both linear portion and quadratic portion. So survival fraction is equals to alpha D plus beta D square. Now alpha and beta are the constant. Alpha is the constant for linear damage, and beta is the constant for quadratic damage. And here it is directly proportional to the dose. Here it is directly proportional to the square of dose. Means more dose is required to produce a particular damage. Now, if you want to understand, now we have to understand what is linear quadratic model. So this is a typical. So if this is the survival fraction, this is the dose. So curve will move like this. You have initial phase, it will be linear, and as the dose will increase, it will follow the quadratic model. Now, why it is like that? Why it is a linear and then it becomes quadratic? That we need to understand. So now here, what happens when you give to certain dose range? Always it is creating single stent break and double stent break. Now, single stent break will be repaired, and double stent break, even one double stent break is left out, it will kill the cell. Now, so whatever the single stent breaks are created in this region, they all get repaired by the cell. So whatever death you are getting only because of double stent break. And because since you are getting double stent break, as you are increasing the dose, then the probability of double stent break is increasing. So you get a linear response. But now what happens to this region? Here your exposure dose is very high. So it is creating lot of double stent break plus lot of single stent break. Earlier in this range, single stent breaks were completely repaired. But in this region, now what will happen in the single stent breaks are created into different stent, and they are also now behaving like a double stent. You got my point? So now this stent is broken at here. This stent is also broken at multiple times. So ultimately, single stent breaks are also converting to double stent break. They are not getting repair, and they are also now contributing to the death. So now what will happen? You have multiple target coming into action. Multiple target coming into action. So that's why it deviates from start deviating from the linearity. You got my point? Here, single hit target works. Here, multiple target works. Because here also, it does not mean that here different targets are not hit by. Here you have a creation of both single strand, but whatever single strand breaks are, they are such small in number, they all are getting repaired. They all are getting repaired. But those single strand breaks are non-repairable in this region because they are now getting involved in both strands of DNA and behaving like a double strand. Is it clear to everyone? So this is linear quadratic model. Whatever low LET therapy you will do, your tumor response will be following like this. As you increase the dose, the response will start deviating from the linearity because of the multiple targets being hit inside the cell and their contribution to the cell. Now you see, now this is the dose, and as we are increasing dose, you have a response are different. Now what I have shown here, they are the different energy of radiations. Okay, these are different energy of radiation. So different energy of radiations will have a different kind of survival curves. So that is why this survival curves of clonogenic assay is very important to estimate the radiobiological effectiveness of any radiation type. Suppose you want to do proton therapy, you want to do X-ray, but of different range. Then you have to develop your own radiobiology effectiveness. You have to calculate radiobiological effectiveness of radiation type by this kind of analysis. Now, this curve is very important for understanding low LET and high LET. Now, you see, this is the survival fraction, this is the dose. Now, this is the curve for the low LET radiation, and this is the curve for high LET radiation. Densely ionizing radiation, and this is the sparsely ionizing radiation. Now, in this case, it's always single hit theory. It's always linear. The reason is that because the energy is getting directly densely ionization. So densely ionization means it's very important that you have to understand. See, I told you DNA has a diameter of how much? 20 nanometer. Now densely ionization means you have ionization happening at a time when the diameter of that ionization is more than 20 nanometer. So what it will create? SSB or DSB? DSB. So DSB means linear curve. This is the fundamental. So densely ionization means the diameter of your ionization will be always greater than 20 nanometer. Or sparsely ionization means the diameter of ionization is less than 20 nanometer. And that's why in case of a sparsely low LET, you have a generation of single strand break and double strand break. However, in case of densely ionization, you have mainly double strand break, and that is why it follows the linear response. And that is why high LET therapy is better than the low LET therapy. Apart from that, there will be less side effect because by controlling the energy, you can deposit the energy directly to the cell tumor cells away from the normal tissue. So now you understand that response. And this response is the linear quadratic response, which is valid for low LET. Now here, this is the linear contribution and this is the quadratic contribution. So if you stretch this linear portion, it will go like this. And this is your quadratic portion. Now, if you see this, if you take a straight line from here, okay. Now this contribution is alpha D and this contribution is beta D square, quadratic contribution. Okay. Now the ratio of alpha D and beta D square is very important in radiobiology to estimate the radiosensitivity of cancers. If the contribution of this alpha D is more, or I can say that if the ratio of alpha by beta is more than 1, then what? The cells are radiosensitive or radioresistant? If this contribution alpha by beta is more, then this curve will tends lower direction. See, beta is the quadratic. Quadratic means more dose is required to produce a particular damage. So beta is more than cells are resistant or susceptible. Resistant because you need more dose to kill the cells, na? So beta contribution. See, fundamentally you have to understand. Otherwise, if you mug up, now you will forget. Beta D square. It means double dose is required to produce a particular damage. So if more dose is needed to produce a particular damage, means cells are resistant. And alpha D means single dose is required to produce a particular damage. So if less dose is required to produce a damage, it means cells are sensitive. So alpha is more, cells are sensitive. If beta is more, cells are existing. Clear? So you understand, very easy to understand. Perfect. Now, for radiobiology experiment, and especially in case of low LET therapy, these all survival curves are valid. Whatever mathematical modeling you want to do, they are valid only up to 20 gray. Beyond 20 gray, there is no meaning of making survival a dose response curve. The reason is that beyond 20 gray, everything will be dead. Necrosis, these all things are being followed when the cells follow a particular mode of death. For necrosis, there is no, you cannot fit a mathematical model. Necrosis means bursting of the cells. Everything is dead. Out of 100, everything is finished. So beyond 20 gray, you cannot, there is no meaning of radiobiology. Radiobiology experiments are limited only up to 20 gray. Now another thing, so this is the formula survival alpha D plus V. This is valid for low LET. Linear model is for high LET. This is all I explained. Now one more thing. So now you have understood the why this is linear and this is because here you have a direct damage, creation of more double strand break. Here you have indirect damage, creation of SSB and DSB. Okay. Now coming to radiobiological effectiveness. This is again very important where clonogenic assay comes into picture. You cannot develop radiobiological effectiveness. You cannot calculate radiobiological effectiveness of any radiation by using any other method. Any other method you can't do by MTT assay. Theoretically you can do, people do, but those are not correct. Whatever radiobiological effectiveness factor you will be able to calculate, that is not incorrect factor. If you are using MTT, if you are using PISA, even gamma S2X assay is not going to give you the correct radiobiological effectiveness factor. You have to do the clonogenic assay to estimate the radiobiological effectiveness of your radiation type. How you will calculate? So for that we need one standard. Always radiobiological effectiveness means you want to compare this particular type of radiation with some standard. So what is the standard in radiobiology? The standard is the X-ray of 250 keV. X-ray of 250 keV is considered as a standard in radiobiology. Okay, and with respect to that only we calculate the radiobiological effectiveness of any other radiation. Suppose ACTREC is using X-ray of some energy and they want to compare what is the radiobiological effectiveness of this X-ray, then they have to use this as a standard. Okay, so how you will do what is radiobiological effectiveness? So formula is given, dose in gray from 250 keV X-ray divided by dose in gray from any radiation that you want to compare. But for what? Dose for what? Dose for a particular radiobiological parameter. You can calculate radiobiological effectiveness by using D10 value, by using D37, by using D50. These are the radiobiological parameters that I told you can get from clonogenic assay. And you can use the same radiobiological parameter, you feed D10. So you can modify this formula D10 in gray from 250 X-ray, D10 of the test radiation, or D37 from 250 or D10. So you'll get the ratio, and that ratio will tell whether the test radiation is more effective than the standard or less effective than the standard. So suppose if RB is more than 1, means it is better than better than 250 kV or worse than 250 kV in terms of it's worse because you have got it is less effective than the 250, and if it is less than 1, if this dose greater than 1 means it's better than test, because this should come lesser value. If this will be lesser and this will be more, then it will have a positive more than 1. If this will be more and this will be less, it will become less than 1. So if this is more, means it is less effective. If this is more, then it is more effective. Clear? So that is how you calculate the efficacy of your radiation that you have installed at your facility. You get this kind of graph, and from this is they have calculated the radiobiological. Perfect. Now, this is very important. So there is some relation in terms of, like I told you, test radiation. So any type of radiation is classified based on LET. Either it will belong to low LET or it will belong to high LET. Now, what is the relationship between, in general relationship between the radiobiological effectiveness and LET? That you should know. So this is the typical response. You see, this is the radiobiological effectiveness and this is the LET, linear. Now, if you see radiobiological effectiveness remains constant till up to 10 keV per micron meter. So LET, what is the unit of LET? You know, LET is the linear energy transfer, amount of energy deposited per unit length of the medium. So its unit is kilo electron volt per micron meter. So x-axis is kilo electron volt per micron meter, and y-axis is the radiobiological effectiveness. So if you plot radiobiological effectiveness as a versus of LET, you will get this kind of curve. So radiobiological effectiveness will not so much change up to a LET value of 10 keV per micron meter, and it will peak at a LET of 100 keV per micron meter. And then what is happening? It's going down. Why? Generally, if you are depositing more energy to the medium, you should get more damage. So RBE should keep on increasing, but it's not like that. After beyond 100 keV, it again starts coming down. That you need to understand. It means it is not like that. You can get infinity radiobiological effectiveness. You can get maximum radiobiological effectiveness only up to a linear energy transfer of 100 keV per micrometer. So you increase the energy of X-ray. But if the X-ray is going to deposit more than 100 keV per micrometer, again, tumor response is going to be low. Why it is like that? The reason is that all these low LET damage mechanism depends upon the indirect action. Indirect action means radicals, another thing whatever tau. Now, if you are creating more than required density of radicals in a closed space, the radical recombination may happen, and it will, like water or ionization of water, create H2O plus and electron. Now H2O plus and electron will combine and they will make again water. This is called recombination reaction. So if you are going to create lot of chemical species in a very small volume, there will be recombination, back reaction it is called, and back reaction will reduce the damage. Second is that I told you the diameter of DNA is only 20 nanometer. Now you have created that much ionization or excitation that it has completely occupied that place. Now extra ionization is not going to increase. In fact, because of recombination, it is going to decrease.
That is the extra amount of ionization you are wasting it.
Wasting it.
It is more than the 20 angstrom diameter. So this phenomenon is called as a overkill effect. And that's because of these are the two reasons why you can have after 100 keV, after it peaks at 100 keV, it starts falling.
RBE is going. So this is another important concept in radiobiological effectiveness. So this is the same thing. By picture, you can see that this is the DNA, which has a diameter of 20 nanometer. Now this is the sparse ionization. If you are increasing the energy, it will get converted into dense ionization. Now at this stage, you have reached the peak. Beyond this stage, you are wasting energy, and you will not. There will be recombination. It may have even lesser damage, and that is why you start getting response like this. Perfectly. So other molecular assays I will be dealing in the next lecture. This is the one method in which you are calculating the cell killing, which is called clonogenic assay. How it is calculated, what are the interpretations you can get, what is the difference between a low LET and high LET survival plots that I have taught you. Now there are other methods, like I told you, flow cytometry based methods. Then there are methods for estimating DNA damage, gamma H2X assay. Then redox changes, how you can estimate the radiation-induced reactive oxygen species. I am talking a lot about the generation of reactive oxygen species, but how you are going to measure? Those all also come under molecular assay. Detection of reactive oxygen species inside the cell, detection of antioxidant inside the cells, which are going to scavenge the reactive. Then monitoring cell cycle pattern changes by PI assay, monitoring apoptosis by Oncogenes with PI assay. Then DNA damage analysis, how you will distinguish single strand break and double strand break. That is a major challenge in radiobiology. One of the famous means anybody who is going to do PhD in radiobiology, they have to differentiate between the single strand break and double strand break. How you are going to do? So they are molecular assays, and I will be teaching you in my next class about those molecular. Clear? Now you can take.
All right. Thank you, Amit, for that exhaustive lecture on clonogenic assays. So I'm going to talk about the 5 pillars of radiation biology. which is important for us clinicians, clinical radiation oncologists. And I won't be ashamed to say that I'll be repeating a few stuff which Dr. Amit has already spoken about. Yeah. And it's always better to repeat things because it gets hammered into your brain and you will tend to remember all these
You
things.
tell them it used to be once upon a time 4 hours.
Yeah, I'm going to talk about that. Yeah. So when we were students, about 20 years back, we were talking only about the 4 hours of radiobiology. Then it became 5 hours of radiobiology. Now in the 21st century, now it is 6 hours of radiobiology. I don't know how many more hours will be added to it. Well, there are something that re-irradiation is also another hour.
Okay.
All right, I'm going to talk about the 5 hours of radiobiology and also give the concepts and the definitions of RB, LET, and OER and their clinical relevance for the clinical radiation oncologists, right? So as in my previous lecture, I gave you that schema of all the 5 hours and then added the 6th hour of radiobiology, which are the pillars of fractionation in radiation therapy. So what are these actual the hallmarks? They are nothing but molecular, cellular, as well as tissue processes that will affect the radiation on not only cancerous cells, but also the normal cell. So remember, when we will be talking about radiobiology, we'll take cancerous cells and normal cells together. We'll see their effect on both the cancerous cells as well as the normal cells together. And these 5 hours of radiobiology are the processes that will affect both the cancer cells as well as the normal cells simultaneously, okay? And these hours of radiobiology will ultimately determine the effect of radiation treatment on clinical patients. Remember, what we want to do, we want to have the maximum therapeutic ratio. Using these 5 hours, we are going to basically determine the outcome of radiation treatment so that we have the best therapeutic ratio, okay? And this is how, by balancing the complication rates and the tumor control, all right? So initially, as Dr. Sareen said, we started when I was a student, we started with 4 hours. They were considered the hallmarks of radiobiology, and it was given by Withers, Rodney Withers. I had the opportunity to see him in one of the conference, and I was a young student at that time, and he was 85 years old. He's now dead as well. So he gave the 4 hours recovery or repair, and that pertains mostly to the normal tissue, but also to that of the cancer tissue, but it is more important to that of the normal tissue. Second is redistribution, also called as reassortment. This is more important for that of the tumor cells. Reassortment or redistribution of that of the cells in the different phases of the cell cycle, okay? Third is the repopulation, more important for the normal cells. Repopulation after giving radiation is basically a compensatory mechanism of the normal cells, but can also happen in that of the tumor cells, and that is detrimental. If repopulation happens in the normal cells, it is favorable because you're trying to repair the damage which has happened in the normal cells and the tissue comes back to its normal homeostatic position, right? And the last one is reoxygenation. Very, very important and most important only to the tumors, okay? These are the 4 basic hours of the radiobiology which was given by Withers. Then came Gordon Steele. He gave the 5th hour, which is the intrinsic cellular radiosensitivity of a given tumor. We have been talking about that hour. Dr. Amit told about D0, D37 and everything for sparsely ionizing radiation. This is the one. That gives the intrinsic radiosensitivity of a given cell. It can be a tumor cell or a normal cell, okay? And then last one is the 6th hour, which is reactivation of the immune response to radiation treatment. Not much is known about it. We are in the very formative phase of understanding this particular process of immune reactivation after ionizing radiation treatment. Okay, this one I had shown in my previous listing. Intrinsic radiosensitivity, repair/recovery. Repair is for the normal cells, I have told you. So recovery is basically for the tissue, normal tissue. So when you go from cells to tissue, we call it as repair of the cells and recovery of the tissues. All right. Then reoxygenation for tumors, then redistribution for the tumors into the particular phase of the cell cycle, which is very important, and then repopulation, okay, of the normal cells. And last but not the least, now we are understanding is reactivation of the immune response following ionizing radiations. All right, this I've already spoken: repair, reoxygenation, redistribution, repopulation, and intrinsic radiosensitivities. Okay. All right, now let's go into details of that. The first R is intrinsic radiosensitivity, which was started, although it came late. Yeah, question? Is it not? Just a minute. Our slides are not changing. Sharing the intent. Screen sharing. Okay. Oh, this one. इसको share करना. Yeah. All right. I'm sorry. I just started with the 5 hours, 5 plus 1, 6 hours of the 6 hours, which are the hallmarks of radiation biology and the pillars for clinical radiotherapy. So now I'm going to start with intrinsic radiotherapy, although intrinsic radiosensitivity, although it was not the initial one, but it's an important one. And what does intrinsic radiosensitivity tell us about? It tells us about the degree of a tumor or a normal tissue to regress after giving a dose of ionizing radiation. And it depends upon so many factors. And it depends upon the clonogenic capacity of that particular tumor or the normal cell, depends upon its proliferation or the reproductive rate, how much mitotically active that is, its metabolic rate, and the type of tissue also. Apart from that, intrinsic radiosensitivity will also depend upon the presence or absence of oxygen or hypoxia in that particular cell. And last but not the least, the radiation dose which we are giving to kill that cell. Okay. So just to understand, cells which have got a fast growth or a highly proliferating rate or high metabolic rate are considered to be highly intrinsically radiosensitive. And they are mostly for tumor cells. All right. There are certain tissues in our body which are similar to the tumor cells. They are also intrinsically radiosensitive. All right. Like, for example, the bone marrow, the testicular cells, okay, the intestinal cells, they are also having a high metabolic rate, high proliferation rate, similar to that of the tumor cells, and they are also considered intrinsically radiosensitive. Just go to the other extreme, where cells are slowly growing, proliferation rate is slow, the metabolic rate is slow, okay, more of hypoxia in them, they are basically intrinsically radioresistant. Intrinsically radioresistant tissues are mostly, as far as clinical radiotherapy is concerned, are mostly for basically kidneys, lungs, spinal cord, and those. Some of those are the examples of intrinsically radioresistant tissues. Okay. Now, again, when I talk about intrinsically radiosensitive, intrinsically radioresistant, they are very relative terminologies. Okay. It's not an absolute terminology. Remember that. Okay. Even if they are radioresistant, that means at a higher dose, they will still have that effect. For radiosensitive, at a lower dose, they will have that particular. Okay. So it's a relative phenomenon, not an absolute phenomenon. Please remember that. So these are the examples. Now, you are seeing on the x-axis are the single dose radiation exposure which we are giving. And these are yellow, and you can see that pink and red ones are the mild, intermediate, and severe effects of radiation treatment. Okay. So the ones which are done in the blue, they are mostly intrinsically radiosensitive cells. Okay. Ones which are here, the growing bone, the breasts, the islands, testes, these are intrinsically radiosensitive. Okay. while the adult bone is intrinsically radioresistant. That means an adult bone, if it's intrinsically radiation, it can basically tolerate large doses of radiation. Okay, and you won't get that toxicity. But after a certain dose, you will definitely get that toxicity. Okay, intermediate, you'll get a toxicity at intermediate doses of radiation treatment. Okay, and intrinsically radiosensitive, at very low doses also you'll get those side effects. Okay, that is the clinical connotation of intrinsic radiosensitivity. All right. Next is the repair. So repair for us clinical parlance is more important for normal tissues, but it is also important for the tumor. So tumor and normal cells can repair, but the way they repair and to the level that they repair is different. And this is what we exploit as radiation oncologists, right? So tumor and normal cells differ in terms of repair after radiation-induced damage. That is because the reparative process of the tumor cells are defective, okay, and they are inefficient. On the contrary, the reparative process of the normal healthy cells is very efficient. So normal cells after ionizing radiation tend more to compensate and get repaired. But tumor cells, because of inefficient repair process or a defective repair process, they're not able to repair. And that is what we exploit to kill the tumor cells and at the same time prevent normal tissue toxicity. When we are talking about repair, there are different types of repair. This is the new terminology I'm now
slide. I think what I'm seeing on YouTube, you all just check. I'm seeing the old slide.
Still now it's not going.
Yeah.
Why is it?
One second.
No, once they told me.
No, it is still.
No.
See, just check one of them. Just check some of you on YouTube live, whether it is the previous talk is there. It is showing this slide. Yeah? It is showing previous one, no? That's what I'm saying. Yeah. Yes.
We just did
Previous
that.
one was stop share. One second. No, so it is seeing the camera is yours, but the slides are.
Now it is there.
No, they will tell. Yamima, you tell. Eh?
It came now?
So his slide is coming.
Now it's coming. Okay, just a recap. For all of you who could not see that, I'm so sorry about it. Apologies for that. So this is about the 5 hours plus 1 hour, the 6 hours of radiobiology, which are the pillars of clinical radiation therapy. And majority, some of them are important for the tumors and some of them are important for that of the normal cells, but there is not a discrimination between them. Any type of R can be for a tumor, but also for a normal tissue. Okay. So the first R which I have spoken about is intrinsic radiosensitivity. It is important both for the tumor cells as well as for that of the normal cells. And I've told you because the reasons why they can be each type of cell can have differential intrinsic radiosensitivity. Okay, depending upon the metabolic rate, the proliferation rate, the growth rate, presence or absence of hypoxia, and many other things which I've alluded to already. The next one is the repair process, right? I'll go back again so that others will know about it. So I said that tumor cells and the normal cells have repair, differ in terms of their repair after a given dose of ionizing radiation. And why they differ? It's because of in the tumors, they don't completely repair because they have got an inefficient repair mechanism. They've got a very defective repair mechanism. On the other extreme, the normal cells have got a very efficient repair mechanism. They would always try to compensate for the damage which has happened to them by ionizing radiation because of their efficient repair mechanisms. And this differential between the tumor repair capacity and the normal tissue repair capacity is used by us radiation oncologists to kill the tumor cells and have cure the tumor, okay, with minimal normal tissue toxicity. So since I've spoken about repair processes, in radiobiology parlance, we talk about 3 different types of repair processes. One is called as the potential lethal damage repair. Second is the sublethal damage repair. And third is the lethal damage. It doesn't repair. Okay, it's only the lethal damage. What Dr. Amit has spoken about is the lethal damage, which is very important. When you have potential damage, potential lethal damage, when you have got sublethal damage, you can have the repair process. Okay, so and that has got a bearing on radiation therapy and also on the dose response. All right. So potential lethal damage, the repair happens only when it depends when the cells are in a given environmental condition after a ionizing radiation has been given. So in a normal situation, the damaged cells will not repair or die. Okay, but when you reform that environment system, as if you're doing it in a flask, you give the radiation and then keep that cells in that environment of the DMA, whatever media, culture media, you're providing that nutrient media. So it has undergone a potential lethal damage, but because you're modifying the environment within the cell, around the cell, by giving them nutrients, they're coming out of that particular potential lethal damage and repairing. And this particular process is called as potential lethal damage repair. Potential lethal damage repair is mostly not seen everywhere, mostly in in vitro conditions, but there is one particular tumor type which shows potential lethal damage repair, and that is malignant melanoma. Okay, that you have to do. So this is how the cell survival curve looks like if there is a potential lethal damage repair, right? And this is how you can do it. You do the PLD, PD experiment. So that's the mnemonic. For potential lethal damage repair experiments, you do a plating delay experiment. So give ionizing radiation, keep the cells following ionizing radiation in the same media, which provides that nutrient, and then after a delay of certain time, bring it out. Fix it, fix that. Then you get to find out the potential lethal damage. Okay. Next is the sublethal damage repair, where the death of the cell depends upon the sublethal damage condition. Is called as sublethal damage. So how does the repair happen? The DNA damage, which is sublethal in nature, can be repaired if no extra injury is given to that particular sublethal damage. Okay. And the recovery kinetics, the time of repair will range between few hours. Okay. All right. And this will mostly happen if it's a single strand break, or if there are two strand breaks which are separated by a particular distance and by a particular time. So sublethal damage repair can happen. So experimentally, you give ionizing radiation, all right? There's a delay is there. Cells, you take it from that, put it into another media, okay? And then after some time, you take it out and plate it. The important thing is that the cell survival curve or the dose response curve of potential lethal damage repair and the sublethal damage repair are more or less the same. Both potential lethal damage repair and sublethal damage repair can lead to a particular phenomenon which we call in the dose response curve or the cell survival curve, which is called as a shoulder. Okay, if this is the one thing. This is the shoulder which you will be seeing that in the cell survival curve. And this is mostly seen in sparsely ionizing radiation or low LED radiations or radiations like gamma rays, electrons, and X-rays. And what does it mean in the clinical radiotherapy parlance is that the shoulder or the repair of the sublethal damage or the repair of this potential lethal damage is something very similar to that of the wastage of the radiation. Okay. All right. Okay. So we know now about potential lethal damage repair and sublethal damage repair. Now the third one is lethal damage where no repair happens. Okay. It is irreparable and is irreversible damage. And what you see if in sublethal damage, basically you see a straight line rather than a curved line, as you see in potential lethal damage and sublethal. So that's how it is. And the main reason for this lethal damage at the chromosomal level is because of the development of that the ring chromosomes and the dicentric chromosomes, which are, since he said that double-strand breaks are important for lethal damage. Similarly, at the chromosomal level, the types of chromosomes which are lethal are the ring chromosomes and the dicentric chromosomes. Okay, just to give that, so that you will understand, it's a picturesque view of lethal damage versus sublethal damage. So you are having when sublethal damage happens, when there is one ionizing event causing a single strand break here, okay, and another ionizing event causing a single strand break in the opposite DNA strand. And because they are separated by time, and because they are separated by distance, there is repair is happening. Okay, they're interacting and then repair is happening. Here, lethal damage, one ionizing event is cutting through both the strands. Simple to remember. And that is the reason it is causing lethal damage. So this is a single track action. The other one is a double track action. Okay. So single track action, lethal damage, it's a linear curve. Dr. Amit has said, similar, it's same thing I'm talking about. I'm just reiterating those facts. And then double track action, sublethal damage repair, it's a non-linear or a quadratic. Okay. The third one, redistribution or reassortment. This is more important for that of the tumors. Less important for that of the normal tissues. Okay, remember, everything will be balancing now between tumors and the normal tissues. Any questions here? No questions. Okay, so the tumor cells or the normal cells are always cycling in the cell cycle. Okay, some of the cells, some proportion of the cells will be in the G0 phase, some proportion of the cells will be in the G1 phase, some will be in the early S phase, late S phase, G2 phase, and M phase. Okay, but the importance is that each phase of the cell cycle basically has determines the radiosensitivity. Okay, due to various aspects that will be taken up later on. Okay, just for the understanding purpose, each phase of cell cycle has a differential radiosensitivity. For example, G0 phase, when the cells are sleeping, they are resistant. The G1 phase, where the cells are entering from G0 to G1, they are also resistant, but they are less resistant than that of the G0 phase. Okay. Now come to S phase. Okay, where DNA synthesis is happening, they are more resistant. Okay. Once the cell enters into the G2 phase, where division is happening and the mitotic phase happening, so these two phases are extremely radiosensitive phases. And experiments, clonogenic assays have been done to show the radiosensitivity of various phases of the cell cycle. You can very well see late S phase is magenta color. So you can see that it is like this, where larger doses are required to kill the cells. So it is more radio resistant. Now, if you see the M phase and the G2 phase, the blue color, at lower doses itself, the cell survival curve is following this way. So more number of cells are getting killed at lower doses in the G2M phase. So it is more radiosensitive phase. Less number of cells are getting killed in the S phase, so it is more radiosensitive. Got it? These are very important. So just to give a table for you, G0, G1 phase are resistant to radiation. Early S phase is moderately resistant, more resistant than the G0, G1 phase. Late S phase is the most resistant to radiation treatment. G2M phases are the most sensitive to radiation therapy. So when the normal cell is given ionizing radiation, you can have three effects. One, recruitment happens, where the stem cells of some of the tumors in the G0 phase gets recruited into the radiosensitive phase of the cell cycle. Remember, there are so many checkpoints also there. I'm not talking about those checkpoints, okay? Just to give you an understanding about reassortment, I'm talking about this. Those things are completely different, right? So the stem cells of some tumors in the G0 phase get recruited into that, the radiosensitive phase of the cell cycle once you give the ionizing radiation. So cells are blocked in this radiosensitive phase, and as a result of this, they die. Cells in the G2 phase are highly likely to be killed or sterilized by the first radiation dose, because this is the most radiosensitive phase of the cell cycle. Okay. And then after some time, the rest of the cells will now reassort and progress to the so some of the cells which are killed by the first dose of ionizing radiation, they are in the G2 and M phase. What will happen to the other cells which are in the other phases, which are resistant? No, then they will, before the next dose of radiation, they will be again pushed into the G2M phase, which is the radiosensitive phase of the cell cycle, and again they will be killed by the ionizing radiation. Same thing will happen third ionizing event. fourth, fifth, sixth. That is the very reason why we give fractionated radiation doses. What do you mean by fractionation? When we have tumorocidal dose is 60 gray, I don't give it in one day. I divide it over how many days? 30 days. We give 2 gray per fraction today, 2 gray per fraction tomorrow, 2 gray per fraction day after, same way to 30 days. And we finish off that radiation treatment. One of the reasons is this. This is one of the reasons why we have to fractionate the radiation doses. Next is the repopulation. Repopulation is more important for that of the normal tissues. This is more important to reduce the normal tissue reactions caused by the ionizing radiation. So it is nothing but the renewal capacity of the clonogenic cells, which have the clonogenic forming capacity. It is, when we talk about repopulation, it is at the cellular level. The same thing when we talk about tissue at the tissue level or the organ level, we call it as regeneration. Okay? The repopulation of that of the cells will result in regeneration of that of the normal tissues. Okay? And why this happens? Because of once the repopulation of the cells happening, of the clonogenic cells are happening in the normal tissues, they will ultimately result in that of the regeneration of the tissue. And you don't see, and what will happen ultimately? The end product is that once repopulation of cells, regeneration of tissues, whatever side toxicity the normal tissue has developed because of ionizing radiation, it gets healed. It is because of the regeneration. Okay. So if repopulation happens in the tumor, it is detrimental because it is basically causing the tumor to grow back. But if the same repopulation is happening to that of the normal tissues, it is favorable because it is allowing the normal tissue to regenerate. Whatever toxic side effects, whatever toxicity you have seen in that normal tissue, because of this repopulation and regeneration, it is healing. So that is a good thing. Okay. So you have to understand these things, right? So what are the reasons for repopulation? There are 3 reasons for repopulation, and it's what is its clinical implications. So once the tissue is exposed to the radiation, the cell kinetics, which is actually reminiscent of the normal tissue epithelium, is stimulated. So ionizing radiation stimulates, a compensatory mechanism kicks up, okay, and it stimulates. I'll talk about that later on, okay. In the next lecture, I'm going to. It basically stimulates a compensatory mechanism for repopulation to happen, okay. So how does this happen? There is a regenerative reaction of the clonogenic cells to initiate the repopulation by certain growth factors. One of them is KGF, keratinocyte growth factor. There are many others. I'm just giving an example. Second, there is reoxygenation will happen over the course of multiple ionizing radiations which you are giving. As a result of this, the normal tissue gets regenerated. And third, there is activation of another particular protein. This is an important protein which is called as EGFR, epithelial growth factor receptors, and there is increased signaling after ionizing radiation. And this will also help in the regenerative response, repopulation response of the cell, and ultimately regenerative response of the tissues. So these are the 3 reasons which I've told for repopulation, okay? But there is a time scale of event which is happening, okay? This will happen both in the tumors, this will happen both in the normals. Once you're given ionizing radiation, this repopulation doesn't kick in immediately. It kicks in after some time. Usually, it is around 2 to 3 weeks after giving radiation treatment. Having said that, this sort of phenomenon has been seen with sparsely ionizing radiation only, 1, which are low energy radiation, X-rays, electrons, and gamma rays, and 2, only with particular type of tumors, not all tumors. Squamous cell carcinomas of the lungs, squamous cell carcinomas of the head and neck, squamous cell carcinomas of the cervix. This has not been much seen with other type of tumors. Okay, so I cannot say that this is a blanket statement for all the tumors. But for head and neck tumors and for squamous cell carcinoma, this particular repopulation is detrimental. And this sort of repopulation is called as accelerated repopulation. This ionizing radiation is making the tumor cell to repopulate at a double the speed, which is quite detrimental for tumor control. We call it as accelerated repopulation. But the same accelerated repopulation can also happen in normal tissue cells, which is very good. Okay, so that you have to remember that. So from the clinical point of view, the total radiation dose should be delivered over a controlled period of time. See, if you are able to give that entire radiation dose within that 3 weeks, this is the best thing to happen. But usually we cannot give it in 3 weeks time, because before 3 weeks we are finishing that accelerated, we are basically bypassing that accelerated repopulation. We are not allowing it to happen. But we cannot do it because the normal tissues also take some time to repopulate and regenerate. They take about 5 to 6 weeks time. So that's the very reason why we have to alter our radiation fractionation schedules to basically counteract this accelerated repopulation of the tumor cells. Okay. Next, reoxygenation. So if you make a tumor model, just to keep it very simple, the tumor has multiple compartments. It will have an oxic compartment where the cells are very well oxygenated. Then it will have another compartment which is called as a hypoxic compartment. The third compartment is there is no oxygen, that is anoxic compartment. Okay, so let us see here. The blue ones are the ones which are basically oxygenated. This is the oxic compartment. The inside ones are the hypoxic compartment. Okay, so there is a given particular proportion of oxic cells in that particular compartment, and there's a proportion of anoxic or hypoxic cells in that particular compartment. When you give that radiation, the oxic cells, being radiosensitive, they will be killed. The tumor shrinks. Okay, and then what will happen is that the blood vessels will start accumulating. They will send more oxygen to that of the anoxic cells. The anoxic cells or the hypoxic cells now become oxic. Again, the second dose of radiation will be given. It will again sterilize or kill the oxic cells, which are then again the tumor shrinks. Then again blood vessels are formed. The blood will take the oxygen to the hypoxic cell. The hypoxic cells become again oxic. So this is a vicious cycle. Okay, and this is the second reason why we should fractionate radiation, because every fraction of radiation treatment will reduce the proportion of hypoxic compartment. Okay, and this will ultimately lead to that of the tumor cell killing. Okay, I'm not going to talk about that. All right, so this is done. I've shown you how it happens. So empirical observation is that, and there are various ways to understand the oxygen carrying capacity of the tumor cells. There are so many in vitro as well as in vivo assays are there. I will talk about that later on in the other next in the future classes. Okay, so there is empirical observation that oxygen levels in the tumors can be enhanced in the period after radiation. Why? Because the tumor shrinks, blood vessels are formed, they carry oxygen to that of the hypoxic cell, they become oxic. Okay, this particular process is called as reoxygenation. Reoxygenation is very important for fractionated radiation therapy. Okay, and the differences in reoxygenation of originally hypoxic tumor cell and the normal tissue cell is again very important for the fractionated art. Okay, usually tumor cells are more hypoxic than the normal cells. Okay, therefore the effect of fractionation is not much for normal cells as opposed to that of the tumor cells as far as oxygenation, reoxygenation, and hypoxia is concerned. Not talking about repopulation. Okay, all right. So the fractionation sensitivity for normal cells is slightly less as opposed to that of the tumors as far as hypoxia is concerned. And during this fractionation course, whatever lethally damaged cells are there, they are removed and the blood supply increases. I've explained that. And this takes the oxygen to the hypoxic cells, make it toxic. Again, the next course of radiation will come. Again, that will kill the sterilize the tumor cells. Okay, so the initially radio-resistant hypoxic cells slowly become reoxygenated and become sensitive to radiation, thereby shrinking the tumor and producing the cure. Okay, or controlling the tumor. Okay. Last is the sixth R of radiobiology. It is the reactivation of that, the immune system. It's more important for that of the tumor tissue. We don't know much about it. We are still studying it. We really don't know whether it is beneficial or not, or whether it is harmful. But going by whatever research has been done, it's like a double-edged sword. It is both beneficial as well as it is actually, it can be worse also. Okay. So when a tumor is irradiated, the tumor has a tumor microenvironment. Okay. It is not only the tumor, it will have a stromal cells and all those things will be there. The tumor microenvironment is exposed to the ionizing radiation. What will happen is that it will not only cause a local effect, but it will also cause a systemic effect, because there will be lots of immune cells that will be released from that of the tumor microenvironment. They will go into a systemic circulation and can cause systemic effects elsewhere in the body. If there is a disturbance in the balance between immune suppressive and immune stimulatory effects within the tumor microenvironment, then it can lead to certain biological changes. Okay. So this again depends upon that of the type of radiation which we are giving and the doses of radiation which we are giving and the radiation protocol. When we are giving very high doses of radiation, especially when we are giving very high doses, like more than 6 gray or around 10 gray in stereotactic radiosurgery or stereotactic body radiotherapy, where we have to ablate that tumor with very, very high doses, these type of radiation doses can have an anti-tumor effect by immune stimulation. But lower doses can be detrimental because it can basically activate the immune suppressive environment of that the tumor also. So they can have a pro-tumor immune reaction. All right. So this is just to show how this thing works basically. When you are giving ionizing radiation to that of the tumor, this will again depend upon that of the dose which we are giving. At high doses, it can cause immune system stimulation by basically when the tumor cell dies, it basically expresses certain proteins called as the DAMs. These are the examples of the DAMs, and that will basically result in an inflammatory signaling process. And this is a cascade of process which will start. And this will have an anti-tumor response, resulting in activation of various types of immune cells, especially the NK cells, the CD8 cells, the CTLs and others. Even the dendritic cell activation will be there. And that will help, that will result in tumor elimination. This sort of process basically results in tumor elimination. But if the doses are less, this can actually activate the immune suppressive microenvironment. Okay. And this immune suppressive microenvironment can be because of increased expression of checkpoints like PD1, okay, or PDL1, CTLA4, and all those things that can result in basically tumor propagation. So it's like a double-edged sword. All right. So remember that we are still learning about it, and this is the 6th hour of radiobiology as of now. All right. So there we finish all the 6 hours. Any questions here? Okay. Next, I'm going to talk about the 3 fundamental principles of radiation therapy, which is linear energy transfer, the relative biological effectiveness, and the oxygen enhancement ratio. Again, all these 3 things are important as far as clinical radiotherapy is concerned. Okay. So Dr. Amit has already spoken about relative biological effectiveness. I'll just go through that and I'm going to talk more about linear energy transfer and the oxygen enhancement ratio. All 3 are interrelated. All 3 are interrelated. Okay. One will basically affect the other. The second will affect the third one also, and vice versa. Okay. It's not moving. Okay. What is so linear energy transfer and relative biological effectiveness, RBE. So when you talk about ionizing radiation, they are spatially distributed. Okay, the spatial and that is one of the quality parameter of the ionizing radiation. And when the ionizing radiation passes through a biological material, it gets absorbed and it basically tracks through that biological material. Okay, and we really do not know what will be the pattern of its track. Okay, it can be anything. All right. So a radiation quality is determined by this particular parameter called as linear energy transfer, and that will allow us to discriminate between different types of radiation. And it is one of the factors which can basically determine the accuracy of radiobiological predictions. All right. So Zirkle gave this particular terminology called as linear energy transfer. And it is in simple terms, it is nothing but the amount of energy which is transferred per unit length of the track it traverses. Very simple. Okay. And its unit is, as Dr. Amit said in his previous lecture, can anybody again, do you remember that? What's the unit? Kilo electron volts per micrometer. Okay. It's the amount of energy per unit length. Okay. And it accounts for the density of energy transfer along the track of that charged particle within that biological material. And this includes not only ionization, but it also includes excitation. And this happens till the end of its range, of that particle. Okay. So linear energy transfer will account for the density of energy transfer till the end of its range, and is denoted by kilo electron volt by.
And it might change in its track.
It might, that's why it's uncertain. It is uncertain. That's what. So it is one of the methods to predict its quality by the way it will proceed within the biological range. Okay. It is important for us, not only for the biologists, but for clinicians also. So that's what is important. How LET also determines the fractionation. Okay. So high LET radiations like neutrons, protons, carbon ions, direct damage, directly acting ionizing radiations. So the cell survival curve will be straight. Low LET radiations, they will be curved. And why they will be curved? Because of what type of repair I have told you, sublethal damage repair will be happening. Okay. And this, because low LET radiations are fractionation sensitive, you have to fractionate the radiation dose, okay, to get a given biological effect, as opposed to that of the high LET radiation, where you do not actually, which is theoretical, okay, do not have to fractionate to get that particular biological dose. In practical sense, for high LET radiations, the number of fractions can be reduced. Until it can be, I'm not saying it should be reduced, can be reduced, okay, to get the same biological effect when you have larger number of fractions for low LET radiations. Okay. So as I said, there is few fractions are sufficient with fractionated high LET radiation, because they will have little normal tissue sparing after that. Right. Now coming to what is RBE. Again, I'll just go through that definition. It is relative biological efficiency or effectiveness also, and the ratio of the physical doses relative to the reference radiation dose. Dr. Amit said 250 kVp. Yes, it is one of the standards, but people also take cobalt-60 also as a reference standard, required to given that with particular biological effect. Okay, like it can be a D10, D37, D50, anything. Okay, but the biological effect or the biological endpoint has to be the same. Okay, so if a new radiation has a RB of more than 1, then less dose is required to produce that effect. So typical RBs for protons is between 1.1 to, it's 1.3 they say, but it can go up to 1.7. The range of the RB is basically of protons is between 1.1 to 1.7. For neutrons, it is even more higher, 2 to 3. And for carbon ions, it's even more higher, it's 4. So RB is variable. And when it is at the maximum, when the RB is maximum, the dose per fraction can be reduced. You can reduce the dose per fraction. So coming to the relationship between LET and RB, again, I'm reiterating all these things already has been spoken about by Dr. Amit. At LET less than 100 keV per micrometer, as LET increases, RB increases. But beyond 100 keV, as LET increases, RB does not increase, rather it reduces. Who is going to give me the answer? Again, just to remember, just to reiterate that. And the second one is the overkill effect, because more number of ionizations are happening within that close 20 angstrom DNA double helix diagram. Okay. Yes, that is just to show this particular overkill effect. You are seeing this when you are having a higher LET of 200 keV per micron, more number of ionizations are happening within that particular 20 angstrom. So that much amount of ionizations is getting wasted. And as a result of this, RBE decreases. And this is what is called as overkill effect. So you can very well see as the LET increases, the RBE increases. Peak is at 100 keV. And then after 100 keV, it reduces because of this particular phenomenon. Okay. What are the factors on which RBE depends? So radiation quality, photons versus protons. It's a no-brainer. You can very well say that high LET radiations have got higher RBE, low LET radiations have got lower RBE. Depends upon the type of tissue and the biological endpoint which we are choosing. Since RBE varies with dose per fraction and also dose rate. We are not brought in this dose rate thing now. Usually this dose rate thing comes, the terminology comes more in brachytherapy when we are giving radiation internally, okay? So for external radiation, we talk about dose per fraction. For brachytherapy, we talk more about dose rate. So both these factors basically can vary your RBE, okay? And it varies considerably throughout that irradiation field, okay? Especially for photons and also for that of the protons. Then oxygen effect is less pronounced for radiations possessing high RBE. So the influence for those radiations which are high LET radiation, densely ionizing radiation, the influence of oxygen is less. So that is what is coming out from this particular statement. The oxygen effect is not important for high LET radiations, for those radiations which have got a high RBE. And it's also, but as I said, from that particular graph, the relationship between RBE and LET is quite complex. It increases first, peaks at 100 keV, falls down again because of the overkill effect and the recombination effect. All right. Again, what is the importance of it for dose per fractionation? As I said, at low doses, high LET radiation is very effective in killing cells, while low LET radiation is ineffective. Okay. At higher doses, the difference between high LET and the low LET radiation actually comes down a lot. Okay. There's not much of a differential is there. But in case of at low doses, there's a lot of differential between high LET and low LET because of the high RBE of the high LET radiations. Okay. And then as at very high doses again, RBE is no longer dependent upon the dose. Even at very low doses also, because the RBE is very high, you can get that cell kill at very low doses also. But this is a very theoretical phenomenon. This graph goes up to 0, but it never happens that way. But the crux of the statement is that when the RBE is at the max, the doses required for that particular biological endpoint is very low. Okay. And as RBE increases, dose per fraction to get the same kill reduces, as told, and it approaches 1 from this particular graph. Okay. So this is very important for us clinicians. So when for highly densely ionizing radiation where the RBE is very high, we can reduce the dose per fraction. Next, coming to oxygen enhancement ratio. So this is an important parameter, oxygen effect in radiation therapy, and the influence of oxygen on the tissue biological response it differs with different qualities of radiation. Okay, high LET radiations have low oxygen effect. Low LET radiation have got higher oxygen effect. And this particular influence of oxygen on the biological response of the tissue is represented by this particular terminology, which is called as oxygen enhancement ratio. The oxygen enhancement ratio is the influence of oxygen effect on the irradiated tissue. And it is given by this particular formula, dose that produces a given biological response under hypoxic condition divided by the dose that produces the same biological response under the aerobic condition. The biological response again remains the same. It can be a D10, it can be a D37, it can be a D90, whatever, but it has to be the same both for the numerator as well as the denominator. Okay. So what happens? What is oxygen? How is it related? OER is related to that of the LET. So as LET increases, oxygen enhancement ratio reduces. What does that mean? the dependence on that of the oxygen for the cell killing is less for high LET radiation as opposed to that of the low LET radiation. Okay, so what is it? It says that LET is high. Okay, so if LET is high, then OER is low. And if LET is low, OER is high. Okay. So these are this table actually shows the relationship between LET and the OER. For low LET radiation, it is OER is about 2.5 to 3.5. And for intermediate LET radiation, LET, as for example, fast neutrons, it is about 1.6. For high LET radiation like alpha rays, protons, and carbon ions, it is 1. Okay. So less is the higher is the LET, less will be the oxygen enhancement ratio, and less will be its dependence on that of the oxygen within that tissue. Understood this? Then OET also, as I said, radiosensitivity also depends upon the cell cycle. The OER also is also a function of that of the various phases of that of the cell cycle. And this particular thing has been only done in sparsely ionizing radiation, especially the X-rays. So the OER for S phase is about 2.8 to 2.9, which is the radioresistant phase of the cell cycle. And OER at the radiosensitive phase, which is the G2M phase, is between 2 to 2.4, which is more of the radiosensitive phase. So if you go by the various phases of the cell cycle, the OER will be the highest for S phase, followed by G1 phase, followed by the least in the G2M phase of the cell cycle. Meaning that the requirement of oxygen for this phase of the cell cycle is quite less as to kill that particular tumor cell, as opposed to that of the S phase where the requirement of oxygen is very high. Okay. And this is how all three are related to each other. The different types of radiations, RB, OER, and the LET, basically LET values. You can see that 250 kVp X-rays, electrons, and any megavoltage radiations, their RB is around 1. There are OERs basically go on reducing, and the LET value basically is around 2 to 0.2 to 2. Okay. For protons, it is about 1.1 to 1.3. The oxygen enhancement ratio is 1. And you can see the LET value is between 0.5 to 4.7 again. So there's a large variation in that. For the carbon ions, it is between 1.5 to 5.5. The oxygen enhancement ratio is again 1, which is also similar for that of the fast neutrons, which is OER is about 1. But if you see that for the carbon ion, the LET value is about 1.166. And for the fast neutron, it's quite a bit heterogeneous. We not only talk in LET, we don't talk about fast neutrons for one value. We basically talk about track average and the energy average. So here, the track average for fast neutrons is about 12, and the energy average is about 100. So that is the relationship between LET, RB, and OER. Okay, and this is how the curves are, the shape of the curves are for the LET, RB, and the OER. So again, RB goes on increasing, peaks at 100 keV and then reduces. OER also does the same thing. At around 100 kVp, it goes on reducing. Okay, that's how the relationship between OER, RB, and LET is. And that's how these relation LET, RB, and OER allow us to modulate our radiation treatment, especially in terms of fractionation. This is just to show how as the quality of the dose distribution improves, even with low LET radiation, you have got poor quality of dose distribution in the tumor. But with protons, the quality of dose distribution is very high. Okay, although the RB is not very. So this is also important for us as far as radiation therapy planning is concerned when we are using different LED radiation treatment. Just to summarize the last part, both X-rays and gamma rays are sparsely ionizing radiation with low LED values, and just reiterating everything so that it gets into your brain. Some particle radiations, example neutrons, alpha particles, carbon ions, or heavy ions have high LET. High LET radiations are biologically more effective per gray of radiation than low LET radiations. And this is actually measured by the relative biological effectiveness. For most of the high LET radiations at low doses, RB is in the range of 2 to 10. Okay. And RB increases as LET increases up to 100 keV per micrometer and then falls down because of the various reasons which we have discussed above. And OER also decreases rapidly over the same range of. Thank you. Questions?
There are 30 fractions for meta-data. So the fact each 5 fractions to give a rest of the day. So that is required for what purpose?
Okay. So all these breaks of 5 days in a week, you can actually get 7 days in a week also. Because why we give is because of the basically we don't want to work on Saturdays and Sundays. That was started by the Britishers, and that is the very reason. But having said that, the important thing is that we have to give a break of 24 hours for the reasons for repopulation.
Out of the 5 hours, so 6 hours, like which hour is the most important for the break? Like, for the break is the most important for which hour? For the repopulation purpose, for reoxidation purpose?
All are important. So one is important for the normal tissues, and the other is important for the tumor tissues. Like reoxygenation is important for that of the tumor. Reassortment or redistribution is important for the tumor tissues. But repopulation is important for that of the normal cells. Okay. So that all these hours are important. At least these 3-4 hours. Okay. Let us not talk about intrinsic radiosensitivity and immune reactivation. These 4 hours are all important for this fractionation.
So the point which she raised, that although it was initially put as a convenience so that weekends we don't work, only we make the students attend this course on the weekend and some of us faculty. So, but humans don't go to sleep on Saturdays and Sundays. Okay. But it was found that although it was started empirically, but it was found that some of the fractionation, like there was one regimen in the UK, continuous hyperfractionated accelerated radiotherapy chart, 7 days a week without any break. And it was found that, there was excess acute toxicity, and maybe that time that they were getting, there was some repair was happening, which we won't understand. And plus, the overall treatment time is also reduced. So by these 2 days break in between, it is also increasing the overall treatment time. It is not just the time between the 2 fractions, but also the overall treatment time, what we call OTT. So for the tumors, overall treatment time as short, it is good, but for the normal tissue, so maybe some of them, they are sort of repairing. And mind you, all these curves, they are looking at the damage to the DNA, but as Dr. Amit had shown, the damage is not only to the DNA, there is damage to the other molecules also, and they are also causing some biological effect. So this is something which is not very well understood. But if you try to reduce the overall treatment time too much, there could be acute, at least excess acute toxicity.
That is one.
So, okay, so I think now we will next talk.
But having said that, it is important for us to know that we have to wait for at least 5 to 6 half-lives of a normal cell to happen, so as to give another course of radiation, so that repair is there. Okay, so that is one of the reasons why we also give radiation, fractionated radiation every 24 hours.
Okay. So I'll close your PPT.
Do you have another one? You have done, huh?
Oh, your talks are over now.
So actually the molecular assays, he wants to take, it's a very long one. He wants to take it the next time.
Or you want to do it next time, molecular?
Want only 1 or 2 I can talk and then.
Yeah, you can, cover one, and after this you have a talk, then to give a break, at least some part of the molecular assays, although that is as part of advanced radiation biology. Let us see whether screen share. Yeah. You want to come?
One or two.
After this, Jayant, what is your talk next?
The normal tissue radiation.
Normal tissue radiobiology and fractionation.
Fractionation is just the starting.
Just the beginning or just a flavor of fractionation. Okay. So there are a lot of flavors that you are getting. After food, I don't know why this is coming, but it is being live streamed. We can see it very well. And I have become an expert in all this now. In this course, I have learned something. Please.
Fine. So again, I will continue with the molecular assays. Clonogenic we have completed. So now we will move to the another assay that we call it, one of the target is the DNA. DNA is a very, very critical target of radiations, and DNA gets damaged due to radiation exposure. So what are the molecular assays that we have in our lab to distinguish single strand break and double strand break? Because single strand break and double strand breaks are the major things. And apart from that, we have oxidation of nitrogenous bases. So I have not put a slide for the detection of nitrogenous bases. I will tell in brief that oxidation of nitrogenous bases can also be detected in the lab. What you have to do is that you have to isolate the DNA from the radiated cells. You have to digest the DNA, and then you will have to run the HPLC. And then you have the standard of the modified nitrogenous bases, and then you can, from chromatogram, you can actually quantify that how much of oxidation of nitrogenous bases has happened. Another strategy is that now we have antibody. We have antibody available for all the modified nitrogenous bases or oxidized nitrogenous bases, and then you have ELISA kit, already commercially available ELISA kit, and you can detect how much of 8-hydroxyguanine or whatever the modified bases in which you are interested, how much it has produced in the radiated cell. So these are the method, HPLC based method and antibody ELISA based method is used for quantification of oxidized nitrogenous bases in the lab. But I have brought a slide for the detection of single strand break and double strand break in the labs. This is very, very important because we have to distinguish how much damage has happened to the DNA. So now, principle, I will tell you that we can distinguish single strand break and double strand break by using a technique called gel electrophoresis. And I think everybody is aware about the gel electrophoresis. What is gel electrophoresis? So DNA is which charge? Which charge DNA has? It's negative. So DNA is a negatively charged. Okay, and it's not a monomer, it's a polymer. It's a big molecule, it has certain size. So if I will apply electric field, where it will move? Towards positive. And movement will depend upon what? Its molecular weight, because it's a polymer. So this is what electrophoresis is there. Electrophoresis is the separation of polymeric molecule based on its charge as well as molecular weight. If it is a polymer, molecular weight. Now, so this is the typical setup. I think you might have seen. This is called gel electrophoresis. This is horizontal gel electrophoresis. We have vertical gel electrophoresis that we do for the protein, but for DNA we do horizontal. It's not like that we don't do vertical for DNA also. If you want to separate a small nucleotide, then you have to do the vertical electrophoresis. So horizontal gel electrophoresis, we have electrodes. These are the electrodes. So there will be negative and positive cathode and anode, and this is tan. And here we put a electrolytes buffer that will provide the movement, conductions. You will provide the conductivity. And then this is the agarose gel. Agarose, agarose is a matrix in which we create a wells, and in those wells we put the load our DNA. Now the DNA, once you load the DNA will start moving through this gel. How it will move? Because this gel has certain porosity, and that porosity will depend upon the concentration of gels. So depending upon the size of DNA that you want to resolve, you'll make a gel. The percentage of gel agarose may vary from 0.5% to 2.5%, depending upon the size of DNA that you want to resolve. So once you load the DNA from here, and you apply electric field, DNA will start moving through this gel. And once electrophoresis will be completed, then you have to stain that DNA and you have to visualize, you have to document. And this is how the bands, DNA bands look like. Now, one more thing you have to know that since the DNA is moving through the gel and DNA does not have its own color, you put some tracking dye. to see that how far DNA is moving. That tracking dye is bromophenol blue. Okay, perfect. So once your electrophoresis is complete, you do the staining with the dye, fluorescent dye called ethidium bromide. Now, ethidium bromide, what it does, ethidium bromide? So ethidium bromide, you know, this is the principle. This is a dye. This ethidium bromide intercalates between the base pairs of the DNA. And when you excite it with 300 nanometer, it gives fluorescence at 605 nanometer. And that fluorescence you visualize in the form of bands. Clearly? Now, this is how we see. Now, knowing this electrophoresis technique, now our job is that how we can distinguish single strand break, double strand break, and oxidized basis. We can detect even oxidized basis by using gel electrophoresis. Now, the technique that you are going to use here is the simple the principle. So I will give you just step by step. Suppose you want to detect double strand break. It's very easy. Because in case of double strand break, both the strands of DNA are broken at the same location in both the strands. So it means DNA has got fragmented. So now what you have to do, you have to do this electrophoresis at neutral pH. Neutral pH means, you see, you go to back. So this tank buffer is going to be neutral. So under neutral conditions, what will happen? The controlled DNA or the DNA from the unirradiated cells, they will be intact. There will be no fragmentation. So they will move faster or slower? Slower, because they are high molecular weight DNA. There is no fragmentation. There is no double strand break because of the controlled DNA. These are from unirradiated cells. But the DNA from the irradiated cells, and if at all it has a double strand break, it is going to be small molecular DNA. So they will move faster. So in case of radiation, in case of irradiated DNA, you will get more bands because the DNA is fragmented. In case of unirradiated DNA from unirradiated cells, you will get an intact single band. So that is how you detect the double strand break. Very easy to remember. You have to do the electrophoresis at what pH? Neutral pH. Now you apply alkaline pH. You treat your DNA with alkaline pH first and then do the electrophoresis. What will happen? Due to alkaline pH, both strands of DNA will get separated. Now, it does not mean whether it's a double strand break or single strand break. Everything will become, if it is a fragmented, you will get everything moving faster because fragmented DNA will move faster. So amount of DNA damage that you will detect at alkaline pH is going to give you overall damage. Whatever damage, whether it's a single strand break or double, cumulative damage will be detected at alkaline pH. Now, if you subtract the damage from the alkaline pH to neutral pH, what you will get? That is the principle of this assay. Clear? Now, how you will get? So this is what I have put in this thing. All the you will understand it is in this slide. So now, next time you want to quantify the oxidized nitrogenous bases. So for that, you have to use certain enzymes. Commercially, there are certain enzymes available. What they will do, those enzymes cleave the N-glycosidic bond. Remember in the morning, they will cleave the N-glycosidic bond specific to the modified base in which you are interested. You got my point? In my lab, if you come, I have different enzymes. Each of these enzymes is specific for particular N-glycosidic bond of modified base. So what I will do, I will isolate the DNA from normal cells, unirradiated cells. I will isolate the cells from irradiated cells. And now I will digest the DNA with my enzymes. So if there is some modified bases, what it is going to do, this enzyme is going to cleave the N-glycosidic bond of that place. As a result, a purine site will be generated in the DNA. Now, next enzyme I am going to use is called AP endonuclease. And this AP endonuclease will cleave the phosphodiester bond. And now I have converted modified nitrogenous bases into single strand. Clear? So initially you had a modified nitrogenous bases. By using the combination of enzymes, I have now converted modified nitrogenous bases to strand break. And then I will do electrophoresis. So now, please, if you are doing electrophoresis at alkaline pH using N-glycosidase enzyme, then you are going to get what kind of damage? It will have a combination of SSB, DSB plus oxidized nitrogenous base. You got the point? Now, in another assay, you do the same alkaline electrophoresis, but don't use the enzyme. You are going to get only SSB plus DSB. In another experiment, you do the electrophoresis only at neutral pH. You will get only DSB. So now from all these combination, you can know how much DSB has created. If you subtract alkaline electrophoresis minus neutral electrophoresis, you will get only double single strand break. And if you are combining enzyme also and then subtracting, you will get the contribution of oxidized nitrogenous base. Is it clear to everyone? Very easy to remember. So gel electrophoresis is a technique by which you can detect all or distinguish all these different types of DNA damages which happen inside the cell. And I told you that specifically if you want to detect oxidized nitrogenous bases, you can use ELISA-based kit as well as HPLC. Now, so this was all when you are isolating DNA from the cells and then you are doing electrophoresis of isolated DNA. Now, advancement of technology is that we can do electrophoresis of whole cell. Yeah. No, normal cells may basal level of see. If you talk about unirradiated cells will also have some basal modified nitrogenous bases. Okay, so you'll get some basal DNA damage, but after radiation, you have a lot of modified nitrogenous bases. So you'll get more number of AP sites, more number of single strand. So you'll get more damage. Is it clear to you? After irradiation leads to oxidation of nitrogenous bases, single strand break, double strand break, and intra-strand cross. Now, if you want to detect oxidized nitrogenous bases, if you are using N-glycosidic enzyme, wherever the modified nitrogenous bases are there, at that positions, it will create AP sites, apurinic or apyrimidine site. Then you use another enzyme called epi-endonuclease, and those sites will be converted into strand breaks. And then you do the electrophoresis, you will know that, okay, these are the damage because of oxidized nitrogenous base. So control cells will have basal damage. Since control cells are not radiated, they are not going to have very lot of modified nitrogenous base. So now I told you with the advancement of technology, we can do electrophoresis of the whole cell. We don't need to isolate DNA. For isolating DNA, we need millions of cells or you need the tissue. You got my point? But now we can do the single cell analysis. That's why it is called single cell gel electrophoresis. Or in other words, we call it Comet assay. Comet assay is nothing but it's a single cell gel electrophoresis. We are detecting DNA damage of an individual cell. Very powerful technique. You see, when you isolate DNA, there will be a lot of error coming due to your isolation protocol. Sometimes your DNA will get damaged due to problem in your isolation method. But in this method, single cell gel electrophoresis, you can avoid those errors. And here you can detect DNA damage inside a single cell. So technique is principle is same. Same way we have to do the electrophoresis at neutral alkaline by using enzymes, non-enzymes. Principle is same. But instead of DNA, we are doing the electrophoresis of whole cell. So the technique by this is done on a slide, glass slide, on which you do for microscopy. So this is the glass slides. Again, you make a bed of agarose, and in these agarose now instead of DNA, you have mixed cells. So now these are the cells put on a slides. This is in agarose bed. Now you are going to lyse the cells. How you are going to lyse the cells? This slide you are going to put in a lysis buffer. So lysis buffer, what it will do? It will remove the membranes and DNA will be deposited at the location of cells. And then after lysis, you will again neutralize the pH and then you do the electrophoresis. Now, once you are doing these are the DNA, DNA from the single cell. Now, when you are doing the DNA, again the electrophoresis means DNA will move from negative to positive. Now, suppose if you have an intact DNA without any damage, you will get a sphere. But if you have a intact DNA along with the damaged DNA, fragmented DNA, the fragmented DNA will move ahead of the intact DNA in form of a comet. And the entire structure will look like a comet. So these are the fragmented DNA which are moving ahead of the center of gravity. This is the center of gravity of this comet. And this center of gravity is representing the intact DNA, which are not damaged. And this area represents the damaged area. Now, this is the center of gravity of this comet. And now from here to here, this length is called tail length. Each comet has a tail That length is called tail length. This is in micron meter. Because on its microscopic slide, you'll observe everything under the microscope. So this comes in the micron meter. This is called tail length. Okay, perfect. Now, with microscopy, you can know how much amount of DNA is in this tail and in this center of gravity. So, but overall DNA you can calculate and how much amount of DNA is there based on the fluorescence emission. You got the point? Fluorescence, this is the fluorescence. So I can know the intensity, how much intensity is here, how much, and from there we can calculate percent DNA in the tail. So from comet assay, what parameters we get? We get two important parameters. How much amount of DNA is there in the tail, overall entire DNA of the cells. And second thing, what is the length of the tail? Now you multiply these two parameters and you get olive tail moment, OTM. And this is the parameter which will tell how much of DNA is damaged. So if the olive tail moment is more, DNA is damaged more. If olive tail moment is less, DNA is damaged. So intact cells which is not irradiated will have no olive tail moment. And once you radiate the cells, you will get increase in the olive tail moment. Now, just like normal electrophoresis, we are using ethidium bromide. Here also you can use ethidine. So this is the fluorescence of nothing but ethidium bromide, cybergrid. Any DNA binding dye can be used for detection of this method. Okay. Now so olive tail moment, this is the tail moment, this is the parameter that you got it from this analysis. Now suppose I have plotted olive tail moment as a function of time. I have radiated the cells, and after radiation, I did this single cell gel electrophoresis at 5 minutes, 10 minutes, 15 minutes, 2 hours, 24 hours. So now I have the olive tail moment as a function of time. And if I will plot what kind of graph you expect? Did you get my question? I have radiated the cells and I have now done this analysis at different times after radiation. And now I am plotting tail moment as a function of time. So what kind of graph you expect? Yeah, time dependent decrease. So initial 5 minutes, you will have very high tail moment. 10 minutes, it will come down. 1 hour, it will further come down. 4 hours, it will come down, and then it will go and come to the basal. This is called DNA repair kinetics. This is called DNA repair kinetics. Such a powerful technique by which you can know, you can differentiate. Like Dr. Jain told, the sensitivity of normal cells and cancer cells are different, radio sensitivity. How you will do? One is the clonogenic assay, another is this assay. Here you can prove, indeed, you can prove that cancerous cells have less repair efficiency as compared to the statement that he gave. You can actually prove this in your lab, in your PhD thesis, that cancerous cells have less DNA repair efficiency, or in presence of some chemical modulator, what is happening to their repair kinetics. Got my point? So now understood? You can do this analysis, you calculate olive tail moment at different function of time, and then make your own DNA repair kinetics graph. Perfect. So this technique, this is one of the molecular technique which is very important. Now another molecular technique again for the DNA damage is the gamma H2AX assay. Now gamma H2AX, again he has introduced, this is the H2AX is a histone. It's a type of histone molecule. Where is present? Histone is present where? It is a DNA binding protein. It is bound with the DNA. And it is one of the derivative of histone which acts as a sensor of double-strand break. The moment double-strand break is created inside the DNA, I will teach you DNA repair pathway, then I will talk more about this. But at this moment, you have to just listen that the moment double-strand breaks are created, in order to initiate the downstream signaling, the cell has to activate the repair pathway to repair the damage. It recruits some sensor molecule on the DNA. You know, it sends some protein that go and bind to the damaged site so that the repair machinery will come to know where is that DNA damage. And then entire repair machinery will go from cytosol to that site and they will do the repair. So it acts as a sensor molecule and it is recruited wherever the double-strand break is there. And recruited by whom? ATM. ATM is another protein which is a kinase, and that protein does the phosphorylation of this molecule. You know, so this H2AX histone is present throughout the DNA. Wherever the double-strand break has generated, this histone derivative will get phosphorylated by ATM at its serine 139 residue. Now that phosphorylated histone will act as a tag for all the repair machinery to wherever the phosphorylated histone is, the repair machinery will go and sit there and start doing the repair. So now this phenomenon can be used again to study the DNA repair kinetics, because now we have commercially available antibody against the serine 139 phospho derivative of this H2AX. Got it? So now we have antibody. So what we will do? You will do the immunofluorescence. What is immunofluorescence? Immunofluorescence is that use of antibody, fluorophore tagged antibody to study any kind of parameter. It's called immunofluorescence. We are going to use fluorophore tagged antibody to study the parameter in which we are interested. So here we are interested to this is the histone, and we want to detect wherever the phosphorylation has happened. So I'm going to use the anti-phospho-histone antibody, and that antibody will go and bind to wherever it is there. And then after again, I will make a slide and I will look in the microscope. So this kind of images will come. So this is the entire cell, but you can, what is this blue color? Yeah, this is the nucleus of the cell. How we are coming to why she is saying that it's a DAPI stent. So DAPI is again a DNA binding tag. So DAPI will go and bind to the DNA inside the nucleus. So entire nucleus will glow up like a blue fluorescence, because DAPI has a blue fluorescence. So now in this cell, this entire blue region is an entire nucleus. Now in this nucleus, you can see that white dots. Can you see that white? These are nothing but the gamma H2X foci, where the antibody against the gamma H2X has gone and bound, and it has given the fluorescence. Now with microscope, we can count all these dots, which is known as number of gamma H2X foci. And again, that foci can be plotted as a function of time to get your repair kinetics. Now this analysis can be done not only by microscopy, confocal microscopy. You can convert this method into flow cytometry based detection also. Then you can detect cells having positive for gamma H2A foci. Okay, so now you can do this analysis both by flow cytometry, both by confocal microscopy. So this is the typically how we do. So you culture the cells, then you give radiation. This is the control which has not been given radiation. These are the cells which have been given radiations. After that, you fix the cells. Fixing के बाद you do the immunostaining. You treat with the primary antibody, secondary antibody, and then now you detect by the microscope. So this blue is the DAPI staining, and these are, you see, these are the dots you are saying, these are the fluorescence of antibody which is specifically binding to the gamma H2A protein. So now you tell me which cell is having more DNA damage? This one or this one? This one having the most DNA damage, because the number of gamma H2A foci are highest. So now you do this kind of analysis, and now you can have your own repair kinetics. So this is the example now. This is from our own paper. So in which we have done, you see, this is 30 minutes, 1 hour, 2 hour, and 6 hour. Now you see, this is the controls. Can you see any foci? No, because it's the cells which have not been in radiation. Now here you see, we have one molecule which we are developing clinically. So now this molecule, this is a drug molecule. Drug itself is causing any damage? No, very basal, means you can say negligible DNA damage. This is radiation. We have given 2 grams for radiation. Now you see the damage, and then damage is coming down, because the cell is going to repair by itself. Now here I have combined my drug with the radiation, and what has happened? From curve, it has not protected the cell. In fact, it has increased the DNA damage. It means it is working like a radiosensitizer. It's working like it is increasing the tumor cell killing in the presence of radiation. So this is how DNA repair, this is called DNA repair kinetics.
Yes, which you had, this is going to be the last lecture for today. And I'm going to talk about normal tissue radiobiology, especially in terms of pathogenesis of normal tissue responses to ionizing radiation. So as I mentioned before also, radiation is not only important for tumors, but it is also important as far as normal tissue is concerned. And to have the optimal therapeutic ratio or the best therapeutic gain, we have to basically see that the ionizing radiation controls the tumor and at the same time spares the surrounding normal tissue, which is around the tumor. And for that, we need to understand what normal tissue reactions are following ionizing radiation. And in order to understand the normal tissue reactions also, we need to understand the pathogenesis behind these normal tissue reactions. So I'm going to talk about the pathogenesis of normal tissue responses to ionizing radiation. And on the way, I'm going to introduce some new terminologies also. You might want to write them as well. All right. Even before we try to understand the pathogenesis of the normal tissues, the normal tissue cellular system has been again classified into different types based upon their sensitivity to ionizing radiation. The first of the classification is based upon the population kinetics of these cells, and it is called as Casaret's classification. Casaret's classification used to be used earlier for understanding the pathogenesis of normal tissue reactions to ionizing radiations. And Casaret, and along with his colleague Rubin, were considered one of the foremost radiation pathologists. So one of the classification is based upon the population kinetics of the cells which are present in the normal tissues. The second classification is based upon the architecture of the tissue. And that classification was given by Michalowski. It is called as Michalowski's classification. And based upon this classification, that is the classification based upon tissue architecture, we classify the normal tissues into two types. One is the hierarchical tissue types architecture. The other is the flexible type of architecture. Okay. So let's start with the Michalowski's classification of tissue organization. So Michalowski has said in his classification that tissues can be only of two types. Should I go back?
You just check. I'm seeing your passion.
I guess they have to share the slideshow. Share
Something you do, Jen, last time also your this thing had stopped. No, it's not moving, no? One second.
It's moving.
It's moving now?
Okay.
Good.
30 seconds.
No, that I know that. But these are moving, yeah.
All right.
Yes.
So there are two classification systems. One is the Casaret's classification system based upon population kinetics of the cell. The other is based upon tissue architecture, which is called as Michalowski's classification. That is a more simplified classification. And based upon that classification, tissues have been classified into two types. One is a hierarchical type of tissue, and the other is a flexible type of tissue, just to keep it very simple. Right? So what exactly are these? What exactly does this classification system say? So Michalowski said that tissues can be divided into two types. One is the H type, which is the hierarchical type of tissue. The other is the F type, or the flexible type of tissue. What happens in the hierarchical type of system? He divided the entire normal tissue into three different compartments. Okay. The first compartment will have stem cells, which have got infinite capability to reproduce and divide and repopulate. The second compartment was called as the transit cell compartment, where there are differentiating cells, differentiating daughter cells. Okay, they have got a finite capability to reproduce and repopulate. And the third compartment was called as the functional compartment, or the mature compartment, where they do not have any capacity to proliferate or divide. So cells will enter from stem cell compartment, divide into and go into daughter cells, will go into transit compartment, and then our daughter cells will again further differentiate. Daughter cells are also called as the differentiating cells. They will differentiate into mature cells or the mature functional cells, which don't have any proliferative capacity. They have got only the functional capacity, and ultimately the cell is lost. This is the normal homeostasis of a normal tissue. Okay, this tissue has not been exposed to any sort of genotoxic agent, be it UV rays, be it any chemotherapy, be it radiation treatment. This is how the H type of tissue basically functions. There is a hierarchy in that particular tissue. Okay. The second type of tissue is called as a flexible system. There is no hierarchy. Okay. What will happen is that there is only one compartment which will have stem cells, differentiating cells, mature cells, or the functional cells. And then ultimately, what will happen after a particular lifespan of that particular cell, that will be lost. Okay. The difference between the H type and the F type of system is very important. In the H type of system, it is a self-renewing system. You don't require to stimulate it for the stem cells to divide into differentiating daughter cells and then go into the differentiated cells. But in an F type of system, you require some stimulation. The stimulation can be any genotoxic agent to basically allow the functional cells to basically divide and act as a stem cell. Okay, so it is flexibility is there in that type of a tissue. Okay, and when it comes to clinical radiotherapy for us, an H type of tissue is that type of a tissue which is a highly proliferating tissue. Remember what I said in the last lecture? Any tissue which is highly proliferating, highly radiosensitive. Okay, because they are highly radiosensitive, they will develop acute radiation type reactions. I'm talking about normal tissues, okay? So they will develop acute radiation reaction. Come to the other one, the F type of system. These are those tissues which are not highly proliferative. They require a stimulus to basically for division of the stem cells to differentiating cells to that of the functional cells. Okay, so these cells are less radiosensitive and they proliferate slowly and then they tend to have late reactions. Okay, so the H type of tissues are those tissues which will develop when given ionizing radiation, which will develop acute reaction. The reaction can be during radiation treatment, just after radiation treatment, up to 90 days of radiation treatment. Okay. The F type of system are considered slowly proliferating tissues, then having slowly proliferating cells. They are basically late responding tissues or late reacting tissues, and they will have late reactions or what we call it as chronic reactions. Okay. There is a third type of system. Okay. So examples of H type of system, I'm going to talk about that in my next slide, are those which will have which will develop acute reactions, and examples of those tissues which will have F type of system will develop late reactions or chronic reactions. Okay, so there's a complete segregation. Okay, and then there's a hybrid system, a hierarchical system with a flexible system, which will have both the compartments. It will act as a hierarchical tissue also, as a flexible tissue. How is it possible? It's not a tissue which will act as both the things. It's within the tissue itself. There are particular type of cells which will act. One type of cell will act as a hierarchical cell system, and the other type of cell will act as a flexible type of system. Example is kidneys, which has got the glomerulus and the nephrons. The glomerulus will act as a hierarchical tissue, and the nephrons will act like a flexible system. Okay. So if you want to completely differentiate between this H type of system and the F type of system, let us see how it happens. There is a clear-cut segregation in the H type of tissue, wherein there will a compartment will have stem cells. Will have stem cells compartments, transition cells, which are the differentiating daughter cells, and the functional cells or the differentiated cells. While the F type of system, there is no clear-cut differentiation and there is no strict hierarchy, as they rarely divide. The second difference, as I said, you require it's a self-renewing system, the H type of system. It doesn't require a stimulus for the division of that of the stem cells. But on the contrary, the F type of system, it requires a stimulus, wherein the Oh, oh. Yes, it's going to be one resolution.
So how to, yes, floating control, how do we, इसको hide कैसे करना है? So.
All right. As far as requirement of stimulus is concerned, for the F type, you actually require stimulus to stimulate the stem cells to divide. Okay, and then differentiate into the daughter cells, which ultimately get converted into the functional cells or the mature cells, which do not have the proliferating capacity. Okay. So, as far as the proliferating capacity of the functional cells is concerned, in the H type of tissues, there is no proliferating capacity, but for the F type of tissues, it has got infinite capacity to proliferate. And as I said, H type of tissues, these are rapidly turnover tissues, highly proliferating tissues. Okay, so its rapid cell turnover happens in the H type of tissues, while very slow turnover happens in the F type of system. And as far as the time scale of injury is concerned, H type of tissues will show acute injury or early injury, but F type of tissue will show slow or a late injury. Okay, which is chronic injury. All right. The onset of a damage, functional damage, is actually independent of the radiation dose in the H type of tissue. Why? Because it is a self-renewing system. You don't require a stimulus. Okay, so radiation may not, it's not dependent upon that of the radiation dose. Okay, but here in the F type of tissue, it is dependent upon the given radiation dose. Okay. And as far as the dose response relationship is concerned, in case of a H type of tissue, because these are highly proliferating, they're highly sensitive, so we don't see that sublethal damage repair happening. Okay, I spoke about the sublethal damage repair. We don't see that, and that sublethal damage repair tells us about the shoulder of the dose response curve. Here, in case of H type of system, the shoulder is very short because the sublethal damage repair is not happening. Okay, but here, in case of the F type of system, slowly proliferating, more radioresistant, more sublethal damage repair happening. The shoulder which is happening is it is a broader shoulder is there in the dose response curve. Okay. All right. So what are the examples here? The important examples are the intestines, which are highly proliferating tissues, the bone marrow, the oral mucosa, the epidermis of the skin, okay, and the testicular epithelium also. These are highly proliferating tissues, and they basically are normal tissues which show acute or early reactions. The other one is the lung, the liver, kidney, thyroid, and the dermis of the skin. They show chronic or late normal tissue reactions on exposure to ionizing radiation. So now you know about the two types of tissue architecture. One will show early type of tissue reactions. The other will show late type of tissue reaction. But there is a third type of reaction which we call it as a consequential late reaction. I'm going to talk about that later on. Okay. But as far as clinical radiotherapy is concerned, up to there's a watershed line of 90 days. Don't ask me why it is. It's all empirically done. Okay. Up to 90 days, whatever reactions we see are mostly considered early reactions. And these type of reactions are seen mostly in the early responding tissues or the H type of tissues. Beyond 90 days, we call it as late reactions. And these are seen mostly in the late responding tissues or the F type of tissues, liver, lung, dermis of the skin, kidneys, and all. Okay. Let us now talk about the tissue homeostasis under physiological condition. What happens? We'll then go to what will happen once we give the radiation. We need to understand this very well. Okay. Under physiological condition, absolute homeostasis happens in the normal tissue. Forget about any type, early or late. Okay, there will be homeostasis will be there. Okay, and there's a balance between production of the cells and also the cell loss. Once the cells are lost, what it produces, it gives a stimulus to the stem cells, gives a signal to the stem cells, which will start proliferating, dividing. The stem cells will undergo a particular type of division, which is called as asymmetric division. I'll talk about that later. You need to know what exactly is asymmetric division. It's a new terminology, okay? Okay, asymmetric division is that type of a division wherein the stem cell will divide into two daughter cells. One daughter cell will come back to the stem cell compartment, and the other daughter cell will go into the transit cell compartment or the differentiating cell compartment. Okay, in a normal homeostasis, this happens, asymmetrical division of that of the stem cells. Okay, once that happens, the differentiating cells will then go into, after they will have finite proliferating capacity. They will again divide once, twice, three, nth time, and after some time, they will again proceed into the next compartment of the tissue, that is the functional compartment, and ultimately the cell loss will happen. And this will continue in a normal tissue homeostasis. Okay, cell production, cell loss, cell production, cell loss. Okay, this is a physiological situation. In that tissue itself, it will have vasculatures, blood vessels, and it will have also stromal cells, the connective tissues. They will also help the maintaining of this tissue homeostasis, okay, in different ways, by producing certain types of the proteins that I'll talk about later, okay. But what will this type of homeostasis is completely disrupted when you are exposing that tissue to any genotoxic agent. Now let us talk about ionizing radiation, okay. So let us start with the early reactions, okay. What are the sequence of events? When a normal tissue, let us take for example, intestine, which is a early responding tissue, is exposed to ionizing radiation, okay. You're giving ionizing radiation, it will result in certain cellular changes. There will be vasodilation, okay, all right, and there will also be inflammation because of the increased translocation of that of the inflammatory cells towards the site of radiation injury, okay, and this will be basically expressed on the tissue as erythema and other inflammatory changes. Now, this particular thing is called as induction, okay. Now you are given one dose of radiation, but if you stop it, it will directly undergo restoration and heal. But if you continuously bombard that tissue with ionizing radiation, it will go into the next phase, which is called as progression phase. And in this progression phase, what will happen? In this progression phase, what will happen is that hypoplasia of the tissue happens. But why this hypoplasia happens is because of certain subcellular events which are happening in that. Remember my last slide? Normal tissue homeostasis is there. Now, what happens? The normal tissue homeostasis is completely disrupted. What is happening is that there is you're giving ionizing radiation, you're destroying or disrupting the stem cell compartment. There is not enough stem cells produced to replace the functional cells which have got a particular lifespan and they are lost physiologically, okay? What is happening is that you're damaging the stem cells, which is not producing more number of, which is not producing the daughter cells, and these daughter cells are not able to replace the functional cells. As a result of which, hypoplasia of the tissue. There is completely, the tissue becomes smaller, the hypoplasia of the tissue happens. So, most important event in an early stage early reaction is this particular, okay? It is hypoplasia of that of the tissue, and it is because of that of the depletion of that of the cells within the functional compartment, okay, of this hierarchical tissue structure. Then, if you again stop radiation, it will again go, it will start healing, it will undergo restoration and heal, okay. But again, if you're not doing that, you're basically bombarding it few more doses of ionizing radiation. It will go into a third phase, which is called as manifestation, resulting in secondary effects. What is happening? is you have got hypoplasia, okay, which becomes more and more progressive because you're not stopping bombarding that normal tissue with ionizing radiation, right? So it goes on becoming hypoplastic, and then there will be secondary reactions to it. Example is that you got a small ulcer, okay, and then you're going on bombarding that with this thing. There is epithelial denudation is happening, and that ulcer is exposed to that of the surrounding this thing, and it can get infected. So this secondary effects can happen, which can be one of the this thing is infection of that. Okay, and this is called as the manifestation phase. Okay, the last phase, you do whatever. Remember that in a early responding tissue, you do whatever, you give bombard it with ionizing radiation. But once you stop giving that ionizing radiation, restoration of this tissue is the rule of thumb. It will come back, it will repair, it will restore. Okay, that is the rule of thumb for acutely responding normal tissue. And that is called as a healing phase. Okay, and why does this happen? It happens because although the stem cells in the site of the radiation have been destroyed, but there will be some amount of residual stem cells which always be there. And this particular this thing is called as abortive division. Abortive divisions. Okay. Those stem cells, those residual stem cells will then start proliferating and then allow it to heal or restoration of the tissue. That is one mechanism. Second mechanism is basically because you have irradiated a small volume, the stem cells from the surrounding region, unirradiated region, will translocate themselves as a signal. When they get that signal of region, they will translocate from that surrounding place and go to that irradiated site and help the residual stem cells to further proliferate. Okay. Third mechanism, stem cells from a distant area. Okay. They will get the signal, they will come to that injured site and then help us residual stem cell to. That is how healing or restoration of the tissue happens in the early responding tissue. Okay, and the most important pathogenic event is this in early responding tissue, which I have highlighted with blue. This is the most clinically dominant phase also. All right, so since this is a clinically dominant phase, this particular phase I want to explain it. All right, so what is happening, and this has been explained by the target cell theory or stem cell hypothesis, what we call as. Okay. So when you are given, you know about the normal tissue homeostasis. Okay. So once I've given ionizing radiation, what will happen is the first thing is, I told you, what will happen? There will be asymmetric cell loss. Okay. What will happen is that instead of the stem cell dividing into one daughter differentiating cell and one stem cell, it will basically produce only two daughter cells which go into the transit cell compartment. Okay. This will then proliferate for a certain amount of time. And then what will happen is that those cells will go into the functional cells or post-mitotic cells or the mature cell compartment. And after a particular span of life, they will all be dead, they'll be lost. Okay. But since we don't have that many cells in this because of the depletion of the stem cells, what will happen is that the cell loss is more. The stem cells are not able to now replenish the functional compartment. We will have this particular phase of hypoplasia. All right. So the important thing is, in case of early responding tissue, is the overall turnover time from the production of that, of the differentiating cells and the stem cell to the cell loss is very important as far as the time course of early radiation reactions take place. Okay, that is one important thing. And this will be different for different tissues. Okay. The interval between the stem cell production and the cell loss is the one which will basically determine the time course of early reactions. Okay. Second point, the turnover time from the initial stem cell division to cell losses, tissue specific. So what I said, every tissue, the time interval will be different. So the time events of early reactions for different tissue, intestine it will be different, bone marrow it will be different, or testicular epithelium it will be different. Okay. And the third thing is the radiation tolerance of the tissue will depend upon the number of stem cells in the stem cell compartment and also the intrinsic radiosensitivity of the stem cell. Okay. And the last is called as I've said already, the high turnover tissues will actually have these early reactions. Okay. So these four important things you have to remember when we are talking about early responding tissues which show early reactions or acute reactions. What are the changes that happen in the early responding? So once you have basically these type of tissue systems have been irradiated or exposed to ionizing radiation, there is increased expression of different types of proteins like IL-1 alpha and other type of interleukins, tumor necrosis factor alpha, COX-2 and NOS, which are all the above three are produced by the macrophages, okay, while NOS is produced by that of the endothelial cells. What they do is that they have a paracrine effect. And as a result of this paracrine effect, they increase the expression of EGFR and other addition molecules like ICAM1. And what they basically act as transcription factors to modulate the DNA repair, and ultimately healing will happen. All right? Yeah.
The previous lecture, it was like radiation affects all three DNA, lipids, and proteins, and overall the functioning is getting affected. But to be able to increase protein expression, it's a bit counterintuitive that how is that happening?
Yeah, that's what I told you, no? The repair mechanisms in that of the tumor is defective. This is the repair mechanisms for the normal tissues is quite efficient, right? So that's why they undergo repair. Even if there is the protein is that there is a damage in the proteins or the lipids or the DNA in the normal tissues, the mechanisms are much more efficient. So I've told you that radiation actually causes the early reactions to these normal tissues, but we have to quantitate it. And this process is called as quantization. Okay. So let us go through this. Let us dissect this graph. We have got two important quantities. One, we will talk about severity of the acute reactions, and the other will talk about the incidence. Okay, do you know the difference? Severity and incidence. Severity can be a grade 1, grade 2, grade 3, grade 4 type of acute. Incidence is whether or not it has happened. What percentage of patients will develop that acute reaction, and we have to quantize, and it is dependent upon certain things, right? Let us talk about the severity. On the y-axis is the severity, and x-axis is the time in weeks after giving the radiation treatment. Okay, so let us take one particular reaction effect. This is the, let us say it is a grade 2 reaction, okay, of the earlier. Only one type, okay, but it will differ for a next type of grade severity of reaction. So let us take a grade 2 acute reaction.
Grade, you know.
So yeah, that's why. So let us, so one particular severity of reaction, okay.
Moderate reaction.
So let us take it as a moderate reaction.
Reaction means side effect.
Or toxicity or side effects or a complication, okay. So you are given ionizing radiation. The development of this reaction, the development or the initiation of this reaction is not dependent upon dose. You can very well see at low doses, at intermediate doses and high doses, it is more or less the same. They are all together. Are you seeing that? All the graphs are kissing each other, right? But the healing is different. At lower doses, the healing is faster as opposed to intermediate doses, as opposed to that of the very high doses. Healing is slower when the dose is much higher, but the initiation is not dependent upon that of the dose. Okay. The second thing is the incidence. So on the y-axis, it's the percentage of individuals who have been exposed to ionizing radiation developing a reaction, acute reaction or acute toxicity. And x-axis, it is the dose. Okay. Up to a certain dose, the incidence of acute reaction is nearly zero. After that, it slowly increases. Maybe it is only out of 100 patients, only 10 patients will get. But then there's a gallop happens. Between say 5 gray and maybe 10 gray around, you'll have an increase of from 5% to that of 50% will develop that reactions. So this is how acute reactions develop as far as severity is concerned, as far as incidence is concerned. Okay. So all this is dependent upon that of the stem cell inactivation. All right. So let us now summarize it. Early normal tissue reactions. So tissues which have got high proliferative capacity develop acute reactions or early complications or early toxicity. Okay. And the major mechanism is cell loss or cell depletion. And the imbalance between impaired cell production and the ongoing cell loss leads to tissue hypoplasia, which is the most clinically dominant phase of this particular event. And the symptoms in this tissue is based upon radiation-induced impairment of the cell production in the face of ongoing cell loss. And last but not the least, complete healing is the norm, is the rule of thumb in tissues which are highly proliferating, which are acutely reacting, which we call it as acutely reacting tissues. Okay. It's a bit difficult, but I would suggest that you go back and read again. Okay. They are typically seen in tissues which have an hierarchical architecture. Examples are the epidermis of the skin, intestine, bone marrow, and oral mucosa. And as I said, they are transient. Acute reactions are transient. But if they persist for a longer time, they lead to another type of reaction, what we call it as a consequential late reaction. Okay. And the latent time for the development of this clinical reaction is a function of tissue biology, but not a function of radiation dose. Okay. And the healing time for this acute reaction is a function of radiation dose. All right. So the severity and the time to healing is dependent upon the radiation dose. Another important thing for early normal tissues is that the reactions dependent upon the overall treatment time. Longer the treatment time, lesser will be the severity of that of the early reactions. Okay. Shorter the treatment time, more severe will be the reactions. Okay. And as I said, healing eventually occurs, which is because of all these reasons. The surviving stem cells in the irradiated volume, the migration of that of the stem cells from distant sites, for example, bone marrow. This happens with the bone marrow stem cells, and the stem cells which are migrating from that of the unirradiated area surrounding unirradiated. Okay. Any questions on early or normal? There will be definitely. It's not so easy. I would suggest that you go back and read it all. Okay. Then the late normal tissue reactions. So late normal tissue reactions are typically seen in tissues which have got a slow turnover, and in terms of microscale classification, it is seen in tissues which have got a F type of system or a flexible type of architecture. All right. Examples are the dermis of the skin, liver, thyroid, and the lung. But the pathogenesis is a bit complex in this. And the important thing about late normal tissue reactions is that it never heals. It never heals. That's the most important differential point. Let's see what happens. So again, it will go through various steps, right? So when you are giving a late normal tissue, for example, it's a lung. It's a late normal. Example is a lung. You are exposing it to ionizing radiation. It will produce something called as, this particular first step is called as induction. It will affect the parenchyma of the lung. It will affect the endothelium of the lung. It will affect the fibroblasts of the lung. And what will happen is that when it affects the endothelium, it can result in loss of capillaries. Okay. Okay. When it affects the fibroblast, it can lead to differentiation of the fibroblasts into fibrocytes, resulting in increased collagen production. Okay. And collagen deposition. This, along with the macrophages, and the reactive oxygen species and the reactive nitrogen species, which are basically produced from this endothelial cell, all together can lead to parenchymal damage, a step which is called as progression. Okay, and then when you're giving further radiation for this type of tissue, it will ultimately lead to manifestation wherein the architecture of the tissue is completely disrupted, and and as a result of this, there will be complete loss of function of the tissue. Okay, and last, healing never happens in a late responding normal tissues, especially when we talk about kidneys and lungs. All these and urinary bladder, these are late responding tissue. Once they have been damaged by the ionizing radiation, they don't heal. Okay, and why it is important for us is that in many cases we have to re-irradiate the tissue, tumors which are in close location to this tissue. Tumors which have developed back and are in close proximity to these tissues, which are late responding tissues, it is very difficult to re-irradiate because these tissues, because whatever re-irradiation will be giving, some amount of radiation will go to these tissues also, and they don't heal from the first dose or first effect of the radiation. So it's very difficult to re-irradiate tumors which are in close proximity to these late responding tissues. On the contrary, tumors, if they grow in close proximity to early responding tissues, we can easily irradiate because healing and restoration. So that is the clinical importance of understanding the pathogenesis of acutely responding tissues and late responding tissues. Just an example to show for lung, there are so many. Brain is also late responding tissue. Just I wanted to show one example. But when you're giving radiation treatment, what is happening? It is affecting not only the parenchyma but the capillaries also. And once the endothelium is basically exposed to this ionizing radiation within the brain, there is increased expression of all these protein molecules. There is decrease in the capillary density. As a result of this, there is loss of function. Apart from that, the fibroblasts produce TGF-beta. They undergo differentiation into fibrocytes, increased production of collagen, which is deposited into the lung parenchyma and results in loss of function. This is aided by macrophages and the production of ROS and also RNS, which also basically increase the which are nothing but inflammatory molecules, and they basically also result in loss of function. Okay. Last but not the least, ionizing radiations in the lung affect the type 2 pneumocytes, and these type 2 pneumocytes are very important for production of the surfactants, lung surfactants. Okay, and because of these type 2 pneumocytes are destroyed or damaged by the ionizing radiation, there is no production of the surfactant, and ultimately there is loss of function. This is how the late responding tissues basically are affected by ionizing radiation. So the loss of function is the thumb rule or the norm in late responding tissue, but restoration and healing is a thumb rule for early responding tissues. All right. So let us talk about the time course of events and the dose dependence of late radiation sequelae. So let us see. Again, we are quantizing it. Okay. So you have got a grade of response, which is let us talk about moderate severity of late reaction. Okay. Here. Okay. And this is the defined clinical response, clinical effect. That is a moderate grade of toxicity. And you are basically following up that particular tissue, late responding tissue, over a period of time after ionizing radiation. So this is after maybe let us say about 10 years, okay, because it's a late responding tissue. The other one is after 5 years, okay. So what is happening is that the grade of response increases with that of doses. This is the curve for the high dose, okay. So the initiation basically, as in case of higher doses, the effect goes on increasing, as opposed to that of lower doses. What is the difference? Here, the initiation of the late reaction is dependent upon the dose. In the early responding tissues, it is independent of the dose, but is dependent upon the type of tissue architecture. All right, that is the difference here. Okay, and let us So on the right side, this is the responders, that is the incidence of late reaction. Okay. So y-axis, there's the percentage of patients who will develop that late reactions. And in the x-axis, it is the dose. Okay. So, and they are being followed up over a period of time. When you're giving less dose, say this is about 30 gray radiation, and this is about the 70 gray radiation. So when you're giving lesser dose, the time to develop that late reaction is delayed, as opposed to giving higher doses, which is 70 gray. The time to develop that reaction is shortened, late reaction is shortened. So the incidence or the percentage of patients developing late reactions will be higher at shorter time interval following higher doses of radiation, as opposed to patients who get lower doses of radiation. I want you to go back, read, and then you can please ask me questions on this. So it's a very, very difficult graph, but at least you will get to understand the difference from this graph, the difference between early reactions and late reactions as far as radiation dose and the time is concerned. Okay. So late radiation effects are progressive in nature. It doesn't heal. And the latent time to late clinical effect is dose dependent, radiation dose dependent. Higher doses, faster. You will see higher increasing severity, okay, as opposed to that the lower doses. Okay. Then progression rate of late radiation effect is also dose dependent. More the dose, faster will be the progression of that thing, late effects. And for a given radiation dose, the incidence of late reaction increases with the follow-up time. Higher the dose, faster will be the late reaction in larger number of patients. Okay. Coming to summary of late normal tissue reaction. So there is dose-dependent shortening of the latent time to clinical manifestation of this effect, and they are inversely dependent upon the dose. More the dose, there will high. It's bit counterintuitive. Okay, this particular statement. It is inversely more the dose, greater will be the severity of that of the late reaction. Rate at which the severity progresses, again inversely dependent on the dose. Higher the dose, faster will be the development of the late reaction. And it is, apart from the dose, it is also dependent upon the dose per fraction. Higher the dose per fraction, greater will be the late effects. Okay, so it is radiation fractionation sensitive. These tissues are highly fractionation sensitive. Okay. The third type of reaction is called as the consequential late reaction. And basically what happens is that if an early reaction, acute reaction, basically progresses over a period of time, that leads to something what we call it as a consequential late reaction. And it is basically influenced by the severity of the acute reaction and the duration of that of the acute reaction. All right. Although the name is consequential late effects, it is more synonymous with that of the early effects. The markers of early effects are surrogate markers for that of the consequential late effects. I'll talk about that in my next few slides. Okay. Late reaction, as I've said, late reactions happen in particular types of tissues. Early reactions happen in another particular type of tissues. Consequential late reactions also happen only a few types of tissues. So not all tissues will show consequential late reactions. Okay. Consequential late reactions can be modulated by altering the severity or duration of the early effects. So markers for early effect are surrogate markers for consequential late effects. Okay. For example, if you're changing the fractionation, intensive fractionation protocols like accelerated radiotherapy can increase the severity and the duration of oral mucositis, which can lead to that of consequential late. This is one of the examples of it. Apart from this, apart from the severity and the duration, additional trauma can also basically progress to consequential late effects. All right. So this is how it happens. When you're giving ionizing radiation to a particular type of tissue which shows consequential late reaction, which will affect the epithelium. Okay. And what will happen is that there will be disruption in that the proliferating cells within the turnover tissues, This will also damage the endothelium, the fibroblast, and the parenchyma. Okay, which all happen in the early effects. But when this early effects, all these things continue for a longer time, when the severity is also higher, with the addition of additional trauma, this can lead to what we call it as late radiation or consequential late radiation. All right. So as I said, consequential late reactions usually doesn't happen in all type of tissues. So the consequential late reaction more common in urinary bladder and intestine, less common in oral mucosa and lung, even lesser in case of the skin. Okay. So these are the tissues which show consequential late reaction. And as I said before, the markers for early reactions are surrogate for consequential late reactions. Although the name is consequential late reaction, it is a misnomer. It doesn't basically, the markers for late reaction are not surrogate for consequential late reaction. So I can just show you that. Okay. So if you are basically modifying a vascular function, so the acute, there is no effect on that of the vascular in the acute reaction. Similarly, consequential late reactions also are not affected, but late reactions are basically affected when you're modifying the vascular functions. Similar way, fractionation. Acute reaction or acutely reacting tissues they show low radiation fractionation sensitivity. Similarly, tissues which show consequential late reaction are also, they show low fractionation sensitivity, but late reacting tissues show marked fractionation sensitivity. Okay. In the same time, if you increase the overall treatment time, the acute reaction, the severity of the acute reaction also changes. Similarly, the consequential late reactions also changes severity, but minor or no changes happen in that of the late responding tissues. Okay. And if you are able to modulate the acute effects, it definitely modulates the acute reactions. Okay. But because of modulation of that of the acute effects, you are able to also modulate the consequential late effects. But this doesn't happen in that of the late reacting tissues. Okay. So that's why the consequential late effects are basically a surrogate of or an extension of that of the acute effects rather than a surrogate of the late reaction. Okay. The CLA is a misnomer basically. So what are the take-home messages for these? Latent time, again, so that you just remember that latent time of early effect is independent of dose, but dependent upon the tissue biology. Severity and duration are dose dependent. Time to healing in case of early effect is dependent upon the dose. It is clinical dominant phase is tissue hypoplasia and happens in highly proliferating tissues or tissues with high cell turnover. Late, they are observed after months to years, usually after 90 days, 3 months. Okay. They are progressive, irreversible. Healing never happens in late responding tissues. Okay. Examples are basically lung, liver, kidneys, and late effects are based on the interactive response of the parenchymal cells, the vascular endothelium cells, and the fibroblasts. The pathogenesis, as I told you before, is complex, and it's because of the complex interaction between these. Right. The latent time to late effect is dependent upon the dose, and the rate of progression of late reaction is also dose dependent. The severity of late reaction is dependent upon the times. Okay. So given the time, more time, so again the severity will be less, similar to that of the acute reactions. Right. So that is the first part of normal tissue pathogenesis. So how much time do I have?
How much?
Another maybe 15 slides or 15.
I think.
Or do we do it in the next one?
Look at their place. They are all best thing. I think they all tired. क्या अभी हम लोग बाद में भी तो पढ़ाना होगा ना? I also want to speak for a few minutes.
I just really want them to assimilate this early reaction, normal reaction.
ठीक है, अगर इसी से relevant है तो बताओ.
All right, I will just see. And fractionation is also there, sir.
So fractionation is a full this
बाद में अलग. Okay, I'll just finish with
yeah, this one. So because then also exam will be there.
Yeah. So only 10 slides. Okay. So-
पकड़ लिया कुछ भी कर सकता है इसके साथ
So what are the time factors in normal tissue response to radiation? Again, the time factors basically correlate with that of the recovery of the tissue, repopulation, regeneration, and restoration. And restoration can be long-term restoration also from the hours of radiobiology. Again, they are being repeated. Okay. So how important it is, you can see that they are the pillars for radiation biology. And this is not the first time I'm going to repeat it. It will be repeated more and more. Okay. So what are the time factors? So we divide the time factors into three different types. Okay. One is the short scale from minutes to hours. Whenever we are giving a single fraction of radiation treatment, what happens is that when we are giving a single fraction of radiation, there is incomplete recovery of radiation happening from SLD. And as a result of this, it reduces the radiation tolerance to late signal. Okay. So intermediate time scale is from days to weeks. This happens during when we are dividing the radiation doses over a period of time, which is called as fractionated radiotherapy. Here, tissue regeneration is happening and they modulate the radiation tolerance to early reactions. Okay. And the last is time scale of from months to years. There is long-term restoration is happening in the tissues and which make them highly resistant. So that you can give re-irradiation in these particular tissues. Okay. So long-term progression of the damage even at subclinical level in some of the tissues will decrease the re-irradiation tolerance. That means in these tissues where restoration is not happening, there is always a progression of the damage which is happening. As a result, you cannot re-irradiate these tissues. Okay. So what are the changes which are happening in this normal tissue tolerance? Okay. Let us take, for example, the spinal cord, which is a late responding tissue, and oral mucosa, which is an early responding tissue. Okay. And this is the interval of time, what is happening. So in the early responding tissues, what is the recovery kinetics? It is more or less flat in case of early responding tissues. Slightly, it is there for late responding tissues. There is some recovery is happening in the spinal cord. Okay. But when you see in the early responding tissue, which is the oral mucosa, between 1 to 10 or 20 days, there is good amount of restoration is happening. So because of repopulation. And then spinal cord is one such exception of late responding tissue, which shows long-term restoration. You can very well see how restoration is happening. In my previous listing, I used to say that long-term restoration never happens in late responding, but spinal cord is the only exception. And here is the case where you can give re-irradiation. Okay.
You figured out
the spinal cord restoration, it is again based upon the stem cell hypothesis. There is a long-term restoration is because in the spinal cord is the only structure where the abortive divisions is quite high. They allow for the restoration of this long-term restoration of this. So repopulation is at the cellular level, regeneration is at the tissue level. So regeneration response of early reacting tissues to fractionated RT results in increase in radiation tolerance with increasing overall treatment time. So more the treatment time, so better will be the radiation tolerance of the early responding tissues. Okay. Now there are certain clinical observations. So let us see what happens. Dr. Sarin talked about one particular type of regimen, which is called as the continuous hyperfractionated accelerated radiation treatment, CHART. Okay. What is happening here is that when you are giving, CHART is actually very good for tumors because you are trying to reduce the overall treatment time. Before that, think of accelerated repopulation picks up, okay? But it is detrimental for that, the normal tissues. It's very easily, you can see that. So this is conventional fractionation where we are giving total of 66 gray, 33 fractions, which is 2 gray per fraction. CHART, which is 54 gray, 36 fractions in 12 days. In 12 days, we are giving 2 to 3 fractions per day. What is happening is that these patients who are getting this CHART regimen have got 70% of patients will have grade 3 mucositis, severe mucositis, which is grade 3 mucositis. Okay, and that is because it is not allowing the repopulation to happen of the normal cells. Okay, let us see the other extreme. Okay, there is which is called as the split dose radiation. What is happening here is that when we give radiation initially, say 12 days of radiation treatment, we see about 90% of patients will have grade 3 mucositis. All right. After 12 days, we give a gap of 2 weeks. What is happening here is that accelerated repopulation is now kicked in for the normal cells. And then when you give radiation treatment, we don't see any radiation mucositis. It is because of the accelerated repopulation of that normal cells in the area which has shown the toxicity, right? So these are two clinical observations to basically corroborate all the biological findings which we have been talking about. All right. So how important is repopulation? You can see that. And when I'm talking about repopulation and I'm talking about healing, healing is the norm in early responding tissues. So then we have to put some quantity to that healing. So what is that? Is that when I'm giving radiation to a human oral mucosa, about 0.5 to 1, 2 gray per, when I'm giving 2 gray of radiation, at least 0.5 to 1 gray of radiation is being basically restored, okay, in terms of the radiation dose. All right. So I mean to say that when I'm giving radiation treatment, the normal tissue tries to at least completely restore itself. It may not be possible, but at least it tries to compensate for the ionizing radiation damage which we have caused with the ionizing radiation. So if I've given 2 gray, it will try to compensate for that 2 gray also. So which will nullify that the effect of that 2 gray. But it never happens always like that. So at least 0.5 to 1 gray will be nullified for that 2 gray. Okay. So how does this repopulation is regulated? And there are experimental models based upon the target cell hypothesis or stem cell concept. So what is happening here is that when I'm giving ionizing radiation, there is depletion of that of the stem cells. And that depletion of that of the stem cells is because of that of the asymmetric loss. And I told you what is asymmetric loss. You know that, huh? And so this asymmetric loss, and then again, there will be tissue hypoplasia that will lead to, as a compensatory mechanism, for stem cell acceleration. Both asymmetric loss and stem cell acceleration will compensate for that of the radiation dose as far as repair is concerned or repopulation is concerned. The second thing is that the tissue hypoplasia will also signal abortive division in the damaged cells. Abortive division is when the residual damaged stem cells are trying to divide, and that will compensate for that of the cell loss. And these are the two mechanisms, both by dose compensation and by cell loss compensation of cell loss, repopulation is happening in these tissues, which ultimately lead to regeneration or long-term restoration of tissues. All right, so take-home message as far as repopulation is concerned. So the radiation tolerance of early responding normal tissue increases with that of increasing overall time that you can understand. And as I said, repopulation is very much tissue specific. For head and neck tumors, usually it starts 3 weeks after starting the ionizing radiation. And this repopulation is called as accelerated repopulation. Okay. And how come repopulation is happening? It is because of the abortive divisions of the doomed or the residual stem cells which are present in the area of the irradiation, and also from the surrounding stem cells which is happening. Okay. So tissue hypoplasia results in stem cell acceleration in abortive divisions, result leading to repopulation, and stem cell depletion leads to asymmetric loss of cell, which again helps in repopulation. So these are the two methods by which repopulation and long-term restoration happens. Thank you. So fractionation is another question topic where it's not only about fractionation, it's about clinical fractionation, where I'll be talking more about altered fractionation, which is more clinically relevant. Any questions?
Now you can ask questions while I'm on this. Consequential late reaction.
Consequential?
Late reaction.
So it's see the oral mucositis which is there, okay, and which is there for say less than 90 days. But if it persists beyond that, you can, that is what is called as a consequential late reaction. It can also happen in the urothelium also, bladder urothelium.
No. Any other questions? So, I'll maybe while I'm just putting some feedback from students, I'll tell you about the exam module and subsequent courses. Questions, feedback. You think we should continue doing these weekend courses? Okay. Okay. Well, I'm sure we will find time and energy to do these courses if you all want it, because ultimately, let me, where is this cheap? So I have prepared something. What will be the Gmail was working. So, thank you all of you for really still not become a pro. Okay. So, a TMC certificate of successfully completing a module will be given to registered participants who pass the online MCQ based exam for that module. Okay, it could be only physics or radiology, so that is there. So those who have registered. So it will be the 3 modules for the MCQs, which is one is medical physics, other is genetics, other is radiobiology. So who may give the exam or who has to give the exam? For some it is optional, others it is not optional. So all the TMC MD students have no choice, they have to give all the 3 exams. Okay, which is physics, radiobiology, and cancer genetics. Same way TMC HBNI health science PhD students, sorry, I did not write PhD. They have no choice, they have to give module B and C. This is part of their core course. Okay. Now, there are some people who have done their MD or their research fellows in TMC, they're working, they have attended, some have registered. So for them, it is optional. If they feel like giving an exam, they can give either none of the module or 1 or 2 or 3, whichever they want to do. Okay. And for all the other participants who have volunteered to spend so much of time, it is entirely optional. You may or may not wish to appear for 1 module, 2 modules, all the modules, whatever you want. Okay, it is purely optional and there is no sort of obligation. Sometimes we feel, okay. So how it will be done? So it will be an online MCQ based exam. We have to figure out the modalities, otherwise conducting online MCQ exams through some other agency is very expensive, and this course is free for everyone. You want to democratize education and training. So it will be either through their polls on Zoom or Google Form, because you get individual responses, when you're seeing that. So how we will do it, because it could be an open book exam also, but when you start reading it, the questions will be such that it will take you that much time to go through this thing. Even if you have chat GPT or AI chat GPT, it will take time. So the challenge is for us to frame the questions in such a way You can't just quickly, Google it and get the answer. Okay. So like if you say, what is the half-life of this? You can immediately search and in 10 seconds or 30 seconds. So
My questions are already prepared.
okay. So the questions would be such that in whatever 30 seconds or something else, only if you have understood that concept and there'll be more concept based or something you'll be able to answer. Otherwise, you will not be able to answer in 30 seconds. Of course, if you read for 15-20 minutes, you can answer, but there won't be that much of time for that. Okay. So and what did we cover? Whatever we have covered in the course, and like yesterday and today also some things I did not tell. I said you have to go and read. Jayant was very generous. He told you all everything. So I have realized that if something is given to you on a platter and which you have not understood, you have just read it, you think, oh, I know it. And then you, without making a real effort to understand and actually understand, So some of the things. So what was there? The YouTube video is there. Videos are there. Everything you'll have to all, go back and this thing. And I'm very happy. Yesterday's physics YouTube video already 2,800 people have viewed. I am sure today genetics and radiobiology also equal number will see in the next 1 or 2 days. Okay. So you read that. And the exam date also, I thought it is better we finalize. So it is Saturday, 6th of July, which is just over 4 weeks. Time also I've kept keeping in mind that your 5 p.m. to 6:15, if most of those who are in the hospital, they would have finished the work by 5 p.m. and you wouldn't have started partying as yet. Okay. So after the exam, that day you all can go for a party. Okay. You have prepared 1 week or so you will prepare. So that day is the exam. And there'll be 25 minutes for each module. And we've kept it, I was thinking our students, you can keep it, offline and for others, but I think everyone should be on the same this thing. Whatever advantage or disadvantage of online or offline, everyone should have the same. Okay. And for this, we will make a new Google form. And what happened in the previous Google form, many people had, because they had to write their email, many people had typed their email incorrectly. So many of the mails that we sent, they bounced back. And then it also allowed people to register again. They were, oh, I have not registered. So almost 1,700 people who registered, of them around 80 or 90 had registered twice. Okay. So I had to manually curate. It takes a lot of time. So this time we will, I am doing everything, so making Google forms, sending mails, everything. So I will make it in such a way that this doesn't happen. So we will make, and in that there'll be an option. Again, we will collect your individual details now, both sort of structured way we will do it. And because later on certificates have to be given, your proper full name of your hospital or institution where you work, which course you are doing, this thing, others. Many people are in others. Others
keeping 50% as minimum.
Yeah.
I think it should be at 70%.
No, so then they will get gold medal. No, minimum. So our depend how easy or difficult the exam is. Okay. So if many of the exams, if you score 70-80%, it means you are truly very brilliant. So average person. So at least half of what they are expected to know. Then these are trainees. Over a period of time, they will learn more. But at least this much they know. So, and exams will be only for those who have registered again for the MCQ exam, just because they have participated. So that Google form, if some of you have registered for this course and you are watching it on YouTube and have not received the Google form, then you will have to send a mail and then you will get it in the new. And those who cannot get minimum 50% marks, or those for some reason on 6th of July, they're getting married or something else, or the sister is getting married or something, anything could happen. They cannot come, they have to give a valid reason in advance that I am unable to appear on 6th of July. And for these people, after a gap of another 4 weeks, we will keep another exam. But it should be for valid reason, not just 3 days before the exam, you thought, oh, I haven't sort of prepared or something else. I'd gone to Goa, not for that reason. Okay. Give a valid reason. We will believe whatever you say, but please give a valid. Okay. And for the top 5% scorers in each module, we will have an online interview for them to give a meritorious award. I don't know what it would be. I don't know what it will be, but I think a special that we'll see whatever it is. So what we'll give, because it is only online and I really don't have much experience. So if suppose someone has a very good AI assistant, But someone who is doing that, the moment you talk to that person for a few minutes, you come to know, well, someone has scored 90%. And when you talk to them for 5 minutes, you know that, well, how on earth this person scored. That we have seen many times in our PhD interview exam. People have scored very high All India rank of A or 50 or 70 in CSIR gate. But when you talk to them, you are absolutely lost. So I mean, I myself was wondering how come they scored so high, and when you talk to them, they have so this thing. So we will do that. Of course, you'll get a certificate if you are more than 50%. But if you are really that good, you get a special award. And I mean, it's nothing great, but I'm sure you will preserve it. That when I was doing my MD or a PhD, there was 1,800 participants, and in my module, I was in the top 5%. And someone who gets the first in that, I don't know what they will get. I can't give a gold medal. I don't have the authority to, but some special award will be. Maybe we'll party. Okay, so the next course date and topics will be announced in about 4 weeks' time. So now we will make a full structure of this thing, because the entire syllabus we are covering, we will do that. And I would like to thank you, each one of you, on my personal behalf, on behalf of my course coordinators, Dr. Jayan, Dr. Amit, and Tata Memorial Center, for the overwhelming response and your enthusiastic participation. The trust you had on us that we will teach you and train you. And I hope we have done our job. And please give your candid feedback. And now, because of email, I can't sort of keep tab of so many mails and they come in the thread. So on the Google form that you will share, there will also be a section which is a free form. You can give specific candid feedback. So some things I know we should have done that, but in the interest of time and to cover everything, it was just not possible. And it's a very diverse background. So, I mean, I have allowed all kinds of people. Anyone who is interested or want to learn something, I have no business to say that, no, you may be interested, but you should not learn. Okay. We have someone who is an MBBS and then doing MTech in IIT. Okay. We have people who have done PhD, people who are doing PhD in different diverse sciences, people who want to get into PhD. And before that, they want to understand what they're getting into. Okay. So, in India, everything is so regimentalized, whereas in the West, you see people are free thinking. They will explore different things and then they make up their mind what they need to do. Whereas here, everyone is taught what you should do, what you should not do, whether you're fit for this. I mean, for training, anyone wants to do that. In courses also, people can choose so many courses, mix and match. Here, you don't have a choice. Right from when our 10th standard to even when we are doing PhD, we have so little choice to what we are interested in. So everyone is this thing, but we hope this is a new method of education and training. And I think that's how the future is. When you all become teachers, please be better than us. Okay, so thank you. So we will end the meeting, stop share and end the meeting.